இல்ல எனக்கு கேக்குதா? நான் Hello, Sangeeta ma'am, can you hear me? சார் குட் மார்னிங். நல்லா இருக்கேன். நீங்க எப்படி இருக்கீங்க சார்? வந்துருச்சு. ஒரு வழியா கொண்டாந்துட்டேன். ஒரே ஒரு நிமிஷம். ஒரு நிமிஷம். வந்துருச்சு. ஆ ஆமா. அன்னைக்கே சொன்னாங்க. பொறந்தனே சொன்னாங்க. ஆமா. அடுத்த செகண்ட் சொன்னாங்க. ஆ. அப்ப அடடடட. சொல்லிட்டாங்க. கிராண்ட் டாட்டர் இல்ல? பேர் என்னங்க சார்? பரணி நட்சத்திரமா? அதான் நான் பரணி. அப்படியா? செம்ம. சூப்பர். ஆமா. சூப்பர் ஸ்டார். ரொம்ப நல்லா இருக்கான். நல்லா இருக்கிறாரு. வர்றேன்னு சொல்லியிருக்காங்க. இப்ப இது குவாரண்டைன் இல்லைங்க சார்? நல்லா முடிச்சிட்டு வேக்சின் போட்டாங்க. பண்ணல. அவன் தான் வந்து எதை பார்த்தாலும் இது வேண்டாம், அது வேண்டாம், இப்படி வேண்டாம், அப்படி வேண்டாம்னு இருக்கிறான். அதை பத்தியே பேச இஷ்டப்படல. சரி, அதனால ஹவுஸ் சர்ஜன் ஹவுஸ் சர்ஜன் முடிக்க போறோம். இல்லங்க சார், இங்க முகப்பேர்ல தான் போயிட்டு வந்துட்டு இருக்கேன். இப்ப அடிஷனல் சார்ஜஸ் அஸ் வைஸ் பிரின்சிபல். ஹெச்ஓடி அண்ட் கார்ல தான். டிரைவர் வச்சு காலையில 7:15 புறப்பட்டா 8 o'clock வந்துடுறேன். இங்க 4 o'clock முடியுது. 4:30 வரல ஒரு Sangeeta,
Ma'am,
shall I start?
ma'am, HOD ma'am will give the intro, ma'am. Afterwards, my introduction, and we will introduce you, ma'am.
Okay. முத்து, இந்த மொபைல் வெளியில வச்சுக்கோங்க. ஏன்னா 2 இது பேக்கப்காக லாகின் ஆயிருக்கேன். பக்கத்துலயே இருந்தா இது எக்கோ அடிக்கும். இது அப்படியே லாகின் ஆகி இருக்கட்டும். சரி. ஒரு 9:30 வரல யாரையும் ரூம் கொள்ள விட வேண்டாம். தபால் எதுவும் கொடுத்தாங்கன்னா வெளியில வாங்கிக்கலாம்.
A very cheerful morning and a great day to all enthusiastic delegates gathered here. We, on behalf of ACS Medical College and Hospital, IATP, are proud to welcome you all to the second day of this grand event, E-TROPACON 2020, with the theme of emerging and re-emerging parasitic zoonoses. Having completed the first day of the session successfully, today on the agenda, we have the few guest lectures and symposiums in the morning with paper and poster presentations from young budding researchers all over the country in the known. Welcome you, sir, Dr. Vinod Khanna, Dr. Leela, Dr. Kavita. Good morning to everyone. Shall we go on to the scientific session? Are you all ready?
Yes, madam.
Okay, good.
Yes, madam. Good morning, madam.
Good morning, Kavita.
Morning.
Without any further ado, introducing the learned chairpersons for the morning, Dr. Kavita, Professor and Head, Department of Microbiology, Government Kumaramangalam Medical College, Salem, and Dr. Leela, Professor and Head, Department of Microbiology, SRM Medical College, Katangulathur, Chennai. Over to you, madam.
Good morning, all. It's a great pleasure to be a part of this National Conference Tropacon 2020, and I have the pleasure in introducing the first speaker, Dr. Vinay Khanna. For the first scientific session of the day, we have Dr. Vinay Khanna with us. He is currently an Associate Professor of Microbiology in Kasturiba Medical College, Manipal Academy of Higher Education, Karnataka. He completed his MBBS, MD from the same college and graduated his MBA from Sikkim Manipal University and his senior research fellowship from PGI Medical Education and Research Center, Chandigarh. He is currently faculty in charge of Parasitology Division and Division for Medical Entomology, life member of Indian Association of Medical Microbiology and Indian Academy of Tropical Parasitology, and he is a member of Board of Studies of the prestigious university, the Manipal University. He is actively involved in research on free living amoebae, JIT infections with parasites in school children and community in Manipal and Udupi area. He is also involved with vector research with special reference to malaria and filaria. He has 13 years of teaching experience in medical microbiology. Dr. Vinay Khanna is a reviewer of various international and national journals and has over 42 publications in national and international index journals, 30 plus presentations in various national and international conferences with 20 plus projects in hand. We now request our distinguished speaker to deliver his lecture on human angiostrongylliasis, which was discovered in 1935 and which is an emerging parasite producing epidemiology also. We would like to hear from you your experiences and knowledge about this emerging parasite, sir. Over to you, sir. Please do come.
Dr. Vinay, unmute yourself, please. Dr. Vinay, you are still on mute. Unmute yourself.
Sir, at the bottom of the screen, you can see that mic option. No, you just unmute that. Just click that.
Yes, yes. Are you able to hear me now?
Yes, sir.
No, it is
Yes,
fine.
sir. Yes, sir. We are able to hear you. We are pleased to hear you. Thank you. Thank you. Please join us. Sir.
Okay. Now are you able to hear me? Is it fine?
Yes, sir. Kindly
Sir, you are
proceed,
quite
sir.
audible and clear.
Yes, sir. Yes, sir.
Okay. All right. All right. Okay. Thank you so much. Thank you for the kind introduction, Dr. Leela. Now the thing is that just a second, I need to share my screen. I'm not good at Google Meet. For the first time, I'm using it. Just a second. All right. Okay. So first of all, before my presentation, I would like to thank Dr. Geeta Lakshmi for giving me this opportunity to present for this presentation in front of the esteemed audience. Secondly, I would like to thank Dr. Paricha, who is very kind to me and always shown trust in me. So I'm really thankful to you, sir. Thank you very much. And obviously, congratulations to Dr. Subha Lakshmi and her entire team for conducting these sessions very smoothly. Now coming to my presentation, that is human angiostrongylliasis. So here in this picture, you can see a human brain, human intestines. The story revolves around the brain and the intestines of the humans. And the angiostrongylliasis parasite here, angio means blood vessels. So obviously these parasites will be living in the blood vessels. The second and thing you will see here is a rat. Okay, so this is a rat lungworm which is present, and you can see a very cute snail. So which will be acting as a intermediate host in this parasite. So here you can see news articles where the patient has described their symptoms which are very painful, paresthesias which occur in the patients, and you can see the cases has been reported from Hawaii, and the cases has been reported from the rats, slugs, and all these has been described in these news articles. So the story revolve around these parasite, which is a zoonotic parasite involving rats, snails, mollusks, obviously humans. Now, coming to a case report which has been published in a very esteemed journal, Lancet Infectious Diseases. A 20-year-old US Marine posted in Japan presented with a headache, a mild fever for 3 days. He had no myotonia or convulsions. He has given painkillers and sent back, but the patient came back after 2 days with increased severity of the symptoms with numbness in arms. Lumbar puncture done and the CSF showed eosinophil count of 34%, lymphocytes of 60%, and monocytes of 6%. The patient was diagnosed with a case of viral meningitis and started on antiviral therapy of acyclovir and dexamethasone for 6 days. After treatment, the patient headache was relieved and the temperature came back to normal, but his numbness of arms persisted. The patient was US citizen, so he was flown back to US, but his symptoms of headache, numbness remained persistent, but he had a urinary retention and constipation, which was a new symptoms. So the diagnosis of radiculomyelitis was made and lumbar puncture was done, and which showed high eosinophil count of 30%. PCR was done to find out the common causes such as herpes, CNS TB, cytomegaloviruses, but these tests were negative in this case. So then reassessment of the dietary history was taken from the patient, and the patient revealed that he has taken crabs, exotic dishes like frogs, crabs, snails in Japan. So PCR was done to rule out the rat lungworm. It came positive, and diagnosis of eosinophilic meningitis was made in this case. So patient was started on dexamethasone therapy and parental pymioate. There was some improvement in neurological symptoms, but symptoms of urinary retention and paresthesias persisted, and these paresthesias are quite painful in this case. This was because he was diagnosed late in the course of illness. So after seeing this, you can understand that this patient had a case of eosinophilic meningitis, which is caused by Angiostrongylosis cantonensis. So as Madam was telling that it was first described in China by Chen in 1935 from rats, and it was identified in the CSF of the patient with eosinophilic meningitis by Nomura and Lim in Taiwan in 1944. In 1955, Macrae and Sanders identified the life cycle of the worm in rats and defining snails and slugs as intermediate host. And then there was outbreak of eosinophilic meningitis in Tahiti and Hawaii, where the epidemiological survey was conducted by Rosen, Legertz, and Boris. And they hypothesized that this eosinophilic meningitis happened because of eating fishes. But then there was one more scientist named Alicata. He observed that fish is commonly consumed in those areas in Hawaii. So definitely it is not because of eating fish. And then he observed that the patient who has presented with meningitis had a history of eating raw snails and prawn weeks before the presentation. So Angiostrongylosis is a parasitic nematode that can cause severe gastrointestinal and CNS disease in human, and it depends on the species. So there are around 21 species which has been studied till now. This parasite was earlier known as Strongylus vesorum, which was termed by Balliet in 1866. In 1986, a study was done to on the systematics of Angiostrongylos, and 6 different genera based on the bursal ray morphology of the parasite was created. And these were named as Angiostrongylos, Parastrongylus, Angiocalus, Rotentocoelus, etc. Now, out of all these species, the main species which are causing human angiostrongyliosis are Angiostrongylos cantonensis and Angiostrongylos costarricensis. Now, this is a geographical distribution of the parasite across the world, And you can see the parasite has been reported from Africa, Caribbean, Australia, Hawaii, and southern United States as well. Some of the reports has been also seen in India, but most commonly the outbreaks has happened in mainland China, America, and some cases has been reported from Brazil. But outbreaks are quite common in Hawaii. So there are around 3,000 cases has been reported worldwide of eosinophilic meningitis, and around 27 cases of abdominal angiospondylitis has been reported worldwide. And there are around 116 case series patients were reported in the literature till now. And why this has happened? Why this disease, which is a zoonotic disease present in the rats, has affected the humans worldwide? It is because of globalization, basically. And due to globalization, everything is available, everywhere. Rats and snails are no exceptions. So snails actually if they are present in the endemic area, they have been found in the new area, or they have been transferred to the new areas because of increased travel. And there are stowaway of ships which carries the rat from different areas, also are one of the important cause of transmission of disease worldwide, globally. So this is a picture of Angiospondylis cantinosus. Now here the nematode is a roundworm with 3 outer protective collagen layers. You can see the female with a barber pole, and you can see a spiral inside the female body. And this spiral occurs because of urinary tubules which are spiraling around the intestines of the worm. The males are smaller in this case, and the worm is actually quite small. It is around 15 to 25 millimeter in size, and the width is also quite small. It's 0.25 to 3 millimeter. And if you see the characteristic feature, the posterior end in male consists of bursa and spicules. Okay. And in case of females, you will see the posterior end is conical and slightly curved. The characteristic feature is the presence of bursa. Okay. So hosts in this parasite are multiple rat species, including black rats, brown rats, and cotton rats. Can somebody unmute yourself? Please unmute yourself. Thank you. And the other hosts are gastropods, snails, and slugs, which act as the intermediate host. And there are numerous species of snails which are being involved in the transmission of the disease. And these are Biophilarias, Bulinas, Lamias, Pomaceae, etc. Humans are always accidental host in this case. So the transmission occur between rodents and slugs. So the parasite, the adult worms, lives in the pulmonary artery of the rats, where they lay eggs. And these eggs are cupped out and swallowed back by the rat and enter into the intestines. And from the intestines, as you can see in this picture, they come out in the feces. So eggs either will be ingested by the snails or the slugs outside, or they will hatch into larvae, and these larvae penetrate the snail. Inside the snail, inside these mollusks, these get transformed into third stage larvae. And then again, if the rat happen to eat these infected larvae, these will enter into the brain parenchyma of the rats, where they develop and then migrate from the circulatory system into the lungs. And inside the pulmonary artery of the lungs, they again develop into adult worms. This is a life cycle between rodents and slugs. But then how humans get the infection? Now, humans get the infection by eating contaminated vegetables, and these contamination can happen because of the larva which is being present in the snails or slugs. Okay, and then there are certain paratenic hosts such as frogs, shrimps, crabs, etc., which has a tendency to eat these slugs or these snails. And if humans eat these improperly cooked paratenic hosts, they can also get the infection. And these snails, slugs can also contaminate the beverages which are not sealed or kept properly. So this is how humans get the infections. So now what happened in the humans? So in humans, eosinophilic meningitis is one of the important diagnoses. So what is eosinophilic meningitis? It is defined as when there is more than 10 eosinophils are present per microliter of CSF, or 10% of eosinophils in total leukocyte counts. So the larva migrates in the brain parenchyma, but rarely it can affect the lungs or the eyes. So there are certain tracts or microcavities which are formed in the brain can cause the structural damages. But there are no cavities as such, or focal lesions are not formed in case of cerebral angiostrongyliasis. The focal lesions are absent. So this is an important thing because the focal lesions are present in other parasites which affect the CNS are like toxoplasma, neurocysticercosis, or paragonimiasis, schistosomiasis. So the focal lesions are present in those cases, or nanthostomiasis in that case. But here in this case, there are no focal lesions are present. Only microcavities and tracts are seen. Or sometimes, if the parasite dies in the brain, then granuloma formation and hemorrhage can occur around the parasites, which can cause the symptoms. And sometimes the cerebral nerves can also get affected. The most common cerebral nerve which can get affected in this case is abducens and facial nerve. But the spinal cord is also seen to get affected. That's why the painful paresthesias are quite common in these cases. So I have shown you the news article initially in the slide 1, where you have seen the patient has, describing to the reporters that they have very, very painful paresthesia, and which continued for many, many months, which is the main cause of morbidity in these cases. So how to diagnose neural angiostrongyliasis? So the gold standard is microscopy. The conclusive diagnosis you can make if you are able to find out the larvae in the CSF. But as I told that these larvae dies in the brain, so rarely you find these in the CSF or in the anterior eye chamber. In this picture, you can see eosinophilic granulocytes, which has been stained with the May Grunwald Jimsa stain. So suppose if microscopy is not available, then you can go for serological tests. So serological tests such as ELISA or Western blot test can also be used. There are total 10 antigen which has been detected for angiostrongyliasis. Out of that, 5 are very, very important, which are very, diagnostic and specific for angiostrongyliasis. So these 5 antigens bands are 29 kilodalton, 31 kilodalton, 55, 85, and 200 kilodalton, which are very, very specific for diagnosing of angiostrongyliasis. You can culture the parasite. The eggs can also be cultured in the reference laboratory. The reference laboratory is the ILO University in Hawaii. So there you can, there are these are good labs which can basically do a lot of research on angiostrongyliasis, and they are using various cultivation methods for culturing these parasites. So cultivation of eggs can be done in NCTC 109 medium, which is supplemented with equal volume of rat, horse, fetal calf, or bovine serum, and incubated at 37 degrees Celsius. The eggs, the larva will hatch after 8 days of incubation. You can also culture third stage of larva in RPMI 1640 supplement media, which is supplemented with calf serum. The fourth stage larva can also be cultured in Weymouth defined media. So after culturing, if you feed these cultured larvae to the lab rats, the adult will be formed in around 50 days. by shedding these larvae shed their sheath, and then these will transform into adult worms. The molecular diagnosis is actually done for invertebrate host. Clinically, clinical testing of these molecular diagnosis has not been validated in humans as such till now. But the genes which are being targeted are 1134 base pair fragments from parasite 18S rRNA genes, and which are targeting internal transcribed spacer 1 region. Imaging methods are quite important in these cases. So here in this case, you can see the involvement of the leptomeninges, which causes enhancement of the leptomeninges in both the brain as well as in the spinal cord. And you can see here, you are not able to see any focal lesions. Focal lesions are absent. As I have already told you, the focal lesions are not present. So that's why MRI and CT scan become quite important in these cases, because you need to differentiate it from other CNS parasites such as cysticercosis, paragonimiasis, nymphostomiasis, or toxoplasmosis. All right. So this is another case report which I wanted to highlight here. A 30-year-old farmer presented with diffuse abdominal pain, fever, and signs of peritoneal irritation. On examination, the patient was febrile with distended abdomen and pain, which is painful on deep palpation. So the blood examination showed high leukocyte count with high eosinophilia, that is 34%. The clinical diagnosis of acute appendicitis, the CT scan was also done in this case, and which has shown ileal perforation. So the clinical diagnosis of acute appendicitis with perforation was made in this case. The patient went for appendicectomy and ileal repair. So the macroscopic examination of the appendix and ileum showed fibrinous purulent exudate covering the ileocecal segment and ileal perforation with flat ulcer on the mucosal surfaces. The histology reveals transmural necrosis with arterial thrombosis as well as perivascular eosinophilic infiltrates. So adult worms of Angiostrongylius costarricensis was found in the lumen of the intestinal arteries. The ELISA was positive for IgG, and the patient was evaluated for 10 weeks following the surgery, and all the events remain uneventful, and the patient had a complete recovery after 10 weeks. So this is the important thing. It's a self-limiting illness. So abdominal angiostrongyliasis, you have to remember, is a self-limiting illness. And there are around, I've already told that 27 cases has been reported worldwide with the case series of around 116 patients. But the only thing that differentiate between the eosinophilic meningitis and abdominal angiostrongylitis is that the rat, here the worms lives in the ileocecal mesenteric arteries. Okay, but in case of eosinophilic meningitis or neural angiostrongyliasis, the worms lives in the pulmonary arteries of the rats. But here in this case, it lives inside the ileocecal mesenteric arteries. So the eggs hatch in the ileum, and the first stage of larva pass in the feces. So the larvae are then ingested by the mollusks, and then third stage larvae develop inside these mollusks or snails. So then again, how does the human get infection? These mollusks contaminate the vegetables, the fruits, and they are also taken up by the paratenic hosts such as crabs, shrimps, prawns, or frogs, which may eat these slugs and can get infected. And then humans, if they eat improperly cooked food or these exotic dishes, they can get the infection. So the diagnosis of abdominal angiostrongyliasis is purely based on histopathology. Okay, so here you can see the histopathological examination which has been done on the intestinal tissue stained with H&E staining. You can see adult and eggs in the figure A, and then you can see a thin shelled Angiostrongylos costrensis eggs in the intestinal tissues. This is another picture which is showing a female worm in the appendix tissue, which is stained with H&E stain. And there is a higher magnification. You can see thick multinucleated intestines with eggs and within the uterus. And the main important thing you need to do here is to differentiate these larvae, Angiostrongylos larvae, from Strongyloides stercoralis. The larvae of Strongyloides stercoralis has a double lateral alley. Okay, if you see, the lateral alley will be double. But in case of Angiostrongyloides, it is a single lateral alley which is present in the larvae. That is one thing. Second thing, a lot of granulomas are present in these cases. If you see the histopathological, if you do the histopathological examination of the intestine, you'll see a lot of granulomas, but the granulomas are actually absent in case of Strongyloides stercoralis. And the third thing, these larvae are not, will never come out in the feces in case of Angiostrongylos. But in case of Strongyloides stercoralis, you will see the larva in feces of human. That's how you will differentiate between these two important parasites. And obviously, you have to take the patient history, exposure to the mollusks, exposure to the rats, etc. Prevention is quite straightforward. You have to, it's a very preventable disease. You have to wash your fruits and vegetable properly, and then you have to look for the snails or slugs in the fruits and vegetables. If you can see them, don't eat, just remove. Okay, just throw those vegetables. The second important thing, you have to boil your food. Okay, your exotic food at least for 3 to 5 minutes. Continuous boiling has to be done. And if you don't have time, then you can boil for 165 degrees Celsius for 15 seconds. And then you also has to get rid of rats around your house. Snails and slugs has to be manually removed from the vicinity of your house. The treatment options are quite straightforward. In case of eosinophilic meningitis, the randomized trial has been done, and they have shown that you have to combine antihelminthic therapy with albendazole, given as 15 milligram per kilogram per day in divided doses for 2 weeks, along with the corticosteroids, which significantly reduce the symptoms of eosinophilic meningitis. So in some cases, the treatment has been given even for months together. So in case of abdominal angiostrongyliosis, the anti-helminthic drugs are not that effective, and it's a very self-limiting type of illness. So the patient improves on their own without any complications. So that was about my presentation. Thank you so much. If any questions are there, I'm happy to take. Any questions? I think I've cleared all my doubts to everyone.
Thank you, sir.
Thank you, ma'am.
Thank you, sir. You have nicely elaborated on the emerging parasite, and it has the wide range. That is, one, it produces a meningitis. Yes, and the other one, it is a self-limiting intra-abdominal. So it can be, it goes without any notice, or otherwise it becomes one of the biggest differential diagnoses for TB meningitis. And the more important thing is, it doesn't, the molecular diagnosis is not validated still.
Yes, ma'am.
That is what we depend on nowadays.
The important thing here is that you need to differentiate from the other CNS parasites.
Yes.
Like Xanthostomiasis. Xanthostomiasis is one of the important parasites. You have to basically differentiate. Cystic toxoplasmosis. These are the important. Yes, ma'am. But the only thing you have to look for the basically you have to look for the Yeah, you have to look for the eosinophil count. You have to look, basically, you have to rule
Image.
out the exposure to the other, intermediate hosts, etc. in those cases. And then focal lesions in the brain has to be seen in case by imaging techniques, etc.
Yes. So combination of imaging technique. And how far is the antigen detection ELISA kits available, sir? Is it available easily in the market for us to
Okay, ma'am.
depend on?
Yeah, ma'am. Actually, in India, there are around some of the case reports and case series has been published from Kottayam, Kerala, Mumbai Lilavati Hospital, as well as Indo-American Hospital from Kerala, and they have diagnosed these cases purely clinically, along with other parasites. So eosinophilic meningitis has been reported in India, but they haven't used any, kits or PCR-based method or any other method to diagnose this case. And they have the case reports are basically along with the other parasites. So in India, I'm not sure whether the kits are available or not.
So we depend on the history and on the
Exposure,
imaging techniques.
yeah, yes, imaging technique as well as exposure to the host, intermediate
So
host.
we have to think about this parasite also, because your epidemiology picture showed there is a full India is covered, one of the tropical
Yeah, it is. The picture has been taken from the
parasites.
CDC, madam.
CDC, yes.
Yeah,
Thank you very much, sir. You
thank you.
presented
We
it
have
very nicely in a very simpler language. And thank you so much for giving it in a nutshell. Thank you so much.
Thank you. Thank you so much.
Dr. Sangeeta?
Ma'am, can we move on?
Yeah.
Yes, ma'am. Can we move on to the next speaker, ma'am?
Yes. Shall I introduce the next speaker?
The next session of the day, the next session of the day is a guest lecture on Ganthostomiasis. To enlighten us on the topic, we have a distinguished speaker, Dr. Tuhina Banerjee.
Yes. Shall I start? And it's a pleasure for me to introducing Dr. Tuhina Banerjee, the next session of the day on Gnathostromiasis. And Dr. Tuhina Banerjee is currently working as a professor, Department of Microbiology, Institute of Medical Sciences, Banaras Hindu University, Varanasi. She is in charge of the parasitology section and is the infection control officer too. Her area of interest includes intestinal parasites with special reference to entamoeba and hookworm, and hospital adaptation strategies on multi-drug resistant organisms. She has published over 52 articles in various national and international journals, and she is also the assistant editor of the Journal of Tropical Parasitology. She is also a member of National Academy of Medical Sciences. She has won various awards like IIMM Merrick's Young Investigators Award by the Institute of Merrick, Council. She is recognized as the prominent researcher in AMR field in humans by the Department of Biotechnology, Government of India, Scoping Report, November 17. We are pleased to invite you, Madam, to talk on the Gnathostromiasis. Please, over to you, Madam.
Yes, I'm just trying to share my screen.
Ma'am, over to you, ma'am.
Yes, I'm just trying to share my screen.
Ma'am, if you have any problem,
Yeah. Is my screen visible?
Yeah, yes, ma'am. You can proceed.
Okay. A very good morning to all of you. And I'm really thankful to the entire organizing team of E-TROPACON 2020, Geeta Lakshmi Madam and her team, and also the entire IATP fraternity for giving me this privilege on this virtual platform. I think my slides are visible to all of you. Can somebody confirm?
Yes, ma'am. Yes, ma'am. Visible.
Okay.
Yes.
I'll speak on the topic nathostomiasis, and I have just added a subtitle to it as an emerging foodborne imported zoonosis. Emerging because of the increasing case reports over the years, and definitely foodborne because of the association of nathostomiasis with delicious names, drunken crabs, sushi, and definitely it is imported because of increasing reports in non-endemic areas with history of visit or travel to endemic areas. So I just drafted my lecture under the following headings as to introduce the parasite, the epidemiology, the life cycle, the major clinical presentations, the modes of diagnosis, treatment and prevention, and the final conclusion. If we ponder on emerging foodborne parasitic zoonosis, the very theme of this conference is parasitic zoonosis, and many of these parasitic zoonoses are foodborne. So if we ponder on why is there such rapid emergence of foodborne parasitic zoonosis, we find that international travel, especially across the tropics and subtropics, have increased tremendously. And parallel with it is the growth in taste, exotic food taste of the world. And what can be the best example in this pandemic than the Wuhan market itself? Along with this is a parallel development in agriculture business, in pisciculture, better transportation facilities of the marine products globally from one corner to the other. And along with all this, there has been population migration throughout the globe. Population migration doesn't only mean the physical movement of the people, but it is the movement of the entire culture, their ethnicity, and extensive sharing of this cultural habits. And therefore, the emergence of this foodborne parasitic zoonosis. In this context, nathostomiasis is a parasitic infection which is caused by the third stage larvae of helminths Nathostoma species. And it is endemic in those places, especially in Southeast Asia, where there is the practice of eating raw or partially cooked shellfish, or even freshwater fish, or those dishes involving snakes or eels or frogs. So this is the characteristic figure of the third stage larva of the Nathostoma spinigerum, which is the most common species. And this shows the characteristic head ends with definitely characterized 4 rows of circular hooklets, which is very specific for this organism, which I'll cover. So nathostomiasis is a parasitic infection by Nathostoma species. Now, if we look at the global epidemiology, we find the first case of nathostomiasis was interestingly discovered in right 1886, that too in a tiger in London Zoo, as a finding of a post-mortem examination having multiple abdominal nodules. This was later followed with the first case in humans in Thailand in 1889, and then after a long pause, the second case was in 1934. But what is interesting is India reported the first case right in 1945, and then a series of cases followed till date. So the global distribution over the world shows that definitely Southeast Asia and Orient countries, China and Japan, include India, are definitely endemic. There has been increasing reports of African countries also with this condition. And interestingly, Latin American countries in South and Central America are also increasingly reporting evidence of this parasite, especially Mexico, where there is a practice of consuming raw fish marinated in lime juice. And Japan, in fact, has one of the highest incidence of cases of Nathostomiasis. Now, there's a very interesting association with sushi bars and Nathostomiasis. Sushi is a delicacy, a Japanese delicacy, involving rice and partially cooked or raw fresh fish. So, because of the huge growth in sushi bars in the West, it was expected that Nathostomiasis cases would have been increasingly reported from this West. But this did not happen, because it was noted that it is not the sushi which is responsible, but in which place you consume the sushi, which is more important, whether you get Nathostomiasis or not. For example, sushi bars or restaurants in local endemic places who use local raw fish or fish from salty brackish water, where there is no well-regulated food policies, there the fish is more contaminated with this parasite rather than the West, where the sushi bars use good quality water, expensive materials, as well as they have well-regulated policies for having a control over the quality. Therefore, the association with sushi and this parasite varies depending on the quality of the sushi that is being consumed. If we see the status in India, well, this condition is quite prevalent. We have 14 cases of ocular presentation of Nathostomiasis already reported from the country, and 2 cases of cutaneous Nathostomiasis. And the map very well says that it is distributed well in many states, with reports from Meghalaya in Northeast, involving many reports from West Bengal, Orissa, Maharashtra, and South India, involving Puducherry, Chennai. Now, from Central India, we have both presentation or reports of cutaneous as well as ocular presentations of Nathostomiasis, and a single case has also been reported from Delhi of cutaneous manifestation in a person from Manipur for cutaneous Nathostomiasis. So this is prevalent in India too. If we consider the major Nathostomas species, it is 12 or 13 species in total, which is distributed geographically among humans and animals. And in this, 4 major species are those which cause infections in humans. The Spinigerm species is the most common cause, while the species Hispidum and Nipponicum has also been reported, especially from Japan, and Doloresi from other Southeast Asian countries. Most of the reviews say about the and binucleatum. The binucleatum specifically being reported from Mexico as the commonest species causing Nathostomiasis in humans. Now, these species are capable of causing both cutaneous and visceral manifestations in man, and infections in animals are non-specific, like gastritis or lack of appetite in the cats and dogs or felines or fresh fish-eating mammals. Most of the cases are incidental findings of abdominal nodules as a post-mortem finding in pigs and wild boars. But studies on infections in animals is very limited, and therefore non-specific findings are in major. If we discuss about the morphology of the parasite, the egg, this is the egg with the distinct knob at one end, but this is found in the stool of the definitive hosts, not in humans. The definitive hosts being cats, dogs, pigs, or any felines or canines with habit of eating freshwater fish. The third stage larva is actually what is the infective stage in man, found in man, having
hosts being cats, dogs, pigs,
distinct structure of head and a blunt end. The head bears bulbar structures with distinct 4 hooklets, which I've shown in the figures and will subsequently show. This is approximately of the size of 5 millimeters, 5 to 10 millimeters. The adult forms, male and female, vary between 15 to 25 millimeters with distinct mouth parts and a blunt end, and having esophagus and their respective reproductive organs as visualized. Now, coming to the life cycle of this parasite. So as I mentioned, the pigs, cats, dogs, or other wild animals, freshwater fish-eating animals are the definitive hosts in whose stool the unembryonated eggs pass. These eggs embryonate in water, they hatch and release the first stage L1 larva. This is ingested by the small crustaceans in water, especially the cyclops or water fleas, where the larva molts twice to form an early L3 larva. This, when ingested by the second intermediate host, which are amphibians or fish or reptiles, there the larvae develop into advanced L3 larva. This may be consumed when they are eaten by paratenic hosts like ducks, chickens, snake, or directly ingested by pigs and the other definitive hosts. The cycle continues in this manner. Now, where does man come in this cycle of infection? Man is an accidental host always. So the advanced L3 larva, which goes into aberrant migration inside the human body, can enter by consumption of the secondary intermediate host. And there is no further development of the larva inside the human body. There are 2 alternative routes in addition to this common life cycle, which has also been proposed and seen. One is man can also get infected by direct ingestion of water infected with the crustaceans and L1 larva, or there can be direct penetration of the larva in food handlers from the infected meat. So these are the 2 alternative routes which can also occur in infection in man. Now coming to the clinical presentation, the major presentation in most of the cases is cutaneous manifestation, and the classical one is the migratory nodular paniculitis. So you have redness nodules at one place, which over 1 week goes to some other place. It's very non-specific and can occur in any part of the body. Now this this picture of this person, this is a British male tourist who had visited Botswana and then presented with this feature. We can see a very distinct swelling on his forehead and then on the shoulder due to the migration of the larva. Now this swelling, this nodules form due to the mechanical damage caused by the migration of the larva as well as the host immune response. So this migration can occur in any part of the body at any time. This photograph is from an case report from India, where a vegetarian woman presented with non-specific nodules which migrated for nearly a month on and off. She attended the OPD, was treated non-specifically, but finally when the nodule presented on her palm, and this was a distinct serpiginous skin eruption which was found, which on incision drainage actually liberated the worm. So when this happened, then the case came into being. So the cutaneous manifestation is one of the commonest presentation of this parasite. However, because the larva blindly migrates inside the human body and can invade any organ, it is highly motile and highly invasive. So there can also be a plethora of other visceral manifestations like the genitourinary manifestations, mostly presenting as hematuria, The pulmonary manifestations presenting as pleuritic chest pain or hemoptysis or pleural effusion. The gastrointestinal manifestations, most commonly vague abdominal pain. The ocular manifestations, of course, with red, itchy, painful eyes. The auricular manifestations can be mastoiditis or just a larva coming out of the external ear. And the most serious, there can be central nervous system manifestation too, which can ultimately result in fatal meningoencephalitis, though rare. Now, this is again a picture from a case report in India where you can distinctly see the larva of the parasite in the anterior chamber of the eye. Now, the ocular presentation is the most reported presentation among all the visceral manifestations simply because of the fact that the eye is the only organ where the larva can be viewed. You can visualize the larva. And definitely, the increasing reports of ocular manifestations is an indirect hint that the prevalence of this parasite causing other manifestations is definitely underestimated. If we come to the diagnosis, of course, suggestive history with a classical triad of intermittent migratory swellings, along with a non-specific eosinophilia, and a travel history to Southeast Asia or other areas which have endemicity with gnathostomiasis, suggests the probable diagnosis, and this suggestive history is very important. Of course, definitive diagnosis is isolation and identification of the intact larva from the human body, but this is not always possible. All the cases which have been reported in the country mostly show the presence of this larva, or the case reports have been diagnosed based on the identification of the larva. The larva shows distinct mouth parts, as I have already mentioned, the bulbar mouth parts, with distinct 4 roots of circular hooklets present at the head end and a blunt tail end around 5 to 10 millimeters in length. But this suggestive history of eosinophilia is very non-specific, and as already mentioned by Dr. Vinay for angiosprongidiasis, this is mostly non-specific, but of course the intermittent migratory swellings along with the travel history is very suggestive of the probable parasite. In vitro culture is not mostly used for diagnosis, but has been used for isolation of proteins, the excretory secretory proteins, which could be used for serological tests. Now, several excretory secretory proteins of this parasite was being tried and tested so that some serological tests come up, but there was a lot of cross reactivity with other helminths like Paragonimus, like Toxocara, like even Angiostrongylis, as Dr. Vinay pointed. Therefore, after a lot of trials, a specific third stage larval antigen of 24 kilodalton molecular mass had the greatest specificity and reacted only with gnathostomiasis sera, which is available in an immunoblot technique format and is extensively used in European countries and in some reference centers of United Kingdoms and Thailand and Switzerland for serological diagnosis of this parasite. However, this is not widely available, not in India, and as such, the chemicals or the constituents for the serological test is not available at present other than the reference centers. Now, lately there was one of recent publication of a ICT format, the CAN test for gnathostomiasis kit, which came up with the National Institute of Infectious Diseases, Tokyo, in collaboration with pharmaceutical companies, where a recombinant protein of this parasite has been used in format of ICT for diagnosis of human infections, where it simply gives a test like any other ICT, a control and a test band for cases of gnathostomiasis. Now, this showed a sensitivity specificity of around 95%, a sensitivity still higher of 97%, and is still being used only limited to Japan, not widely available, while this immunoblock technique using the 24 kilodalton antigen has around 90% specificity and 95% sensitivity, but has very limited use only in these areas. Therefore, a better approach is for the exact species identification is the molecular approach, and definitely the cytochrome oxidase gene, the COX-1 gene, and the entire ITS2 region. We know the inter-transcriber sequence region is very, very unique for species identification, which is mostly used for this parasite. This is a publication from this year from a study in Mexico where a duplex assay has been standardized to detect their Gnathostoma species, and it is this study which has highlighted that it is the binucleatum species which is in majority in Mexico among the cases of human Gnathostomiasis. So molecular diagnosis definitely is very promising for this parasite for the exact species identification too for epidemiological purposes. With this diagnosis, coming to treatment. Now for long, there was no specific treatment for this parasite, but later, after a series of studies, it was seen that albendazole and ivermectin, either of the drugs, was effective in treatment, with albendazole at twice a day for 21 days. In here, whatever case reports we have, the treatment has been successful with this drug. However, in endemic areas, recurrence or reinfection or treatment failure is very difficult to diagnose. And therefore, if the symptoms recur within 7 months of treatment with either of these drugs, it is usually considered as treatment failure, and a second course of these drugs is started. And a pragmatic approach that is followed is, if there is no symptoms within 12 months of treatment, then it is usually considered as a successful treatment. So seeing all these treatment limitations and the prevalence of the parasite and the increasing emergence of case reports, it is better to prevent the condition, and consumption of completely cooked food seems to be the only way for prevention of this infection, because rapid freezing at -20 degrees Celsius of the fish did not work. Even marinating the fish in lime juice, which Mexican countries thought would suffice, did not prevent the infection. So it is only the consumption of completely cooked, properly cooked food which kills the parasite, the fish. Therefore, the take-home message for this condition, for this parasite, is that because 100% cure rate is not achievable with the available anti-helminthic treatment, prevention of the infection is of immense priority. And we all know that it is very difficult to change the eating habits of people. Therefore, a better approach would be to raise the awareness of the public for avoidance of raw, uncooked fish and the preference for cooked food, so that people on their own change their habits, being aware and not being forced upon. Another very important point is that there are very few clinicians outside the areas of endemicity who are familiar with the condition. And the classical triad of migratory nodules, eosinophilia, should hint at diagnosis, but this is often missed or there is delay in diagnosis because of unawareness even among the clinicians, which could result in potential serious consequences. And definitely there is a large and wide variety of reservoirs and intermediate hosts, which makes it almost impossible to eliminate this parasite, and therefore prevention is very, very important. Despite this discussion related to this parasite, few unanswered questions still remain. What is the entire spectrum of wild reservoirs is still not known. Highly specific antigens for immune diagnosis is yet to be identified and used. And the effective treatment regimen for complete cure of this parasite is still to be clearly studied and implemented. However, with emergence of this parasite, we understand we'll know more about the condition and we'll have a better understanding of this parasite. So thank you all for this privilege and for your attention. If there's any question which I could answer, I'll be happy to take. Thank you. Thank you.
Thank you very much, madam. You have given a nice alarming or alert carry home message. That is, previously it was an endemicity. It was prevalent in endemic areas, and now it has moved away from the endemic areas also. Because one disadvantage of being globalization and trying out food. As you said rightly, we have to have the food regulatories, and also we should be very, very keen on clear about how to cook the food and eat it, rather than trying out many different menus. And this cross reactivity is also one of the main feature which we have to take it, consider it, and also the diagnostic modalities. And since there is no ICT in India, no matter for diagnosis, so far there is no ICT followed or cartridges available here also. That is still in Tokyo.
Yes.
Fine. And only the prevention. Prevention is the only mode where we can reduce, because as you said, South India is also more infected. Cases, nearly 72 cases have been reported in India at the latest. That is what the Google says. And thank you very much for giving us an elaborate, clear idea and how to be with the parasitology diagnosis. We should not ignore, since many of the clinicians are also unaware. We have to have in mind when there is a migratory nodule and a vision defect all of a sudden. Yes, madam. One query I have to clarify. If it is an ocular infection, since it is migratory, will that go off or it produces a permanent damage? There will be definitely an ocular vision defect.
Yes. So,
Anterior.
yes, of the case reports, most of them, after 17 case reports global of ocular nathostomiasis, 14 are in anterior chamber and 3 have affected the posterior chamber. Of this, most common symptom was, of course, redness, itchiness, pain, loss of vision, but none had any permanent damage. But for that inflammation, we have to administer the physician-administered steroid along with the usual course of anthelmintics. But none reported a permanent damage.
The same ivermectin and albendazole helps for the ocular also. Okay. Thank you so much, madam. And thank you. I thank the organizing team and especially Geeta Lakshmi madam and Kala madam for giving me this opportunity. And nice interactive sessions we had. We listened to nice topics from the very good speakers. Thank you. Thank you very much.
Ma'am, there are some queries from the participants. So one of the participants, Shivapriya Swami, has asked if there is any co-infection with other parasitic infections reported.
For this nathostomiasis?
Yes, ma'am.
Yes. Of the reports, as such, there are no co-infections, but it is very likely that because foodborne zoonotic infections are common and this raw, fresh water fish or partially cooked shellfish is a common source for many of the other parasites. So co-infections can very well happen with these parasites. But of the case reports, they are only single parasites. But it is very much possible to have co-infections.
Okay, ma'am. Okay. And one more person has asked about if there is any veterinary studies from India available, ma'am.
On nathostomiasis? No, right? No.
Yes, ma'am.
No. Actually, veterinary studies mostly are, as I told you, they're incidental post-mortem findings as nodular muscles inside the abdomen, which, when dissected, was having the larva or the adult worm in the definitive hosts. But as such, a methodical to study the prevalence of nathostomiasis in veterinary population, which is very important, we don't have such data.
And one more question from Kiran Madhu, ma'am. He has asked, does it mimic microfilaria in eye?
See, worm in eye, the first thing which comes to our mind is microfilaria. What? Why me? The physicians or the ophthalmologist's mind also comes as microfilaria. So, the redness, itchiness, and the pain which is caused by Gnathostomosis or these invasive parasites is more than any of the microfilaria causing ophthalmic presentations. And worm in the eye can have a differential diagnosis of a microfilaria in the eye. However, the presentations of the larva of Gnathostomosis in the eye is more severe than that of microfilaria.
Okay. Thank you so much, ma'am.
One question, madam. Dr. Tuina, that was an excellent presentation. And you have told that it is very common in Japan where the sushi, that use of the sushi food, type of food is more prevalent. But it is not that common in North America where also the sushi bars are mushrooming, coming up quite well. The thing is that you told in your presentation that the place from where the fish is procured, etc., that has got a bearing on that. That is why it is more common in Japan and not in USA. But I think that is a paradox, madam, because Australia is country and there is sanitation is excellent.
Yes.
There is no fecal contamination. It's very common in Japan, but not in North America, country like that, Canada or USA.
Yes, exactly. Most of the sushi exports are not from Japan. They are from other Southeast Asian countries like Thailand, one of the major exporters of these raw materials for preparation of sushi in the West. And prevalence of this parasite in Thailand is also considerable, where there are no well-developed regulatory policies to put a check on the quality of the fish. So it is not only Japan exporting the fish, the other Southeast Asian countries also export the fish. Where the actual problem lies, it is not only Japan.
Thank you.
Can I add on worm in the eye, please?
Yes, sir.
Actually, if you see Dirofilaria, then Felicia and other worms also, they can infect the eye. And rarely, and you can even get Cysticercus larvae sometimes. So there are a number of parasites which can go to the eye, they stay there. And sometimes they do cause symptoms and sometimes they don't cause symptoms. The main symptoms are occurring, as you have told, steroids are given. Main symptoms occur when the worm dies. Or when there is an allergic reaction. But however, the eye is sort of a separate system. So you may not get that much symptom there. That is the reason actually. It is actually in the superior chamber. So that is the reason you don't get. But then there are other worms like in which do not occur in India also. Then some others which are occurring in Africa also. They just in fact the eye itself. I think nobody more other questions. Anyway, excellent lecture, Dr. Tuina.
Thank you, sir.
Thank you, madam. Thanks a lot.
Thank you.
Ma'am, shall we move on to the next section, ma'am?
Yes, Kavita will present.
Yes, madam. Yes, madam. I'm here.
Okay, ma'am. Ma'am, the next section of the day is guest lecture on Gnathostomias, dictophymiasis. To enlighten us on the topic, we have with us distinct speaker, Dr. Nishant Verma.
Good morning. I would like to introduce Dr. Nishant Verma, Associate Professor, Department of Microbiology, AIIMS, New Delhi. His areas of interest in research of parasitology is he is the principal investigator of magnitude of asymptomatic malaria infections in pregnant women in a community hospital and prevalence of microsporidia and the molecular characterization. He is also the co-investigator for many other projects. He is a guide and or co-guide for many theses of MD and DM infectious diseases. He is He has contributed a chapter in microbiological diagnosis of parasitic diseases in the book entitled Bench to Bedside, Diagnostic Microbiology for the Clinicians. He is a member of the Committee for DM Infectious Diseases Teaching. Over to you, sir, Dr. Nishant Verma. I request him to continue on the topic diactophymiasis.
Thank you very much for your kind introduction. Firstly, I would like to thank all the organizers, including Professor Geeta Lakshmi, for giving me the opportunity to talk about this topic, diactophymiasis, which is a highly neglected disease, very rarely seen in the world, and so very little is known about diactophymiasis. So I would also like to thank Professor Parija because he about diactophymiasis. So that is how I came to know about diactophymiasis. So I will try to share some of the knowledge I gained through the literature which is available. So is my screen visible to all?
Yes.
Yes, Nishant, go ahead. All the best.
Thank you. So the topic which has been given to me is diactophymiasis. This is a zoonotic disease, as the theme of the conference is zoonotic disease. So we are covering many topics, very interesting topics, ranging from angiospermoglosis to gnathostomiasis and others. So diactophymiasis is another zoonotic infection which is caused by a nematode known as Diactophyme renale. The other name is Diactophyma renale. Its common name is giant kidney worm because its main habitat is kidney in the definitive host. It is one of the largest parasitic nematodes seen in carnivorous mammals, including mink, wolves, cats, and others. So it has a wide host range, particularly among the carnivorous animals. It's also the largest known parasitic nematode of human beings, where females can range up to 1 meter long. So it's a very long nematode. So it's hard to miss this parasite if you find one in any case. Just a brief discussion about the history of this parasite. When we see the records, we find that the oldest records date for around 3384 to 3370 BC, where Dictophymidae eggs were reported in human coprolites from a Neolithic site in Arbon bleach tree in Switzerland. So this is the oldest record which is available. Now, D. renale has been known since 1583, and in 16th century, it was commonly known as red scourge. In 1782, it was Goes who first described this worm in a case of a dog infection, where it was demonstrated in dog's kidney. And in 1945, the life cycle of this nematode was described by Woodhead. In 1989, the International Commission on Zoological Nomenclature named this worm as Diactophyme renale, as was suggested by Tollett in the year 1987. When we see the taxonomy, this worm belongs to phylum Nematoda, class Anople, order Dictophymatida, family Dictophymidae, and genus Dictophyme. The morphology of this worm is quite characteristic. When we see the adult worm, as I said earlier, it is one of the largest nematodes which cause human infections. It is bright red in color, so it's very hard to miss this worm because of its characteristic size and the deep red color which you see when you actually retrieve this worm from any person. There is a characteristic tapering at both the ends, which is quite similar to many other nematodes, but the color and the size is very characteristic, so that way you can easily identify this worm if you find one. The female worms are larger, as you as is true for many other nematodes. The females are larger than males, so females can range up to 1 meter long and with diameter of around 0.5 to 1.2 centimeters. On the other hand, male worms are just around 20 to 40 centimeters long with a diameter of around 5 to 6 mm. The eggs of this nematode have some characteristic features, but they may resemble some other nematode eggs as well. For example, if you see the image here, these eggs have a characteristic rough surface, which may confuse you sometimes with the eggs of Ascaris. And they also have bipolar plugs at both the ends. Now, these plugs can sometimes confuse you with some of the other nematodes like trichuris or with capillaria. Now, when you see these eggs, they are again oval to elliptical. Their size is similar to that of Ascaris eggs in the range of around 60 to 80 microns by around 40 microns wide. They are transparent to yellow colored, and they have a thick roughened shell on the surface, as you see sometimes in Ascaris. It's a similar looking kind of shell. The larvae of this worm are just around 6 to 10 millimeter long with around 0.1 to 0.2 millimeter diameter. Now, the life cycle of this nematode is quite complex with two intermediate hosts. The first intermediate host is an invertebrate aquatic oligochaete worm, such as Lumbriculus variegatus, while there are a number of other paratenic hosts which act as second intermediate hosts for this parasite. Particularly, frogs and fish are the important animals in the life cycle. Humans are an incidental host for this parasite, and there are hardly 40 cases, maybe less than 40 cases, which have been reported worldwide. And humans acquire the infection incidentally when they consume some raw or undercooked paratenic hosts, such as fish, which contain the infective L3 stage larvae of this parasite. An interesting point to note is that the life cycle of the Dictophyma renale may take up to 2 years to complete in the environment. Just going into details of the life cycle, for example, we have a definitive host such as cat. If there is an infection of Dictophyma renale, the prime habitat for this worm is the kidneys of the animals. Now, the adult worm resides inside the kidneys, and after maturation, they start laying eggs. Now, these eggs are passed with the urine of the definitive host. They reach the environment, they enter into the water bodies, and here they will undergo further development to become embryonated in the environment. Now, with time, these embryonated eggs are ingested by the first intermediate host, which is an invertebrate aquatic oligochaete worm. And after the ingestion of this larvae, embryonated eggs, these larvae develop inside this worm, and further these worms may be ingested by the paratenic hosts such as fish or frogs, and these L3 larvae, they come out into the digestive tract and they become encysted inside the tissues of these paratenic hosts. Now, these paratenic hosts with the encysted L3 larvae, when they are ingested by a definitive host, they start developing inside, they pass through the digestive tract, penetrate through the digestive tract, and ultimately come to lie inside the kidney. So kidney is the final site where most of the times this infection will settle in. Now, man can acquire infection again, like other definitive hosts. It can acquire the infection by consumption of some raw or undercooked paratenic hosts, such as fish and frogs, and the life cycle is again similar. So the patient may start passing urine which contains the eggs of this parasite if he's getting the infection inside the kidneys. Something about pathogenesis and pathology. As I said, it's a very rare parasite which has been reported. Very few cases in humans, and very limited information is available about this nematode. However, based on the literature which is currently available, we can say that the cuticle has an important role to play. It protects the nematode inside the digestive tract, and it probably has a role in the invasion and migration of this nematode inside the host body. Hypothesis regarding how this larvae migrate and how do they localize inside any particular site. So, depending on the site, it's primarily the site which has been identified where the penetration is happening. For example, if the eggs have been ingested and the larvae hatch out and they penetrate through the digestive lining, so depending upon the site of penetration of these larvae inside the digestive tract, different sites may be involved. For example, if the duodenum wall has been penetrated, it's the right kidney which is more likely to be involved. If the greater curvature of penetrated, it's the left kidney which is more likely to be involved. While in case of lesser curvature of penetration, liver is the commonest site which has been reported to be involved with this infection. Now, the strong inflammatory reactions happen inside the renal parenchyma while the adults are residing inside the kidneys. And because of the presence of these kidneys and strong inflammation, the affected kidney eventually loses its function. To compensate for this, the other kidney undergoes hypertrophy. So this hydronephrosis actually develops inside the other kidney. So eventually the patient may suffer from renal failure and may succumb to this infection. The worm has also been reported inside the body cavity and if the peritoneum is involved, it may lead to peritonitis and adhesion formation may happen. So this complication may also occur in case the body cavities are involved. Now, something about the eggs which are shed in the urine of the definitive host. Now, these eggs have some characteristic features, and they help the eggs to survive or tide off the adverse situations inside the environment. So they develop inside the water, and here the thickness and roughness on the shell helps them in survival. Because the wavy character increases the surface area. Now, this increased surface area helps it to extract dissolved oxygen in areas where the percentage of oxygen is low inside water. So this roughness and increased surface area is going to help that. Now, there are depressions and projections on the egg surface. They are again helpful for the worm as they help in the attachment of these eggs to the plant surfaces. And by doing it, by doing this, it increases the chance that these eggs are ingested by the intermediate aquatic oligochaete host. So this actually helps the worm to complete its life cycle and assists in the transmission. Now, several parasite proteins have been studied. Most of these are lipid binding proteins which have been identified, and their functions are not very clear. They probably have some role in energy storage distribution or cell-to-cell signaling or interference with the host's immune system. So all these roles have been suggested, but nothing convincing is available in literature. So these are some of the suggested functions of these proteins. One of the protein known as the P17 protein is a possible heme-containing hemoglobin. Now, it has been suggested that it may have some role in carrying the oxygen, and it is this protein which has a role in making the adult worm appear quite bright red. This bright red color is probably contributed by the P17 protein, which is a hemoglobin. Another protein, P44, has been seen in the pseudocoelomic fluid, and its role has been linked to the lipid distribution within the nematode and also in the stimulation of the inflammatory and local tissue responses inside the host. Now, coming to the disease spectrum of dactylophymaceae in human beings, the clinical presentation is highly varied. We have very limited number of cases reported, but even then the clinical presentation has been highly varied. However, majority of the patients present with renal involvement. So in more than 80% of the cases, renal involvement has been reported. Besides that, cases of subcutaneous tissue involvement and retroperitoneal cavity involvement has also been reported, but those cases are quite few in number. The patients generally present with non-specific signs and symptoms, and most of the times, the commonest complaint you see is the presence of loin pain and hematuria, which has been seen in up to 60% of cases. Otherwise, the symptoms and signs are highly non-specific, ranging from abdominal pain to fever, weight loss, urine retention, and pyuria. In extreme of cases, even death has been reported due to renal failure in case both the kidneys function is compromised. And also sepsis is a possibility in case of severe cases here. In animals, the disease spectrum is again similar, with renal involvement being the most common presentation, which again progressively damages the renal parenchyma, leading to renal failure. The encysted worms have been reported in abdominal cavity, subcutaneous tissues, mesenteric lymph nodes, uterus, and ovaries, and even the cases of peritonitis have been described. However, many animals may remain asymptomatic while suffering from dactylophymaceae. So it has a wide distribution. The animals may remain asymptomatic, and they may act as a pool or reservoir from which you will have a continuous transmission of dactylophymaceae in the environment. So that's why it's an important disease to know, so that in case we encounter such diseases, we are in a ready position to take care of that. Now, coming to diagnosis. Now, diagnosis, again, we have limited options available for diagnosing dactylophymaceae. In case of human cases, it's primarily the microscopy which has been used for diagnosis. Other thing which makes the diagnosis challenging is that it's a rare disease. It's a very rare disease and very limited awareness amongst the clinicians as well as in lab personnel. So because of this limited awareness, many times we are not even suspecting dactylophymaceae. So there are some clues which you can take from the patient. For example, if there's a history of consumption of undercooked or raw fish, which is a thing which is common to many other zoonotic infections. So if you have a history of such a habit, then you should think about zoonotic infections, including dactylophymaceae. If you have some imaging findings, such as enlarged or calcified kidneys, this may again suggest, or you should have a differential of dactylophymaceae in mind in case some other things are matching in the history. On urine examination, you may find pyuria or proteinuria, which is again a non-specific finding, but these findings in combination may be useful when you are trying to approach a case where diactopamine acid is a differential. Now, microscopy is the most important technique which has been used worldwide for confirming a case, wherein you demonstrate the eggs of diactophyma renale in urine. Now, this is the most common approach and most commonly reported way of diagnosing diactophymiasis. However, occasionally adult worms have been passed in urine. Now, this is a very uncommon thing to see. However, this is one of the approaches by which you may sometimes detect a bright red colored adult worm in the urine, and then you can confirm that this is a diactophymiasis case. Another option is histopathology. For example, if there is a tissue involvement, such as subcutaneous tissue involvement or mesenteric lymph node involvement, in those cases, if you are performing histopathology, you may be able to demonstrate the presence of the adult worms. Now, serological options are also available, but they are available only for animal studies or in animal sera. For human use, no serological techniques are still available. So there is a lack of serological assay where we can use it for human studies or for human diagnosis. For animal sera, there is an indirect ELISA where anti-D-renal antibodies, which are based on a soluble antigen derived from the esophagus of adult diactophyma renale worm, has been used, and it has shown a high sensitivity and specificity of the order of around 92 to 93%. But sadly, we don't have this assay available for human cases. Now, molecular diagnosis has also been attempted using a PCR followed by sequencing of the small subunit ribosomal DNA and the mitochondrial cytochrome C oxidase subunit 1 gene. They have been described, but they have a limited application. So this is a table which is showing a compiled information about all the diagnostic approaches we have. So molecular assay, not only they have limited application because you require extra expertise and ready setup for doing these assays in case you are looking for diactophyma, and also it's not that cost effective for routine diagnosis. But yes, these are the options we have till now. So hopefully we will have some more and better diagnostic methods available which can improve the our knowledge about this worm. So till now, the diagnosis mainly relies on demonstration of the eggs of this worm in urine, histopathological identification and other microscopic methods, and demonstration of adult worm in the gross specimens. And molecular identification is not routinely used for routine diagnosis. This is the take-home message for the diagnosis part of this worm. Now, animal models have also been studied, but then they are not well established. There was a study by Abdul Hakim et al., where the authors reported the infection of this worm in Balbi mice, which were being screened for intestinal parasite. Now, this was an incidental finding where Balbi mice demonstrated the presence of these worms, but they are still not have been identified as a good model for studying these parasites. Now, application and evaluation of appropriate animal models is one important aspect of research which needs to be explored, because this will improve our understanding of both the pathogenesis and understanding and knowing the therapeutic modalities which may be used in future in case we want to deal with diactophymiasis in any community. Now, what is the public health importance of diactophymiasis? Like any other zoonotic infection, diactophymiasis has its own importance in public health. It has a worldwide distribution, particularly in temperate regions. It has a wide host range, particularly amongst carnivores and in wild setups. Freshwater fish and frogs are common paratenic hosts. Now, all these animals, they many times are part of our lives, not the wild animals, of course, but yes, the domestic animals, as well as we commonly consume frogs and fish in many countries. Now, these freshwater fish and frogs, if they are not properly cooked, if we are not taking care of the food hygiene, so these things can actually affect the chances or increase the transmission of the disease in any host. So D-renal larvae has also been observed in snakes, freshwater turtles, fish. So it shows that the worm is widely present. It is widely present in wildlife. The possibility of such perpetuation of life cycle in urban areas cannot be ruled out. So we, as I said, it's important to know about diactophymiasis because we should be ready in case we have to face a problem of diactophymiasis on a larger scale, and also to identify the cases and cure them so that they to end their suffering. as I said again, human infections are there, but they are very rare. We have only sporadic cases reported. Most of the cases have been reported in over 10 countries, including China, Australia, Greece, India, Indonesia, Iran, Japan, Thailand, USA, and Yugoslavia. Most of the cases have been reported from China, and this is probably linked to the consumption of raw and poorly cooked fish or frogs in this country. In India, only 3 cases have been reported till date. 2 have been published in index journals, while 1 has been published in a non-index journal. First case was reported from Telangana in 2014, where the patient presented with high-grade fever, hematuria, and worms were found in the urine. Another case was reported in Bareilly, where again the eggs were reported in the urine. This was an incidental finding. And in 2016, in Muzaffarnagar, a case presented with urinary retention, hydronephrosis, and hematuria, and worms were passed in the urine. So that is how they came to know about the diagnosis. Coming to the treatment part, as I said, the dictofamiasis is a very limitedly studied organism and disease. So we don't have any defined management protocol as yet. If the worms are identified, they can be surgically removed. This is one of the options which have been tried. Nephrectomy may have to be resolved to in severe cases where the kidneys have been severely damaged and not available. That is another option which has been used. In a case from Yugoslavia, ivermectin was used and the patient remained asymptomatic during the next 6-year follow-up. Now this is again amongst a couple of cases where ivermectin has been used in treatment. And in some places, albendazole has also been tried, but again, the results have been varied. And again, we don't have any clear information about which treatment will best suit the management of dictofamiasis. And most of the anti-helminthic agents have not been extensively evaluated for the management of these cases. Now, surveillance and prevention, as with other parasitic infection and particularly zoonotic infection, this is a very important aspect. Dictofamiasis, which has a public health importance because of its widespread habitat in the zoonotic areas. And so the best approach, again, as Dr. G.R said, that we have to resort to prevention. So avoid consumption of raw or undercooked fish or other possible paratonic hosts. This is the best approach to follow. In case we can avoid this, we are reducing our chances of getting the infection to a large extent. Also, promotion of usage of clean food and water is also important because it's possible that sometimes you may get infection even by ingestion of the eggs of this worm. The prevention of infection in cats and dogs is also very important because these animals generally scavenge on the fishing industry waste and byproducts. So if you can take care of this important gap by taking care of the disposal of the waste from these industries and avoiding the contact of the cats and dogs with these industrial wastes, probably we can again prevent the transmission in a setup. So what are the unanswered problems for dictofamiasis? There are a lot of questions remaining. There are few answers available. But the 3 most important areas which we need to consider here is that we need to develop effective methods for diagnosing and screening the infections. We also need to develop effective therapeutic modalities, which we know that is going to work properly for the management. And we also need to understand the epidemiology of this worm in human beings so that we are in a better position to know what is the status of dictofamiasis in any setup. So that is something which we need to understand. Thank you very much for your patient listening. I will be happy to answer if there are any queries.
Thank you, Dr. Nishant. It was a really fascinating presentation to see the red worm. Actually, I don't think we have seen any red color worm in our practice. And you have done extensive reading and updates and gave a clear presentation on this. And I think you have also instilled an interest in the youngsters to read up many things and identify new worms. Many times we don't know many things probably, and your presentation has made us to go into all these things. And nowadays the travel has made so many people try new foods. All of them have a fantasy for eating new things, and we have to ensure that it is properly cooked and then it has to be taken, because many diseases, especially parasitic infections, spread by this. And thank you very much, sir. one more thing was differential diagnosis to look for ascaris. We have to keep them updated. We can also tell the clinicians, especially in the nephrology department, to into this in case of hematuria and look for these eggs and see whether it has look more into the morphology and just not go with an ascaris. We can just look at parasitic infection. Yeah. I think with this.
Ma'am, there's a question from the participant side. Dr. Dhruti Seth has asked,
yes,
what sample do we use for PCR diagnosis?
So for PCR diagnosis, either we need the adult worm, or if we have a chance to see the eggs, if we are not sure, then again, those samples may be used for PCR. And thirdly, if we by chance find any larvae in any tissue, then again that PCR strategy can be used for that also.
Okay, sir. Okay. And I guess only that much we have received, sir.
Thank you.
And
yes, and we also have to like look whether the veterinarians. Yes, this the veterinarians also should be kept in mind to see if there is any to find out. Yes, ma'am. Thank you. Shall we move on to our next session?
Ma'am, is it okay? Can we have a short break?
Yes, ma'am. Yes, ma'am.
Okay. So we thank all the guest lecturers for throwing some light on the topic. So let us part for a short break and we can get back in 10 minutes for the next session, which is going to be a symposium on emerging zoonotic helminthic infection. So let's have a short break. And thank you, chairpersons, for giving a lovely introduction to the guest lecturers.
Thank you.
on emerging zoonotic helminthic infection, Dirofilaria. The eminent speakers include Dr. Sumita Kurna, Dr. Saurav Mathu, Dr. K.N. Prasad, and Dr. Tapashi Ghosh. Will be introduced by Dr. Kavita ma'am.
Yes. Good morning, all of you. I have great pleasure in introducing Dr. Kashilnath Prasad, who needs no introduction. He is now presently the Senior Consultant Head of Department of Microbiology, Apollo Medic Super Specialty Hospital. He was the ex-professor and Head Department of Microbiology, Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. His teaching and research experience goes to more than 35 years. And he has 233 publications, 7 book chapters, 14 CMEs conducted and organized. He has guided thesis for 26 PhD students, and he has 25 completed research projects. He has 6 awards, of which S.C. Agarwal Oration Award by IIMM and IIMM Endowment Oration Award are some of them. He has been an invited speaker in 60 national and international events. Is the fellow of national and international academic bodies, including the fellow of Royal Society of Tropical Medicine and Hygiene. His area of interest in parasitology is neurocystic sarcosis. I invite Dr. K.N. Prasad sir to talk on the epidemiology with and description of Dirofilaria. Dr. Prasad, sir?
Yeah. Just, I'm audible?
Yes, sir. Yes, sir. You're audible.
Yeah, just I'm taking out my presentation.
Yes, sir. Yes, sir.
Good morning to you all. So, I thank chairpersons for nice introduction. So, I am going to talk on epidemiology with description of Dirofilaria. Now, you know that so many parasitic infections are emerging and re-emerging. So, the IATP decided to have symposia on emerging and re-emerging parasitic diseases. So, Dirofilaria is another emerging and re-emerging parasitic disease. So, it belongs to the order, it is a nematode, it belongs to the order Spirulida and family Onchocercoideae. The two most important species are Dirofilaria immitis and Dirofilaria rheopens, so far the human infections are concerned. And these two species are also most common in canine and feline species, and they are transmitted from canine and feline species to human through a vector, that is mosquito vector, belonging to the family Felicity. So there are certain non-canine species as well, and rare infections in the form of case reports have been reported, like Dirofilaria tenuis. One report from Karnataka is available on this. Then there is Dirofilaria uris, Dirofilaria subdermata, Dirofilaria striata, Dirofilaria honcollensis. So these are the other species that can cause human infection, but there are very rare and few case reports only. If you see, basically Dirofilaria was first discovered by Francisco Virago in 17th century, and he basically identified this one in the hearts of the hunting dogs, but he erroneously named them as Dictropina renale. Probably at that time the technology was not there to identify, but subsequently when the case was in detail studied and it was thought that probably it was Dirofilaria immitis, and ultimately it was identified and named as Dirofilaria immitis. So Dirofilaria immitis and rheopens, as I have told, that these two species are most relevant because they can cause sometimes severe pathology in humans, and they have got a high prevalence rate worldwide. D. immitis causes usually canine and feline cardiopulmonary dirofilaria, whereas D. rheopens causes canine and feline subcutaneous dirofilaria. So D. immitis causes human pulmonary dirofilaria, whereas D. repens causes human subcutaneous and ocular diarrhea. So usually D. repens infection is most common in human, and it causes subcutaneous and ocular lesions, especially the upper half of the body parts is most commonly affected. These two species are well adapted to human, canine, and feline hosts with immense genetic potential, because these canine and feline hosts, they are very neighbor to us, and sometimes we share the same premises with them. And the stray dogs and stray cats are also very much available in many of the developing countries, and they are free roaming, and they can transmit, as the vector is readily available in the developing countries, and the vertebrate hosts are also available, so the transmission is very easy, and probably many of the cases we miss because of the lack of studies. So life cycle diarrhea species comprises a definitive vertebrate host. As I told, it may be a canine or feline species, or maybe sometimes rarely human, and then a vector. Vector is especially female mosquitoes of the Parasitology family. Increasing incidence of human diarrhea are possibly related to changing climate. As you know that now there is a climate change, especially the temperature is going up, that favor the multiplication and development with parasites, and then change in human behavior as well as increase in vector density. So if you go for the life cycle of this Diarrhea, then you see that Diarrhea imitis and Diarrhea reopens. The two have a difference between their life cycles. Especially Diarrhea imitis, they causes cardiopulmonary diarrhea in canine and feline species. And the adult worms are available in the heart. They develop in the heart of the canine and feline species. Whereas the adult worm of Diarrhea reopens, they are available in the subcutaneous tissue of canine and feline species. Diarrhea imitis usually causes cardiopulmonary nodules or pulmonary diarrhea. We call it diarrhea because they can form nodule in different parts of the lung, especially in segments and segments of the pulmonary artery. And these diarrhea reopens, they can cause subcutaneous and ocular diarrhea in human. And in the heart, the female diarrhea, they produce the worms or the larvae, and these microfilaria usually they enter into the circulation. They can enter into circulation, and then in canine and feline species, again in the subcutaneous tissue, the adult worms they reside in the subcutaneous tissue, and the female worms they produce the microfilaria. The microfilaria can enter into the circulation, and from this circulation, when the mosquito bites to the vertebrate host, then they take up the larvae. This is called L1 larvae, and there is two molting occurs in the mosquito, L2 and L3, and this L3 is the infective form, and when this mosquito bites the next host, it may be a human or it may be canine or feline species, then L3 larva is introduced in the skin, and through skin repens, especially the adult worm develops in the subcutaneous tissue, and if it is Diarrhea imitis, then the adult worm develops in the heart of the canine and feline species. So vertebrate host, mostly it is the wild and domestic dogs, maybe stray dogs also, and canine is most common is the cat. Human is called non-permissive host or is non-suitable host. Why it is non-permissive? Because Diarrhea fail to reach the mature adult stage in human. As a result, the microfilaria are usually not detected. But there are one or two case reports where they have demonstrated microfilaria in human, but rarely usually it is not detected in human. Vector is female mosquitoes of the Parasitology family. There are more than 3,000 species of Parasitology family, and major vectors are Aedes aegypti and Anopheles, Culex, Culiceta, and Bensonia. So there are so many, around more than 2,000 species of the mosquitoes of Parasitology family. This larvae has been demonstrated. So as I have told that the microfilaria that enter into the circulation from the adult worm, female worm, they are L1, and when they are ingested by the mosquito, they develop into L2, around 8 to 10 post-infection days. And after 3 days, then again develop to L3. So stage change means basically the changes that critical from L1 to L2 and L2 to L3. Then L3 migrates to the mouth parts and is ready to infect the next host through bite. Mosquito deposits this infectious larva 3 in the skin wound, and here in the skin, the molting takes place from L3 to L4, and then from L4 to around 3 to 4 post-infection day, they develop L4. And L4, they develop to pre-adults 50 to 70 post-infection day and reaches the canine and pulmonary artery and right ventricles within 70 to 85 days, and they develop sexually mature to adult worms. And this occurs in cortisol in case of Dilophilaria immitis. In case of Dilophilaria repens, these adult worm, they usually reside in the subcutaneous tissue, and they develop here only. So if you see the female, they produce the microfilaria after 6 to 9 months of post-infection. They are ready to produce the microfilaria. And sometimes in the canine or feline species, you may have a primary stage, and especially it is thought that, especially when the female worms, they become aged, they may fail to produce the microfilaria. Especially in Dilophilaria immitis, the lifespan of female worms is 7 years, and in repens, the lifespan is around 2 years. So older females, they may not produce microfilaria. There might be single gender infection, then you may not get the microfilaria. And sometimes host immune pressure can lead to death or destruction of the microfilaria as soon as produced, or prevents the production of the microfilaria by the female worms. Dilophilaria also hosts symbiotic bacteria of the genus Wolbachia. As you know that in lymphatic filariasis there is Wolbachia. Similarly, the Dilophilaria also they have the Wolbachia. And Wolbachia is present in all stages of the Dilophilaria, starting from L1 to adult male and female, and more present in L3 and L4. And what this Wolbachia basically does, it assists in embryogenesis and molting of these larvae from L1 to develop adult. Each molting they helps. And what they get in return, from the parasite, they get amino acid for their own metabolism from the parasite. So this is symbiotic. It is helping in the embryogenesis and molting, and in return, it is taking amino acids from the parasite. So if you see the size and morphology of D immitis, so it is Dilophilaria immitis, they are longer and slender. You see that they are thinner, but D repens, they are shorter and broader, both male and female. If you see D immitis, I have told that adults have lifespan of 7 years, and in D repens, the adult's lifespan is 2 years. Microfilaria of D immitis, usually they can survive up to 2 years in dog, and they have thin cuticle, and microfilaria in D repens, usually they have not given how long but they're usually thought to be transient in cat, but sometimes they may be absent in cat as well. And they have thick cuticle, and the cuticle has got vertical regions with transverse striations. Microfilaria, again, the microfilaria of D repens is longer and slender compared to the microfilaria of D immitis. And D repens has a carved tail by which one can identify, and the rounded cephalic end, whereas the D immitis has a straight tail with spindle-shaped cephalic end. This is just one photograph of Dilophilaria from subcutaneous tissue of a dog. So you can see these male and female worms in the heart of a dog. And you can see that this is the D repens. They have got longitudinal ridges along with transverse striations by which one can identify or differentiate between the two species. So in human, immature D immitis, usually they cannot reach to the adult, so they are immature. They reaches maximum to the pulmonary artery and its segmental and subsegmental branches, and sometimes arterioles, and ultimately they die here, and they causes thrombosis, infarction, inflammation resulting in inflammation. Ultimately, this inflammation results in pulmonary nodule formation, and usually these nodules are single, but sometimes multiple nodules can be available, and they are differential diagnosis of so many diseases in pulmonary or lung diseases, especially when you go CT and X-ray, sometimes you will get these nodules as well. So sometimes it needs to be differentiated from other diseases, and biopsy usually is done and then it's identified. Pulmonary nodule, they are granulomatous reaction around the dead parasite surrounded by a wall of fibrous tissue. As I have told, that D-repens, usually they are available in the subcutaneous tissue and they die there ultimately in course of time, and they can form subcutaneous nodules. Sometimes the parasite may be in the nodule. Viable parasites are also there. The parasites maybe they are viable and they can form a nodule over there. Can reach the ocular regions as well. The D-repens, usually mostly around 65% of the cases, it is reported that they infect the upper part of the body, especially the face, eye, ocular, orbital, conjunctival, on the lid. They have been identified, and also subcutaneous tissue of the face and arms. Both species can also incidentally infect other regions of the body, like abdominal cavity, scutum, buccal mucosa, conjunctiva, eye, and intraorbital have also been reported. So if you see the epidemiology of this canine diaphylosis, canine, you told that canine diaphylosis is most common, or if exclusively, you can see that these countries have exclusively, especially North America, Canada, and then North America, Canada, South America, and Central America. The canine, they have exclusive, they have got the diaphyloria immunities. And if you see the Australia, they also have got the canine population has got the diaphyloria immunities, and it has also been present in some of the European and the African countries as well. Whereas D-repens is common in the part of the China and few European countries, and then some African countries. But in India, we have in canine population, we have both D-immunities and D-repens. Both are there. And one of the epicenter of both these canine filaria, the diaphylosis, is the Russia. You can see that Russia and different states of earlier United USSR, you can find this diaphyloria, both species are present there. So if we come to human diaphylosis, you can see that again the North America and South and Central America, these are the countries where human diaphylosis due to D-immunities is present. So they cause, I have told, they cause pulmonary cases. So usually they cause pulmonary diaphylosis, and it is present in the North and South and Central America. Whereas D-repens is present in Europe, it is present in some of the African countries, it is present in India as well as in Russia. So this is the these are the countries where you get plenty of cases of D-repens. And in Australia also you can get both the species. So if you see the diaphylosis in Europe, just in Europe, you can see that there are around 30 cases of diaphylosis, pulmonary diaphylosis due to D-immunities, and around 1,500 cases till 2011. You have subcutaneous or orbital diaphylosis due to D-repens. So since the D-repens is exclusive, it is found in the Southeast Asian, African, and the European countries. So it is known to be the worldwide diaphyloria. And D-immunities is mostly limited to America, especially in South America, North America, and America, and also Central America, and also it is reported in Australia. So canine diaphylosis, if you see in America, the prevalence is around 1 to 12%. In Central and South America, it varies from 2.3% to around 45%. In Canada, it's a colder climate, so it is less, around 0.2 to 0.8 or 8%. In European countries like Portugal, especially there's a Madeira island, it is reported up to 30%. Spain, especially Canary Islands, it is 33. These are humid and temperate areas, so they are very, development is fast, and usually find that the incidence varies from 30 to 40%. In France, again, it is not very much common, but one national mean is 1.3%. E-repeats are present, and you find that the around D-imities is 50% and 30% is your D-repeats. So this is canine diarrhea-filariasis. In Greece also there is diarrhea-filariasis. In Russia and the other USSR states, they have got the diarrhea-filariasis as well in canine population. So canine diarrhea-filariasis, it is reported from African countries as well, Australia and Asia. And D-repeats is an epicenter. Iran is another epicenter, and Sri Lanka is another epicenter. Around 61% of the canine population in Iran and 60% around Sri Lanka, they have got diarrhea-filariasis. So these two countries in the Asian region, you can say the disease is very much prevalent in the canine population. D-imities in Malaysia also very much common, around 70% of the canine population. Japan also around 45%, and Australia it is 3.5%. So if you come to feline diarrhea-filariasis, it is estimated that around 5 to 10% of those found in the dogs. In any area you find usually 5 to 10% estimated that you may have this infection in the feline as well. And hyperendemic region like Pro Valley region of Italy, it varies from 7 to 27%. Canary Islands of Spain, around 18 to 33%. Japan, it is around 2%. South Korea, 2.6%, and Australia, around 2%. So diarrhea-filaria has also been reported in wild carnivores like foxes, jackals, coyotes, wolves, lion, tiger, leopards, and black bears. So there might be many other species we need to look into. So if you look at the transmission dynamics of the diarrhea-filariasis, diarrhea-filaria is transmitted by diarrhea-filariasis transmission is influenced by two factors. One is human behavior in respect to pets. Approaches to management of pets, how we manage our pets, whether the pets have been given chemoprophylaxis, they have been vaccinated, if the pets are sick, whether the diagnosis is made or not, and sometimes travel to the along with the pets to the endemic region, sometimes imports of pets or dogs from the endemic region, relocation of pets in new residential areas. Especially it has been found that in US on the cyclone hurricane came, then the people from that area, they moved to the other areas, and even the transmission between the pets, healthy pets, and the relocated pets were observed. So climate factors especially helps in the colonization, development, and activity of the competent factors, especially rise in temperature and humidity. This helps in increasing the population, vector population, and larval development also depends on the temperature and humidity. If you see the temperature between 28 to 30 degrees, the larval development is fast. It takes around 8 to 10 days to develop the larva. Whereas if it is 24 temperature, then it takes around 11 to 12 days, and 22 degree around 16 to 20 days, and below 14 degree, it usually the development is arrested. But when again the temperature goes up, it again the larval development can take place. So global warming is a major it influences the parasitic development, especially the expanding amplification and development of parasites, then several of the vectors and lengthening of the annual periods of mosquito activity, thus increasing the transmission in many geographical regions. Urbanization, irrigation, deforestation, then artificial aqueduct systems, all these contribute to microhabitats that favor the vector population growth. So if we come to the Indian scenario, D-repeats is the most commonly reported. Almost, you can say most D-imities also have been reported, but in human cases only I found only one case of D-imities. Otherwise, most of the human cases are due to D-repeats. One case of D-tenuis has also been reported from Karnataka. But majority, if you say around 100 cases, 99% or 98% due to D-repeats. And some of the states like Kerala, the northeast, they are the epicenter of diarrhoeafilariasis. If you see the canine diarrhoeafilariasis repens, the prevalence is 7 to 24% in Kerala. Canine DMETs in Assam and Mizoram, it was 90% in pet dogs and 29% in the stray dogs. Kenny et al. In their review reported 33 cases of human diarrhoeafilariasis in India from 1976 to 2012. So there are 73 cases reported. Those are published, but many of the cases they remain asymptomatic and may not have been diagnosed. So from 2013 to 2018, again 24 cases has been reported, and this is the last report that I found on literature search. This is from Goa. They published that the review that there are 24 cases of diarrhoeafilariasis, and 23 were due to D-repens and only 1 was due to DMETs. So this is the last case from Goa, D-repens, oral submucosal buccal subbuccal nodule, and they removed this subbuccal nodule, and they found that D-repens was identified in that. So conclusion, diarrhoeafilariasis is a worldwide emerging zoonosis. You can see that in canine and feline populations, they are very highly prevalent in many of these geographic regions. Vector density, climate change, and high prevalence of dogs and cats enhances the chance of transmission. Diarrhoeafilariasis is underreported because majority of human infections are either subclinical or mildly symptomatic. It is estimated that almost more than 50% of the cases they have got subclinical or mildly symptomatic cases, and they don't report. Especially D-repens is common, and D-repens causes subcutaneous nodule. So we have many of these may have the subcutaneous nodule, and we ignore it because it does not have any much clinical impact. But only in case of cardiopulmonary or pulmonary cases, but pulmonary cases is caused by the DMETs, and DMETs in India we have not reported yet, but there are only except 1 or 2 cases. Otherwise, DMETs is not that common. So there might be that if DMETs is common in the canine population in India, then there might be that human cases are there, and they might be causing the pulmonary diarrhoeafilariasis. But we may miss these cases because most of these cases may be asymptomatic, or sometimes these cases are misdiagnosed as well, because we need to have a biopsy from the lung, and that is a very invasive procedure, and most of the centers they try to avoid. So sometimes the nodule, the parasite can die, and the nodule may remain with little or no symptoms. So most of the cases probably undergo undiagnosed. So increasing incidence has raised concern, and there is a need for systematic studies focusing on epidemiology, diagnosis, and prevention of the disease. So thank you very much for your kind attention. Thank you.
Thank you, sir, for the extensive update on the epidemiology of diarrhoeafilariasis. And I think in this era of having to beware of diarrhoeafilariasis and choose the canine and feline pets very carefully. And now I would like to move on to the next speaker, who is going to update us on the clinical presentation of diarrhoeafilariasis, Dr. Tapashi Ghosh. Professor Tapashi Ghosh has been engaged in the service of humanity for over 35 years. I'm just introducing you, madam. She's at present working as in charge of Helminthology section, Department of Microbiology, Calcutta School of Tropical Medicine, Kolkata and West Bengal. She attended and presented papers in various national and state conferences, published 18 papers in indexed journals. Parasitic diseases are the main field for research interest, and she took enormous interest and efforts under the leadership of Professor S.C. Parija as founder-president in starting the Indian Academy of Tropical Parasitology, West Bengal chapter in the month of December 2018. Over to you, madam, for the clinical presentation of
Thank you, madam, for your nice. Thank you, madam. Am I audible?
Yes, madam. Yes, madam.
Hello?
Go ahead. Yes, madam. You're audible. You can go ahead.
Thank you for your nice introduction, madam. And good morning, everybody. Greetings for coming new year to my extended IATP family. At the onset, I want to express my deepest gratitude to Professor Geeta Lakshmi, Honorable Chairperson, and the organizing committee of the 14th IITP National Conference for organizing and execution of such a wonderful conference amidst the threat of ongoing pandemic. I again thanks the respected chairperson and the organizer to be a part of the symposium. Professor K.N. Prasad sir has nicely described the epidemiological aspects and the description of diarrheasis. Thank you, sir, for your enriching talk. My topic. Share karo. My topic is clinical presentation of diarrheasis in human. From my end.
Can I see?
Can you see, please? Hello?
Not yet, ma'am. Not yet.
Not yet,
Not
madam.
yet. Can you see my presentation?
Can you see my presentation?
No, ma'am.
Not yet, madam.
Ma'am, you want to share the presentation, ma'am?
I will be shared from.
Ma'am, you want me to share the presentation,
Okay.
ma'am?
Yes, you please share it for me.
Yes, ma'am.
All the best, Tapashi.
I can see this presentation.
Ma'am, please wait. We are just loading.
Yes, thank you.
We are able to see, madam.
Ma'am, if you want to change the slide, say next, ma'am. I will change the slide, ma'am.
Okay, okay, okay. First slide, please.
Just a minute, ma'am. Just please give us a minute.
Can you start? I can't see my presentation actually.
Please wait. We are just sharing.
Can you see the screen, ma'am, right now?
Can you see the screen, ma'am?
Yeah, presentation is coming, no, Tapashi?
Yes, madam, you can proceed.
I think ma'am has left the meeting. Just please hold on.
After this presentation.
Can I join now?
Yes,
Yes,
ma'am, you are
madam.
joined, ma'am. You can proceed.
You can start your presentation, madam. Dr. Tapashi? Madam, Dr. Tapashi, please unmute yourself. You have muted.
Yes, yes. I am audible now?
Yes, now you're audible, ma'am. Yes, you can continue.
I can't see my presentation. Yes, I can see it. Thank you.
You can see. Yes, madam. Yes. All of us can see and you can proceed. We can hear you.
Next slide, please. As already been discussed, diarrhoephilariasis is a zoonotic disease caused by two subgenera of canine filarial worm. Most important members are Dilophilaria, Dilophilaria immitis, Dilophilaria nocitella, tenuis, and repens. Its transmission occurs through bites of various mosquitoes such as Anopheles, Culex, Aedes, etc. that act as vector for this filarial nematode. Most abundant subgenera of this nematode are specific to particular regions of the world. Dilophilaria immitis is essentially cosmopolitan in nature and is particularly prevalent in warmer areas of North and South America, Australia, Japan, Europe. Dilophilaria nocitella repens and tenuis is exclusive to Old World, that is Southeast Asia, especially China, India, Africa, and part of Europe. Next slide, please. Next. Although canines like dog, cat, and jackals are the most important natural host, primary concern to us is human dilophilaris, caused by transmission of disease to human via mentioned host through the vectors. So, in canines, cardiovascular lesions may result in serious complications like death due to congestive cardiac failure. Next. As reported from all over the world, in human, commonly diarrhoephilariasis is either pulmonary or subcutaneous infections, but there have been few reports of human diarrhoephilariasis in unusual sites like large vessels, mesentery, peritoneal cavity, spermatic cord, or liver as well. Next. Coming to the life cycle. The life cycle of diarrhoephilariasis initiates with the vector drawing blood from an infected host and transmitting it to a healthy adult. Next slide. Mosquito bites transfer stage 3 filarial larvae into the skin of definitive host and or accidental, that is the dead hen host like human. It penetrates into the wound and molds into the stage 4 larvae and finally adults. This adult filarial worm reside in the pulmonary vessels. The female nematodes produce microfilaria which enter peripheral blood. Next slide, please. Mosquito ingests the microfilaria during blood meal, but maturity usually does not take place in human. Only 2 cases have been reported of isolation of gravid female worm from subcutaneous nodule. After ingestion, microfilaria migrate to the Malpighian tubules, then develop into stage 1 larva, which subsequently molds into infective stage 3 larvae. The stage 3 larva then enter mosquitoes proboscis and infects another definitive host during the next blood meal. Next slide, please. Next. Previous one, please. In human, previous one, please, the previous one. Previous to this, no, not this one. The next one. Okay, I'm just continuing. In human, the Dirofilaria immitis larvae end up in lung, where they lodge in small caliber vessels, causing infarct and typical coin lesion visible on radiograph. Actually, the slide has been little bit reshuffled. Okay, I'm continuing. Coin lesion, as well we know, refers to a rounded or oval, well circumscribed solitary pulmonary lesion, usually measuring 1 to 5 centimeter in diameter. Calcification may or may not be present. We commonly use this term for describing the solitary primary or metastatic lung malignancy, primary pulmonary hematoma, bronchial adenoma, etc. And the parasitic causes are often ignored. Most human infections are asymptomatic and discovered only if chest imaging is. Please, next slide. Switch on to the next slide, please. Next one. Yes. So this is the coin lesion we are showing here. This I've already described the coin lesion, which is comes in the differential diagnosis of primary solitary malignancy of lung and the pulmonary hematoma, bronchial adenoma, but the parasitic causes are often ignored. Next slide, please. Next one. Yes. So most human infection. Oh, this is the radiograph, picture of radiograph showing diarrhea filariasis patient showing nodular lesion in the right lung found incidentally on the chest X-ray. Chest CT scan demonstrated a solitary pulmonary nodule in the right lower lobe. PET scan also revealed a small subpleural nodule at the right lower lobe with minimal increased FDG uptake. Next slide, please. Next one. Coming to the subcutaneous dirofilaria. No, previous one. Previous one, please. After pulmonary dirofilaria, the most important clinical diagnosis is subcutaneous dirofilaria caused by Dirofilaria tenuis and repens. Adult worm can develop in human, but sexual maturity and the production of microfilaria does not occur. Lesion consists of a coiled, degenerating worm in subcutaneous tissues, typically around the eye or the genitalia or limbs. Nodule can be erythematous, tender, and associated with an abscess. There may be concomitant allergic symptoms, including urticaria and fever. Next slide, please. These are the few pictures showing the clinical representation of subcutaneous dirofilaria. First few pictures, that is the lesion, subcutaneous lesion on the eyelids. The worm is coming out from this lesion. And in the right part, there is a serpentine subcutaneous lesion showing the worm inside. And after dissection of the temple of the forehead, the warming also coming out from the subcutaneous nodule on the forehead. Next slide. Yes, that is the lesion on the nasal part, on the over the thigh, on the finger and the nape of the neck. The different area, almost the exposed areas, are primarily affected. Next slide, please. Next. Yes, this is a subcutaneous cystic nodule excised from the scrotal sac. Next slide, please. This is the deep muscular lesion, although little bit unusual, but the Dirophyllaria immitis has produced such deep muscular nodule which has been excised later and dissected, bringing up the worm from the cystic lesion. Next slide, please. These are the already Sir Prashad has described this gum region, this buccal portions, and the soft palate lesion, submucosal lesions, where the oral dirophylariasis can be visualized well in these pictures. Next slide, please. This is the ocular dirophylariasis, often common with this subgenera ripens and emmetis, tenuis, sorry. And this is the subconjunctival region where this dirophyllaria worm can be isolated or taken up. Next slide, please. So, hence, summing up the entire clinical feature, thus most of the cases are undiagnosed. That is to be emphasized. Hence, there is a need to increase awareness about this parasitic infestation. Ultrasound imaging, radio imaging confirms the diagnosis, and surgical excision of the cyst is treatment of choice. Field-based epidemiological studies among human and also in the domestic cattle and pets are necessary for understanding the magnitude of this disease. There is evidence of Dirophyllaria ripens, which is spreading faster than Dirophyllaria immitis from the endemic area to the adjoining region. So and last but not the least are the entomological investigations like vector species abundance. Infection of Dirophyllaria among them is also very, very important to be emphasized. Next slide, please. So finally, I extend my heartfelt gratitude to the following people for their contribution and continued support and assistance. Professor Parija Sir, Vice Chancellor Sri Balaji Vidyapith Pondicherry, founding President of IITP. Professor Geeta Lakshmi, Founder, Vice President of IITP, presently present as Vice Chancellor, Dr.M.G.R. Educational and Research Institute. Thanks to Professor Ujjala Ghoshal for being continued support all along. Thanks to Dr. Ardhendu Kumar Majhi, Faculty, Department of Microbiology, in charge, Protozoology Section, Calcutta School of Tropical Medicine, Kolkata, and Srimati Moitreyi Chatterjee, the project scientist of this Parasitology section of STM. And lastly, it's an honor to present in the symposium sitting in the Temple of Tropical Medicine in India, representing the Parasitology unit from Calcutta School of Tropical Medicine. Thank you.
Thank you, Dr. Tapashi. Yes, we could feel the pride that you have in being patronized by so many people, great scholars. And thank you so much for the plethora of pictures that you have shown. It as I think all of us would never forget to see and look for these subcutaneous nodules in or the cyst or tissue or the worm. in the eyes, the oral cavity, the scrotum, and the neck, or anywhere else. I think there should be a complete examination of the skin and look for these worms and think of dirofilaria with a history of contact with dogs, cats, and mosquito bites. Thank you so much for the very good presentation.
Thank you, madam. Thank you all.
Was really a feast for us to see.
Thank you very much,
Thank
Dr. Tapasi.
Thank you, Dr. Tapashi.
Can I talk?
I would like to go to the next speaker. Dr. Sumita Kurana, Institute of Medical Education. Yes. Yes, sir. Yes, sir. Yes, sir. Hello, madam. I think I'm muted.
Yeah.
Nice.
Can you see my screen now? Can you all see my screen now?
Can you introduce your family?
Dr. Abhijit, can you see my screen?
Yeah, we can see the screen, ma'am. You can proceed now.
Yes, ma'am. Yes.
Yes, Dr. Sunita, I just introduce you before you start your session.
Yeah.
Yes, Dr. Sunita Kurana is professor in the Department of Medical Parasitology at Postgraduate Institute of Medical Education and Research, Chandigarh. She completed MBBS from Indira Gandhi Medical College, Shimla, and MD from AIIMS, New Delhi. She is Fellow of Indian Medical Society Science Academy, Fellow Indian Academy of Tropical Parasitology, WHO Fellow at Karolinska Institute, Sweden. She's published more than 100 papers in peer-reviewed national and international journals of repute. She's joint editor of Journal of Tropical Parasitology and reviewer of many journals. Her area of interest includes opportunistic parasitic infections, emerging and neglected parasites, ocular parasitic infections, gastrointestinal and genitourinary pathogens. Over to you, Dr. Sunita. I request Dr. Sunita to update us on the diagnosis, lab diagnosis of dirofilaria.
Thank you, madam. Thank you so much for the nice introduction. And at the onset, I would like to express my heartfelt gratitude to IATP President Dr. G.R, Dr. Geeta Lakshmi, and for the whole organizing committee, Dr. Subha Lakshmi, who have been taking all the effort for the effortless streaming and conduct of this conference. Though a virtual one, it's a pleasure to be part of this conference always. And coming straight to the topic, my topic is emerging zoonotic infections, the diagnosis of dirofilaria. It's in this current scenario of COVID, which is also a zoonotic infection, the topic is really well suited. And as Dr. Parija said yesterday, that the next pandemic might be that of a parasite. So let's better be prepared for the same. So this has already been introduced by Dr. Prasad and Dr. Tapasi very well. Just mentioning that Dirofilaria gets its name from a Latin word dirus, which means fearful, and phylum means thread. And these worms are really very long, so that makes them easily identifiable amongst the plethora of worms that we have coming out of the human bodies. As already mentioned by previous speakers, the most common species are Dirofilaria immitis, causing mainly pulmonary dirophylosis, and subcutaneous dirophilariasis caused by Dirofilaria repens and tenuis. Now, there are questionable paradigms related to human dirophilariasis, because basically there were three concepts of human dirophilariasis. That the first one is that Dirofilaria immitis is associated with pulmonary nodules, and Dirofilaria repens is associated with subcutaneous nodules and ocular lesions. Second was that human infections are caused only by immature worms, or in other words, the worms never get mature or gravid in the human body. And third, that human infections are sporadic and accidental. However, these paradigms are currently changing. As a consequence of the data obtained in the last 10 years shows that the epidemiology and everything is changing regarding Dirophilariasis. And a number of species are being incriminated in human infections, which include the Dirophilaria ursi, Dirophilaria subdermata, striata, and a new species which has been identified from Hong Kong, that is Dirophilaria hongkongensis. And these species are basically identified on the basis of their size, and obviously on the molecular basis also, but they are distinguishable on the basis of their size also. The size of female worms is larger than the males, and the range is vast, ranging from 100 millimeter long to almost 700 millimeter long, and they are also more wide than most of the filarial worms that we see in human body. That is, they are thicker than the filarial Brugia, Wuchereria, Onchocerca, Loa, etc. Another characteristic feature that has already been described by Dr. Prasad is that they contain characteristic cuticular ridges, which are not seen in other filarial worms, which differentiates them from other subcutaneous filarial worms that are isolated occasionally from human bodies. And these characteristics, these longitudinal and circular ridges are very characteristic of species also, not just of the genus Dirophilaria, but they also discriminate between species. The only exception of absence of the only exception is that in Dirophilaria immitis there is no external longitudinal ridges. So, as already described by Dr. Prasad, the Dirophilariasis in animals is the norm, and human infections are the exception, but they are emerging infections. And even in animals, the infection is maybe asymptomatic a lot of times. Like we see asymptomatic microfilariemia in humans. Here also in animals also, dogs, the major host, the canines, the felines may be asymptomatic, but when symptomatic, they cause pulmonary artery blockage leading to an illness that include cough, exhaustion upon exercise, fainting, coughing up blood, and or the other species may lead to subcutaneous asymptomatic infections in animals. As far as humans are concerned, if you look at this picture, the heart of this is the heart of a dog, which is showing a lot of worms which have been isolated from this heart. And since the worms are gravid in their natural host, they produce. So this is a blood smear showing microfilaria in blood of a dog. So it is easy to diagnose infections in their natural host by demonstration of microfilaria from blood, or by detection of antibodies or antigens in the blood, like we do for Butyraria and Brugia in case of humans. But now the dirophilariasis in humans is not easily diagnosable, because the microfilaria are not seen in the peripheral blood of humans, because the worms never mature enough to a stage where they can start producing microfilaria. So that is the biggest problem in that. That is the biggest reason why the infection, the diagnosis of dirofilaria infections in humans is not as easy as other filarial worms. Now, a clinical presentation has already been described by Dr. Tapasi, and it is she's clearly shown so many slides where where it has been the worms have been, the lesions have been shown on the eye, or on the face, in the temple region, in the genital areas also, and also the pulmonary lesions or pulmonary nodules presenting as coin lesions. She's shown a lot of beautiful pictures. In some more of these subcutaneous, subocular, subconjunctival nodules, nodules in the periocular region, the supraorbital regions, and the worms coming out. Now, what you see in these lesions are the adult worms which come out of these lesions. And in case of pulmonary infection, dirofilaria infection, it is in humans also the infections may be asymptomatic, and only during a workup of some other infection, or because they will form the differential diagnosis of malignancies and other infective syndromes, the dirofilaria infection may be diagnosed, because the dirofilaria would be present as coin lesions, would appear as granulomas appearing as coin lesions, which may contain either live or T-generating worms. Following embolization, some patients may present with vague systematic symptoms like malaise, fever, chills, and respiratory distress also. So here is a picture Dr. Tapasi has already shown, showing coin lesions. And the figure 2 shows the thoracoscopic evidence of unusual coil pulmonary lesion. Now, mind you that these coin lesions, though are seen in dirofilaria infections, but they may be seen as in other infective and non-infective etiologies like tumors also. So it is important to keep in mind other differential diagnosis also, and but ruling out dirofilaria would also be another important point. But the diagnosis is confirmed by identification of worms, which are mostly immature, because I said that worms cannot, it is a zoonotic unnatural infection in humans, so worms do not really mature in human beings. And most of the times diagnosis is made by identification of worms in surgical or autopsy specimens. I already emphasized that microfilaria are not found in the blood. Only 4 such human cases have been reported where microfilaria were demonstrated in the peripheral blood of humans. The other modalities which are used to diagnose dirofilaria infection in humans are serology and lately molecular diagnosis. The other supporting data include the supportive tests include presence of eosinophilia, which is of moderate grade and is seen in about 20% of cases of pulmonary dirofilaria, and some of the cases of ocular and subcutaneous dirofilaria may also show eosinophilia. And presence of eosinophilia points towards the parasitic etiology of the lesions. Now, histopathology is the mainstay of confirmation of diagnosis and examination of tissue from areas of inflammation. If it is a case of pulmonary dirofilaria, then it would be the lung tissue, or else it would be this subcutaneous tissue or the nodules. And the characteristic feature tissue is the presence of worm in the inflammatory region, in the presence of granulomatous lesion. And dirophilaria are identified where they are thick laminated cuticle, broad lateral ends, and large muscle cells. Now, this large muscle cells, presence of these large muscle cells, presence of lateral ridges, and a thick, smooth laminated cuticle is very characteristic of dirophilaria and can easily be identified by expert eyes on histopathology sections. However, they may be missed by the naive pathologists because this is not a very common infection, and normally people are not really trained to see the dirophilaria in histopathological infection. Some special stains can also be performed because the cuticle of nematodes contains chitin, which can be stained with non-specific whiteners such as calcofluor white. So some special stains can give a clue towards presence of a parasite in these lesions, and then the parasites can be narrowed down to dirophilaria by presence of characteristics that I already emphasized previously also, up to the species level also. However, a successful worm identification depends on several factors like degree of worm decomposition, which makes their identification more difficult, similar morphology of cuticles of various species. In addition, some key features that are considered specific for worm species identification, including cuticular ridge size, ridge numbers, ridge to ridge distance, are highly variable at different areas of the same worm and even within a same transverse section, thereby rendering these criteria sometimes unreliable for diagnosis, and only expert eyes may be able to pinpoint the parasite per se. However, this is a picture to show how the various characteristic features of dirophilaria can be seen on sections. And if you look at this arrow, this shows the cuticle. This is the cuticle, and these are the ridges. And you see this is the rectangle shows the ridges, and this circular thing which with the cross in the middle is the gravid uterus containing the ova in the mature in the gravid female worm. And this is the longitudinal section where you can see the gravid uterus and the cuticle. Similarly, you can see the tissue dirophilaria species in excisional biopsy of testes in 1-year-old boy with paratesticular swelling, and you see a lot of nodules containing the worms inside these nodules. Another thing that can be performed is the immunohistochemical staining to confirm. Now, since these filarial worms carry Wolbachia bacteria, which is essential for the fecundity of the parasite, as well as and in the bargain, Dr. Prasad also told that in the bargain the bacteria gets the essential amino acids. So these immunochemical staining to confirm the existence of Wolbachia or its molecules can also be used to confirm the presence of filarial worms in the tissue sections, and a positive reaction indicates the presence of dirophilaria. However, for all these things, one has to obtain biological material or a biopsy material. Now, serology, which is very important in case of animal diagnosis, is not so well developed for human diagnosis because no such commercial kits are available for human use. And though it is known that humans demonstrate a strong antibody response, so most of the studies have been in the research on the research institutes, and different antigen complexes have been used to detect antibodies and diagnose human dirophilariasis. In some studies, it has been shown that the problem is that these serological tests employing the crude antigens cross-react among different Dirophilaria species as well as with the other parasitic elements of humans, especially Toxocara canis, which is a cause of visceral larva migrans and can present as similar clinical presentation. The use of recombinant antigens improves the improves the specificity of the diagnosis, and a recombinant antigen using the cloned gene encoding DI35 and 35 KDA previously identified and cloned antigen has shown high sensitivity and specificity for diagnosis of Dirophilaria infection. And also for human pulmonary dirophilariasis, and from sera of patients showed some antigens by Western blotting in the range of 22 kilodalton, which proved to be an excellent biomarker for pulmonary dirophilariasis. And this protein exhibited in a study, it was shown that this protein exhibits 100% sensitivity and 90% specificity, with positive and negative predictive values of 75% and 100% respectively. Then even in case of D. repens, mind you, is the species which is mostly reported from India. Most of the cases, more than 95% of the cases of human Dirophilaria infections reported from India have been reported to be due to Dirophilaria repens, which produces mainly subcutaneous and the ocular lesions. And for D. repens, certain specific polypeptides have been identified by Western blotting, and which allow discrimination not only between subcutaneous Dirophilariasis, but also other parasitic and non-parasitic diseases. So antigen detection, one such commercial antigen detection kit is available for use in canines. However, this has not been used or evaluated for humans because the human cases are so far and so few that it is difficult to have a proper prospective study designed to analyze some antigen or some antibody detection tests. However, this is widely used witness Dirophilaria manufactured by Symbiotic San Diego, USA is being used for detection of Dirophilaria circulating antigen in serum of canines. And now with the advent of molecular tests and the capacity building of almost all the labs, now COVID has given an advantage to microbiologists by enabling most of the microbiology labs to perform molecular tests. And now molecular tests are going to be play a major role and may be mainstay of diagnosis of many parasitic infections. And some studies have been conducted wherein Dirophilaria species have been detected by use of PCR techniques with good sensitivity and specificity. And the regions most commonly used for diagnosis include the small subunit RNA or the COX-1 gene. And wherein in this particular study, the 479 base pair product was produced as a result of PCR amplification of the COX-1 region of the tissue obtained for Dirophilaria diagnosis. And another study from Iran has shown the molecular characterization of ocular Dirophilariasis, wherein they used again a COX-1 gene fragment amplification of a 680 base pair fragment, and they could identify Dirophilaria immitis with using the sequencing of this product. And then another species, a novel species, could also be diagnosed based on the molecular analysis of the specimen, and this is named as Candidatus dirophilaria hongkongensis, because till as per my knowledge, it is still not approved as a new species, but hence the name Candidatus, because it is a candidate for naming the new species. And now a study from India has been published in 2019, which states whether Dirophilaria repens parasite from South India belong to zoonotic Candidatus dirophilaria hongkongensis. So in this particular study, they have they took live worms from swellings or nodules in subconjunctiva or subcutaneous tissue from humans. 3 human samples were used, 4 dog samples were used, and one jackal sample was used, and PCR targeting a repetitive segment of 18S ribosomal RNA and COX genes were used, and sequence analysis of 18S ribosomal RNA gene revealed the closest identity up to 98 to 99% with an already published sequence of D. repens. However, based on the sequence of phylogenetic analysis of COX gene, the Dirophilaria infected both animals as well as humans native to Kerala were identified as genetically conserved and closely related to Dirophilaria hongkongensis. Hence, the results of that particular study suggested the existence of Candidatus Dirophilaria hongkongensis in Kerala, South India, in canines and humans. So molecular techniques and molecular diagnosis has a lot of role to play in not just detecting the presence of particular parasites, but also identifying them up to species and genotypic level also. So with these, I end my talk, and thank you very much once again.
Thank you very much, Dr. Sumita. You have gone through all the areas of diagnosis, including macroscopic, depending upon the length of the worm from 100 to 700, the microscopic view of the microfilaria, and the radiological diagnosis with coin-shaped lesions. Then you have gone into the histopathology was the main stage of diagnosis. The role of the through the angiogenes, which may give a PCR, wherein the 479 base pairs has been the diagnosis, and also you have updated us on the latest emerging species of Dirophilaria, which is Hongkongensis. And thank you so much. You have really created a great lot of knowledge to all those who are listening to you. And thank you so much, Dr. Sumita Kurana.
Thank you, madam. Thank you.
Now I would like to think, yes, any doubt?
Yeah, Dr. Sumita, thank you for the very good presentation. And I just have got an observation to make. That I have one observation that it seems that the first report of this Dirophilaria hongkongensis is from a patient from India, and the and as if and it was diagnosed in Hong Kong. So those hit the name as Hongkongensis, but actually it should have some relationship to India. But that name has not been finalized by the International Systemic Society. So I think the name may change because it was first discovered in India. So it has to, the name should have some connotation. Identified in Hong Kong does not mean that, because normally we give the name of any parasite, any virus area. So just an observation that I want to make.
Thank you for updating, sir.
Thank you so much, Dr. Abujia Chowdhury, for the information, and let us hope if the name changes, we'll just update us. Thank you, sir. Thank you, madam. So over to Dr. Saurav Maishi for his next presentation on the treatment and prevention of Dirophilaria. Dr. Saurav Maishi is presently the Chief Consultant, Clinical Microbiologist and in charge, Department of Infection Prevention and Control, Institute of Neurosciences, Kolkata, India. Dynamic and academically sound with technological fluency. He's currently heading the Department of Infection Prevention and Control. Yes. He is the proud mentor of the Namaskar Drive, which has claimed praise of CDC. He received Quality Council of India's Quality Month Award in 2017 and 19. He has extensive experience with neuroinfectious diseases, infection control, leishmaniasis, and cryptococcosis. He has completed training on online marketing, advanced clinical immunology, and FDA good clinical practice. Over to you, Dr. Saurabh. We look forward to your talk on treatment and prevention of diarrhea.
Thank you, ma'am, for this nice introduction. And hello, am I audible?
Dr. Saurabh, you're unmute yourself. Yes.
Am I audible? Hello.
Yes, sir. Yes, sir.
Yes, sir.
You can continue.
Okay. Firstly, I would like to thank you, the chairperson, ma'am, and also the organizing team under the
Yes,
great
you're audible.
leadership of.
Yes, you're audible.
Yeah. So I would like to express my heartfelt thanks to the chairperson and the organizing team under the able leadership of Dr. Geeta Lakshmi and Professor Parija, and the whole umbrella of the team actually. And I came to know that this conference is being organized within a very short time, only within a fortnight. So that is a great commendable job. And I will start sharing my presentation. Hello, it is visible now.
Yes, sir. It's visible.
Okay, so my previous speakers have given a very good overview and the intricate details of the diarrhea, and it makes my topic very easy because ultimately things come the treatment and prevention. And as we have known, the treatment is very limited. Even have to see what it has to offer us. So, diarrhea belongs to a vector-borne zoonosis in humans, and as we can see the whole spectrum, the vector-borne zoonosis comprises of mainly the protozoa, of which helminths comprises of a very low part, but zoonotic filariasis is a major chunk of this thing. And if we see the agents of zoonotic filariasis, diarrhea stops. And here is a cartoon that shows the male diarrhea calling to the female that, my darling, ever since we departed that mosquito and filled up this heart with love, I knew we would be together until the end. So this depicts actually the life cycle, because ultimately the male dies, and we can see the female counterpart is telling that the mouth is on the opposite side because they are so long, like approximately 30 centimeter in length and 1 millimeter in breadth. So we can see the minute thread work here. So talking to the prevention and treatment, we can see the fallen decks which needs treatment, and we need to protect the other non-fallen decks. And human diarrhea treatment is very easy to see. In pulmonary, there is no need to treat the case because it is self-limited, and often the parasite dies, and rarely it is symptomatic. So it hardly needs any treatment, but the differentials are very much problematic and dangerous, ranging from pulmonary malignancy, histoplasmosis, and other granulomatous diseases. So they often need the excision, and that ultimately reveals the diagnosis. Otherwise, there is no treatment for the pulmonary human diarrhoeafilariasis. In case of subcutaneous or ocular, the extraction of the nodule or the worm is the choice. In case of animals, surgical extraction still has a role. In case of dogs, which can cause a fatal Keval syndrome, wherein the worm gets lodged into the tricuspid valve. And if the emergency extraction is not undertaken, it ultimately eventually leads to the death of the dog. And in cats, they mount strong immune response, and surgical extraction, wherever feasible, has to be undertaken. Pharmacologic options are mostly under purview of the animal population. In human, there are no trial or nothing useful pharmacology that is there. And for the mainly the veterinary population, the goals of heartworm treatment is to improve the clinical condition of the animal and to eliminate all the life stages of the heartworms with minimal post-treatment complication. Dogs are usually affected and they exhibit significant clinical signs. As you can see, they can present as congestive cardiac failure, and they should be stabilized before administering an adulticide in form of glucocorticoid, diuretics, vasodilators, positive inotropic agents, and fluid therapy, because the canine cardiopulmonary diarrhoeafilariasis is often fatal. And FDA has approved only one adulticidal drug, that is melarsoline, and that is administered via deep intramuscular injection in the belly of the apexial lumbar muscles in the canine population. This figure shows how the injection is being done into the belly of the apexial muscle, and it is between the third and fifth lumbar vertebra. And we came to know that there is a Wolbachia that is endosymbiont in the diarrhoeafilaria, which we often know in relation to our Wuchereria filariasis. But this Wolbachia was first identified in relation to diarrhoeafilaria immitis only through electron microscope. And as Dr. Khurana told, they are involved in their fecundity, so an adjunct therapy with doxycycline is often helpful to control the infection. And this doxycycline therapy lasts for 4 weeks. A macrocyclic lactone preventive should be administered for 2 months prior to administering melarsoline to reduce new infections and eliminate existing susceptible larvae. And still the additional doxycycline is helpful. Non-arsenical based treatment in which melarsoline is not feasible or the dogs are allergic, that take longer time to kill the adult worm. In cats, we can see the immunological response is much more. So we need to diminish the immunological flare-up by use of steroids. So often the prednisolone is required, and it has to be initiated whenever antibody and or antigen positive cats display clinical signs. And adulticide is not recommended because it is obviously fatal and toxic to the cats, even at very low doses. So there is no scope for adulticide in cats. The American Heartworm Society has been working long because this America has got lot of problems, diarrhoeafilariasis recognized, and this society are working, and their treatment protocol, they have advised that pre-treatment with an macrocyclic lactone and doxycycline, then a month-long waiting period, followed by 3 doses of melarsoline on day 60 and 2 doses 24 hours apart. So In short, the treatment of animal diarrhea consists of 3-dose regimen of melarsoline, and in cat, extraction of the nodule or worm, and a spontaneous cure is often wanted if the cat is not showing any overt clinical signs. In the European Scientific Council Companion Animal Parasites, the SCAP, they have also come out with their guidelines. They are of a bit older, it is of 2019, whether the American Heart Society have the 2020 guidelines. The SCAP also shows 2-step treatment to reduce the risk of pulmonary thromboembolism, because that is the main fatal factor in the canine population. And complication due to pulmonary thromboembolism should be reduced by the restriction of exercise following treatment. So during the treatment, they have to have a rest, and administration of heparin and corticosteroid are helpful. And they have also advice for the doxycycline. Now coming to the prevention, we can know the prevention is of 3 levels. First is the primary prevention, where we prevent disease and harm before they occur by vaccination. However, there is still nothing in case of diarrhea. So only the secondary prevention by screening and early detection, particularly in veterinary population, and tertiary prevention by the treatment and rehabilitation are the things we have to go for. But as we see, the human rarely needs treatment. So, and mostly it's a veterinary aspect that we are discussing. So, do we really need prevention from diarrhea? We are telling that it's an emerging helminthiatic, emerging zoonosis, but do we really need? So, we can see human pulmonary diarrhea presents the coin lesions of diameter 1 to 4 centimeter. And the US data shows this coin lesion can be seen in 1 in 500 chest X-rays. So there will be at least 1.5 lakh patients per year having this coin lesion, not due to diarrhea, but due to other things. And what happens, this 74% of this pulmonary coin lesion or solitary pulmonary nodules are asymptomatic. And interestingly, 27% are the malignancy. And if we see the radiology cannot tell it the true malignant nature. They can assume and can refine their diagnosis. As we can see, the 25 to 40% of the malignant nodules can appear benign. So whenever there is a pulmonary nodule in an asymptomatic patient, and obviously the other supportive diagnostic modalities like complete blood count will be normal, the liver function test will be normal, the ANA profile will be normal, and the microbiological investigation like the sputum, the tracheal aspirate, bronchoalveolar lavage, they will also be negative in both the cases, whether it's a malignancy or it's a diarrhea. So ultimately, for the patient benefit, we have to go for a excision. Now, what we can see, do we need actually excision? Because the 5-year survival for a bronchogenic carcinoma is 15%. Whereas if it is resected, the 5-year survival increases to 50%. And if it is less than 1 centimeter, the 5-year survival is 80%. So ultimately we need excision because that is a more dreadful thing of a malignancy. Again, that in human pulmonary diarrhea is unnecessary actually, because it is self-limited. And if we see the mortality of thoracotomy, it is 0.3 to 7.3%. So it is not less. Thoracotomy itself can lead to a mortality. So in case of human pulmonary diarrhea, it's often unnecessary thoracotomy. So we actually need prevention to exclude these things. And as my previous speaker has told, the different species, the major distributions, and we can see the recognized vectors. lots of mosquito species, including Aedes, Anopheles, Culex, Culiceta, Saurophora, are all involved in the transmission. And along with the mosquitoes, black fly of Simoleum species can cause the subcutaneous dietofilariasis by the spread of Noctiella rc and Noctiella subdermata. And we can see the definitive host here, usual. The dogs, cats are the common one, but raccoons, panthers, bobcats, bear, porcupines are the wild hosts and which are probably beyond our reach. So the mosquito spreads these 4 dietophilaria, Dietophilaria immitis, repens, tenuis, and strata, and blackfly, the Dietophilaria orsi and subdermata. And this picture clearly shows the line of control, which we can control, the canine and the feline population. The wild population demonstrated studies in red foxes, but other animals are actually beyond our reach. Only accidentally we are diagnosing them. Now, why the dogs are being talked so much? Because there is a basic difference between a dog and a cat in relation to dietophilaria. Dogs, we can see the Scooby-Doo over here. Perfect example is attractive and tolerant to the mosquito bites. They have long survival with large, huge worm burden. Different species can be harbored by a dog at same time, and they provide microfilaria to conditions of activity and also the large surface area. And the cat is like that. They have immense host resistance, they mount strong immune response, and actual dietophilaria makes a lots of effort to become manifest because they have long preparation period of 8 months. And the microfilaria is also short-lived and of low level. And also the cat has short lifespan of adults in 2 to 3 years only. So the cats are less favorable than dogs in spread of dietophilaria. Now, definitely the changing epidemiology is there. The geographic extension of the vectors, as we have seen, are no more confined to the Americas and the Europes. And there is increase in helminth population density because of increased veterinary dietofilariasis. And availability of suitable hosts, vectors' adaptability to different environment, vectors' behavior and preferences, and the climate change largely has actually favored dietofilariasis to spread like a wildfire. How the climate change has helped it? The global warming, of which it's very much problem, has decreased the extrinsic incubation period. It has broadened the colonization areas, invasion of the new areas. And this warming actually increased the vector capacity in terms of their physiology. And these are the growing degree day models which survey the vector population and relate to their dietofilariasis prevalence. They are based on the threshold of 14 degrees centigrade for dietofilaria development in vector, 130 growing degree day required for larvae to reach infectivity, and maximum 30 days life expectancy of the vector. And we can see here in a model of central Italy how the prevalence are linked with the growing degree day models. In India, we do not have these maps. Earlier, we have one map for the lymphatic filariasis of this kind, but for dietofilariasis, the endemic regions are now going to implement this, and with a GIS system mapping also. And another problem is the sympatric occurrence of both diurnal and nocturnal biters, the culex and Aedes. Both are involved. What they do, they increase in vector-host contact plus the filarial worm carriage. The culex pipiens in Central and North Europe are behaving like the Southern Europe now. In Southern Europe, they were the active searching of the human blood outdoors. And this pattern actually confirms to the spread of canine dinofilariasis in the Central and North Europe from the Southern. And the recent data shows that highly mammophilic and anthropophilic mosquitoes are infected with Rhizophilidae immitis in nature by PCR studies. And in Europe, these are the 6 mosquito species. They are found to have newly infected with Rhizophilidae immitis. The Culex theileri, Anopheles maculipennis sensulato, Coculi latidia richardi, Edimorphus vexens, Daliana geniculata, and Oclerotatus caspias. So ultimately, we have to prevent the mosquito bite. And the mosquito nets, repellents are of our personal protective measures, and the breeding control insecticide are actually on a large scale, has to be there. And for a traveler, restricted travel is probably an advisable thing. And one thing that this mosquito, they often breed on the stored water, whereas the black fly, they actually breed on the flowing water. So the same things will be applied to the black fly also, the source control, the insecticides. But ultimately what we need are preventive veterinary surveillance, because that will tell us to what extent we have to go and how. For dogs, actually, the annual antigen testing along with microfilaria testing is recommended over 7 months of age of the dog. And a year-round administration of preventive drugs, mosquito repellents, and ectoparasiticide has been endorsed by the American Heartworm Society. For cats, preventive medication for all cats, including kittens in endemic areas, has been advised. And for the preventive therapy, the monthly ivermectin or milbemycinoxime at the oral formulation, and topical moxidectin and selamectin, they can be used also. The dog and cats have different doses. These are actually the macrocyclic lactones. So to end, the American Heartworm Society has brought out different beautiful posters that our dog's heart is a place for love. So it is probably for beneficial for not only the veterinary population, but for us also. And finally, we started with the first, the decks are falling and we have to protect the other decks. But if we realize the real impact of the veterinary dinofilariasis, we can see a stitch in time can save 9. So we can protect the human as well as the veterinary population also, because the veterinary industry is much more lucrative and we have to protect them also. So with all this, thank you to all of you.
Thank you so much, Dr. Saurav. You have created a place in the hearts of all the listeners, I believe, by caring for the heart of the dogs with so much humor and cartoons, so many cartoons incorporated. Probably you are a pet lover of dog, I do not know, and or you have small children, kids at your home, but you've gone into a lengthy description of the prevention aspect and the treatment aspects of the human as well as the dog and cats, the role of various drugs like melarsomin, prednisolone, doxycycline. All the doctors have been, and doxycycline plays a role in controlling it through it. and to also prevent in dogs the role of heparin and corticosteroids for pulmonary thromboembolism, then the various stages of prevention, and I think ultimately we come to the mosquito. So if we do all this, and your talk has also made us to create an interest in research in the veterinarians so that they can do one continuous surveillance regarding the presence of dirophilaria in cats and dogs. And thank you so much for the lengthy and you've gone in depth of each aspect of treatment.
Thank you, ma'am.
Like, there has been some questions in the chat box, like whether D. tenuis infection is there in India. Dr. Pranab has questioned this. Dr. Prasad can go into the. Can I take this question, sir?
What was the question? Please.
Question is whether Dirophilaria tenuis infection is there in India.
Dirophilaria repens. Dirophilaria repens is there in India, and most of the cases
tenuis,
around.
Tenuis. Sir, tenuis. It's not repens, tenuis.
Tenuis. So far, I know one report from. Yeah. One report from Karnataka has been reported on Dirophilaria tenuis. Only one report from Karnataka.
Okay, sir. Thank you.
Okay.
Any other questions? Anybody else, if they want, they can ask any questions or remarks. If not, I would like to wind up. Yes.
There's one more question from the participant. It is that how often do you get to see patients with dirophilaria?
Oh. I don't have any experience of seeing any patients of dirophilaria so far, because as you know that in India repens is most common and mostly is present in the subcutaneous lesion or nodule. So most of the subcutaneous nodule we probably we don't go for consultation to a clinician or all these things. Only when it is in some places like in the eye, ocular involvement is there, most of the cases, or in the face because of the cosmetic reason or something, or in Goa you can get one in the buccal submucosa because there's a nodule in the buccal submucosa, so it creates a problem in the chewing and all these things. So when these things come, then the patient will usually be present to the clinician for diagnosis. Otherwise, in most of the cases, around more than 60 to 70% of cases, these patients are asymptomatic and then they don't go for consultation regarding this. Even we don't go for a subcutaneous nodule or something. So most of the cases we avoid to having consultation.
May I ask a question?
Yes, madam.
What prevalence rate would you advise in meaning in dogs? What prevalence rate in a particular area would you recommend prophylactic treatment for dogs?
I think Dr. Saurabh can address this.
Yes, ma'am. What the question?
Is there a prevalence rate that you go by when you decide on using prophylactic treatment for dogs?
Actually, ma'am, in American Heartworm Society, in their things, they have told that every endemic regions, they have specific maps, as Professor Prasad has shown. The endemic regions have prevalence of even more than 20% among the canine populations. So they are the targeted ones. Actually, the surveillance is not so much strict, incorporating all the sectors. For the canine population, the highly endemic regions has to have the preventive things.
Prophylactic. Does it depend on the species of dirophilaria or is it common treatment?
Actually, mostly the studies around Dirophilaria immitis that has been a important species in the Americas, and the Dirophilaria repens in case of the Europes. So these two are the actually worked upon and surveyed population species, and the subcutaneous nodules of caused by the Noctiella species actually. The Dirophilia repens and other subdermata would see, and those things, they are not still into the preventive aspect yet by the veterinary societies. So it is mainly targeting the Dirophilia remitis and repens.
Right. Thank you.
Thank you.
Any more questions? I would like to wind up this session by by recording my gratitude to Professor Dr. Geeta Lakshmi, Vice Chancellor, Dr. M.G.R. M.G.R. Educational and Research Institute, IITP Vice President, and to Dr. Parija, IITP President, Dr. Kalavati Bector, the Organizing Secretary, and to my co-chairperson, Dr. Leela, and my mentors, Dr. Ravindran sir.
Thank you so much, speakers, for that sizzling discussion. It kept us bright and intrigued. We thank the chairpersons, Dr. Kavita and Dr. Leela, from the bottom of our hearts for moderating this session in such an interesting way. Thank you all. Thank you, ma'am.
Thank you so much. Thank you all.
And now we'll go for a lunch break, ma'am.
Okay, madam. Thank you.
And we'll be back at 2 o'clock.
Yes. 2 o'clock
Hello?
স্টার্ট কিছু তো বলেনি, দুটো বেজে গেছে কি?
Good afternoon. Dr. Ravindran, can you hear me?
Yes, madam. Good afternoon, madam.
So everything is set, no? My audio I wanted to check, that's it.
Yes, madam. Yes, madam.
Okay.
Fine, madam.
Thank you. Thank you.
Welcome back to all the enthusiastic delegates. Research is to see what everybody else has seen and to think what nobody else has thought. The key to becoming a good teacher is by becoming a good researcher. Working in the medical field, one cannot just learn what is present only in the textbooks. We need to keep ourselves updated with newer, intriguing to original research articles. It is now time to listen to all the budding and established researchers about what they have worked on. The eminent and learned judges for the oral presentation and e-poster are Dr. Rosie Venila, Professor and Head, Department of Microbiology, Savitha Medical College, Chennai. She was also the former Dean of Karur Medical College. Our next judge is Dr. Ravindra, Professor, Department of Rheumatology and Immunology. Let us start with the oral presentation.
Thank you, madam. Good afternoon. It's a pleasure having this session. I thank Dr. Rosie madam, Dr. Geeta Lakshmi madam for giving me this opportunity. And I thank ACS College for organizing this grand conference. And in this occasion, I should immensely thank and show my whole gratitude to my professor, who is Dr. Subhash Chandra Parija. Right from my day one, he has been with me, guiding me, and he has brought me to this stage. Thank you, sir. And I thank the whole team, Dr. Kalavati madam, Dr. Subbulakshmi, and all others. And it's a great opportunity given to me. We'll do the best. As Dr. Parija told, parasitology and lot of new parasites were discussed in this conference. And as the speaker before me has mentioned, without research, we can't grow. And this conference encourages young researchers. So this session is on oral presentation about various research, what the younger generation has done. I like Rosie madam to give her valuable comments and opinion regarding this. Madam.
Thank you, Dr. Ravindran. I thank the organizers, the Vice Chancellor, Madam Geeta Lakshmi, the organizers headed under Dr. Kalavati and her able team, and Dr. Parija, who is the pioneer. Dr. Geeta Lakshmi and Dr. Parija together have taken the tropical parasitology to this length over this decade, and I am very grateful for that. I'm happy to see that so many new parasites are being looked at, and young minds are focusing on them. And this is a very valuable input in terms of the research and poster presentation. I welcome each and every participant. More than 10 people have keyed in their papers for the oral presentation and for the poster presentation. So I'm looking forward to listening to each one of you. I thank the organizers for giving me this opportunity to chair this session. And I thank Dr. Ravindran, a very great colleague of mine. And with both of us, we hope we'll have a feast today, listening to each and every one of you. Thank you.
So we can request the presenter of this session.
Good afternoon, sir. Good afternoon.
Please, can you hear me?
Hello.
Jaya Banerjee, please present your slides, madam.
Good afternoon, sir. Good afternoon, ma'am. And good afternoon, esteemed delegates. Today, through this virtual platform, I, Dr. Jaya Banerjee, from King's Hospital, that is Krishna Institute of Medical Sciences, going to present a oral paper titled Seroprevalence of Toxoplasma gondii Antibodies in General Population at a Tertiary Care Center in Telangana, South India. Toxoplasmosis is caused by an obligate intracellular protozoan, Toxoplasma gondii. It can potentially cause life-threatening conditions in high-risk groups such as pregnant women and immunodeficient individuals. Infection in adults usually causes mild diseases, but greater importance lies in the preventing the transplacental transmission, which can cause major fetal anomalies and is vital to identify infection in pregnancy. It has a worldwide distribution affecting 1/3 of the world population. In India, surveillance has been done and which reveals the seroprevalence of toxoplasmosis is 24.3%, varying from 4.7 to 51.8% in North India, whereas 37.3% in South India. The burden of diseases attributable to toxoplasmosis is probably greater than currently suggested. Seroprevalence data from different patient groups could help in the formulation of targeted health policies. With this background, the aim of the present study was to determine the surveillance of anti-Toxoplasma gondii antibodies in various clinical departments at a tertiary care hospital in Telangana, South India. A prospective and observational study was carried out in Microbiology and Serology Department of Krishna Institute of Medical Sciences, Secunderabad, from January 2018 to October 2020, where 112 serum samples received for tox from various clinical departments were included. All the samples were tested for IgG and IgM anti-Toxoplasma gondii antibodies separately. All the samples were processed under strict aseptic precaution and following infection prevention and control measures. All the samples were processed by an automated immunoassay system, MiniVidas, which is based on the enzyme-linked fluorescent assay principle. The assay combined two-steps enzyme immunoassay sandwich method with a final fluorescent detection. Pictures I have shown the serum sample, and this is the reagent kit, reagent strip cuvette, and this is the solid phase receptacle. Coming to results and observations, when we observed the gender-wise distribution of the serum sample included in the study, we found that out of total 112 serum samples, 59.82% were female and 40.17% were male serum sample were included. Coming to seropositivity, we found that is 20.53% of the serum sample were seropositive, which corresponds to 23, including IgM and IgG. So coming to separate IgG seropositivity samples and IgM seropositivity samples, we found IgG seropositivity samples, positive samples, %, whereas IgM seropositive samples were 1.78%. When we look at IgG seropositive positivity of Toxoplasma infection with respect to age and gender, we found highest seroprevalence was found to be of Toxoplasma IgG were found to be in the females of the age group of 21 to 30 years. 8 serum samples in this age group we found positive, which corresponds to 7.14%. Among male, 51 to 60 years of age group was predominant with 5 serum samples positive, followed by 41 to 50 years of male, that 3 serum samples were positive for IgG seropositivity. When we look at the clinical departments, various clinical departments, IgG seropositivity was found highest from the antenatal clinic, around 38%, followed by 24% in the immunosuppressed patients, which includes HIV, AIDS, etc., followed by 14% each from the pediatric and post-transplant recipient. Only 10%, that is 2 cases, were found from the neurological department. IgM seropositivity, positive cases, samples were only 2, and both of them were from the age group of 21 antenatal clinic. Hence, we can conclude our study as overall seroprevalence of toxoplasmosis was 20.53% with anti-toxoplasma IgG 18.75% and IgM 1.78%. Highest seropositivity of toxoplasma IgG was observed amongst the female of 21 to 30 years of age group. It reflects a significant risk of acute infection and complications in general population. Most of the clinicians do not recommend for the toxoplasma profile, torch profile or toxoplasma test for the antenatal patients. But we suggest from this study that it should be torch profile should be a routine screening method, routine screening investigations for all antenatal women. The incidence of opportunist toxoplasmosis is probably underestimated and poorly studied. Hence, more studies focusing on continuous monitoring of seroprevalence strains need to be conducted across the country to assess the risk and prevalence of toxoplasmosis in general population, so that targeted health policies can be formulated. These are my references. Thank you.
Thank you, madam. And it's a nice presentation. One question I need to clarify from you. Is there any difference between HIV and non-HIV in your study, madam?
Yes, sir. HIV.
Prevalence.
Yes, sir. HIV patients, we got in neurological patients, both the patients were HIV associated. But in other, many patients are HIV positive, but toxoplasma negative. That also we found. And only 2 cases are there where the HIV positive along with the toxoplasma IgG positive, sir.
Any difference in the antenatal cases, madam? HIV positive
Antenatal cases, sir, HIV positive, I didn't find anyone.
Madam,
Can you please throw some light on what was the basic, did you find any difference between the IgM positive patients and those who were only IgG positive? And how many of them did you see a space occupying lesion or any other complication?
Space occupying lesions, ma'am, neurological few patients we have seen, but out of that only 2 cases were positive seroprevalence, but they were not toxoplasma positive. And in others, ma'am, IgM, strangely, I don't know why, but IgM, we got very less, only 2 cases I got, and that too from the antenatal clinic IgM positive. And so there is no space occupying lesion we found. But it was like we have to further study in details the case history and all that we have to do. It was not much in depth. So it was our like out of scope. So now we will do in details of the things, ma'am.
Yeah, in essence, Jaya, you are telling that you had only 2 patients who had space occupying lesions. That is neural toxoplasmosis.
Yes, ma'am. Yes, ma'am.
And amongst those 2, none were IgM positive.
Yes.
None were
IgG positive, yes, ma'am.
Yeah, they were IgG positive, none were IgM positive, and you found IgM positivity in antenatal women. When was this study done? When was it completed?
It was started from 2018 January to October 2020.
Yeah, so how long has it been that these antenatal women could be followed up, and where they followed up to check for any visible symptoms, because they were asymptomatic, they were IgM positive. So did you have an opportunity? Were they one of the early subjects?
Not yet, but we'll do, ma'am.
It is important that you should follow
antenatal, especially antenatal
asymptomatic patients actually. Yes, and so here you had one asymptomatic, but who probably was in acute phase. So it would have been a good opportunity.
Yes, ma'am. This is recent case only. So I will like it was, and patient was in September only, and it was in the first trimester. So thank you, ma'am, for suggestion. We can still do follow up something at least.
I had a question regarding the antenatal cases, because that is very important. If you are getting 2 positive for IgM,
yes, sir,
you have to do something about these cases, because this is the reason we are doing an IgM in antenatal cases.
Yes, sir. Yes, sir.
We have to see whether there is any transmission, because ultimately, if here IgM is detected and it's a high titer, probably we give some treatment.
Yes,
during
sir.
pregnancy also.
First, we need to confirm also, and we have to do the ultrasound from the starting from 24
Sometimes you may not be able to confirm, but in spite of that, you may have to give treatment just, to be on the safer side.
Okay, sir. Sir, are you, pardon me, sir, is there any question, sir?
No, I am finished with the question.
Participants, anyone wants to have any clarifications, any questions?
If there are no more questions, then can we move on to the next speaker? Thank
Yes,
you very
ma'am.
much, Dr. Jaya.
Thank you, ma'am. Thank you, sir.
Thank you, Dr. Jaya.
Thank you, sir.
We'll move on to the next presenter.
Good afternoon, sir, ma'am. I'm
Yes, madam.
sir, is my screen visible? Shall I start, sir?
Still,
Can you share your presentation, madam?
please share, madam. Still, it is yet to come, madam.
Okay. Sir, is it fine now? Sir, is it visible now, sir?
No, ma'am.
No, madam.
You have any trouble sharing your screen?
Organizers,
Yes, you have any trouble, Dr. Rangashini? You have any trouble sharing your screen? Have you done a screen share?
Yes, ma'am. I'm
because we are not able to see your presentation. You see, when you click at the bottom of this page, you'll find an option called share the presentation. Okay. Present now. There is an option called present now. Okay. So click on that. Okay,
your
click on
entire
that.
screen.
Okay, your entire screen or your window. So click on the option whichever you want to do in. Only then your present. Okay.
Okay.
Yeah, now you click on the PPT and you share it. In case it is not working, organizers, do you have her presentation?
Ma'am, I have clicked it now. Is it visible now, ma'am? My presentation.
It's not visible as yet.
நம்ம present பண்ணலாம். Shall we?
Yeah, organizers, you can present.
Sir, we will do it, sir.
Yeah, yes.
Dr. Rangashini, can you see the presentation now?
Yes, ma'am. I can.
Would you like to start?
Yes, ma'am. I'll start.
So when you say next slide, we'll change. Okay?
Okay, ma'am.
Please start, madam.
Good afternoon, one and all. I thank the organizing committee and HOD Dr. Delirani madam for giving me this opportunity to present a case report on strongyloids hyperinfection syndrome in a post-renal transplant patient. So next slide, please.
Can we have
so the military?
Excuse me, can we have the slide share? The person who's presenting, please put it on slide share on the full screen.
Thank you, ma'am. The military worm strongyloids stercoralis is a commonly prevalent nematode in our community. and it poses a great risk of strongyloid stercoralis hyperinfection syndrome, which is shortly called as SHS, among immunocompromised individuals and those who are on immunosuppressants. So because of the two important features of the strongyloids called parthenogenesis and repeated autoinfection, it leads to strongyloid stercoralis hyperinfection syndrome and disseminated strongyloids in more serious cases. So suppression of host cell immunity by this glucocorticoid therapy, transplantations, malignancies, and immunosuppressive drugs, they pose a threat of strongyloid hyperinfection syndrome, leading to a great deal of mortality and morbidity. So the next slide, please. Now I'm going to the case history. It was a 28-year-old male who presented in the nephrology department. He was a post-renal transplant patient. 3 years back, he had a renal transplant, and he was on immunosuppressive therapy with prednisolone, mycophenolate mofetil, and tacrolimus. So he presented with symptoms suggestive of graft dysfunction and graft rejection. Next slide, please. So the initial management was done like this. He was preliminarily screened for any active infections, and his chest X-ray was also normal. And he was then started with pulse intravenous methylprednisolone therapy of 500 milligram, 250 milligram, and 250 milligram on 3 consecutive days, following which his graft functions improved. But 2 days later, he developed intolerable abdominal pain, profuse vomiting, and small volume diarrhea, along with tenesmus. Next slide, please. So his X-ray and CT abdomen showed ileus and bowel wall edema. So also stool microscopy was sent. He was started empirically on oral nitrous oxonide, and single dose of oral ivermectin was given. PCR, quantitative PCR for CMV revealed viral copies, plenty of viral copies, and he was started on oral Valgan cyclovir. So despite these measures, he developed high spikes of fever and he worsened. Next slide, please. Then sepsis was suspected, and dose reduction of immunosuppressants was done, and antibiotics was escalated. Now his chest X-ray and CT chest revealed miliary tuberculosis. Miliary mottling was there, so it suggested a possibility of tuberculosis or fungal pneumonia. So he was empirically started on ATT and amphotericin B, but still the patient deteriorated. Next slide, please. So this is the his X-ray and CT chest finding showing miliary mottling. Next slide, please. His stool AFB results revealed oozes of Cryptosporidium parvum, along with plenty of rhabditiform larvae of strongyloid stercoralis on wet mount analysis. His absolute eosinophil count was 600 per cubic millimeter. So he was diagnosed as strongyloid stercoralis hyperinfection syndrome with both gastrointestinal and respiratory symptoms. Next slide, please. So this is the wet mount picture showing strongyloid hyperinfection in stool, and this is the stool modified AFB showing Cryptosporidium oocysts. Next slide, please. So he was started on oral ivermectin 200 microgram per kilogram per day and albendazole 400 milligram twice daily, and his and ATT and antifungal treatment were stopped. So, but still, despite these measures, patient still deteriorated and worsened. He developed hemoptysis. So on sputum examination, plenty of live filariform larvae were present. Next slide, please. So now he was malabsorption and ileus secondary to strongyloid hyperinfection syndrome was presumed. So on extensive review of literature, it revealed parenteric veterinary formulation of ivermectin in humans for severe cases in rare occasions. So hospital ethics committee approval was obtained, and after informed consent from patients' relatives, parenteral veterinary formulation of ivermectin was procured from veterinary hospital, and it was administered at a dose of 200 microgram per kilogram per day in 2 divided doses subcutaneously. Next slide, please. So now patient improved clinically after 3 doses of subcutaneous ivermectin, and he was switched over to oral formulation after 7 doses. So oral formulation was continued for 2 weeks, and after 3 consecutive stool examinations were negative for strongyloids, Gradually, immunosuppressants were introduced. Now, patient did well. So, a brief discussion on strongyloids hyperinfection syndrome. So, strongyloids infection, it begins with the entry of filariform larva, which is the L4 larval stage, by skin penetration, and it enters into the venous circulation, and through the venous circulation, it enters the right heart and lungs. So, from there, it leaves the pulmonary circulation, and through the alveoli, bronchi, trachea, larynx, and epiglottis, it is swallowed, and by that route, it reaches the intestines. Here, the female, it develops into adult female worms, and the female worms by either sexual or sexual reproduction or parthenogenesis. They lay eggs and which immediately release the rhabditiform larva, which is the diagnostic form released in the feces. From then after that, in the soil, they develop into filariform larva. Next slide, please. So autoinfection is a characteristic feature of this nematode worm. Here it occurs with the rhabditiform larva. They penetrate the bowel mucosa and perineal skin and develop into filariform larva, and the cycle is repeated again. So hyperinfection occurs when this is exaggerated due to immunosuppression or steroid usage. Next slide, please. So the clinical features are a wide range. They may be either asymptomatic or they may present with skin rashes, hemorrhages in the lung, alveoli, bronchopneumonia, intractable diarrhea with blood and mucus. And usually strongyloids hyperinfection syndrome presents with severe diarrhea, malabsorption, paralytic ileus, etc. Next slide. Next slide, please. Another thing is disseminated infection. It is a different terminology. Here, the worms penetrate the intestinal wall and they reach the other organs like brain, liver, kidneys, etc. So they lead to brain abscess, meningitis, peritonitis, etc. So gram-negative septicemia may occur because the organisms, when they leave the intestine, they carry the intestinal organisms in a piggyback fashion with them, along with them. Next slide, please. So demonstration of rhabditiform larva in stool specimens or sputum, duodenal washing or biopsy is diagnostic. So, but stool examination, it has a low sensitivity. So stool culture may be required in some cases. So other newer diagnostic methods like complement fixation test, ELISA are also used now. Next slide, please. Thiabendazole, albendazole, and mebendazole can be used for treatment, but ivermectin is the ideal drug of choice at a dose of 200 mg per kilogram per day. Albendazole can be used at a dose of 400 mg per day for 2 days. But in severe cases, treatment may be required for up to 7 days. Next slide, please. Next slide. So, I conclude this by saying that GI symptoms and lung manifestations with rashes in a patient with immunosuppression should raise a strong suspicion of strongyloids hyperinfection syndrome, and the complications are malabsorption with paralytic ileus, so it may require therapy with parenteral agents. So routine screening of transplantation patients for strongyloids stercoralis is advisable. Thank you.
This study, how did you confirm it is strongyloides larva, madam?
The patient was a post-renal clinical history was suggestive, sir, and also by the morphology of the larva, it was clearly visible, sir. The morphology, we could clearly see the oral cavity, duodenal bulb, everything we could clearly see, sir. The genital primordial pore, we could see, make out everything.
Any culture was attempted in this study, madam?
There were plenty of larvae, sir. In this case, we didn't attempt, sir. In another case, we did, but we couldn't culture it. So in this case, we didn't attempt, sir. There were plenty of larvae, so we didn't think of it.
Madam, Rosie, madam.
A very good case, and it's a nice thing that the clinicians had suspected. However much time was lost in their search. There are two questions. One is, initially they had done a X-ray check, and
Yes,
that
ma'am.
did not show the mottling, which
Yes, ma'am.
subsequently showed after 1 week. Is that what you're trying to tell me?
Yes, ma'am.
Two X-rays. Yeah.
Initial screening was normal, ma'am. They did the initial screening, which was normal. But
Yeah.
after that, after
Have
one,
you got to see the two and how far apart were the two X-rays taken? How many weeks apart were the two X-rays? Because sometimes we tend to look at an earlier X-ray and think it is normal. Was it done at the time when the patient presented himself with these symptoms? Did you know the date of the two X-rays? That is of interest to me clinically.
okay, ma'am. Ma'am, I'm not sure of the dates, ma'am, but everything happened within a few days, ma'am.
Yeah,
Only
patient
2
came
weeks
in
period.
patient
Yes,
came
ma'am.
in at 2 weeks apart. I just wanted to know whether the first X-ray was taken after the patient presented himself, or it was a retaken after the patient presented himself, or it was a recent X-ray taken before he had any onset of symptoms. Even with that, we will tend to rule out certain things like tuberculosis or other things. So it was not a so this is one reason why I think as an investigator, as a researcher, it would have been very nice if you had the dates of the two X-ray.
Sure.
I'm not trying to tell you that they were not close enough. I'm only because you should have
Sure.
all points on this. Okay.
Sure.
And the mottling, was it lost on follow-up, or were they still existent even after the treatment?
The patient improved symptomatically.
Yes.
Ma'am, I didn't follow the
This is another thing you would want to, as a curious mind, you would want to see whether the damage done at the lungs was a reversible or it went on to exist and remained calcified foci later, things like that. So it will be of interest to us because you don't see these cases. So, Rangashmi, you should realize these are very peculiar cases which, if recorded and documented nicely, it is eye-opener and a good forum for all of us to know about that. Yeah, of course, this would have happened only a few months ago, but it's nice if you can follow up even subsequently. Try to follow up and see so that you will be able to record when the features, if at all, when they disappear. This is another
Sure,
thing.
ma'am. I'll follow.
And then the rashes you were talking about, was it present in this index patient? You were saying that these symptoms along with rashes should alert us to the hyperstrongyloidosis. Now I want to know whether in this patient there were any rashes.
No, ma'am. This patient didn't present with rashes, ma'am. He had only GI symptoms initially. Followed by that, he developed pulmonary symptoms. Initially, he only GI symptoms.
Yeah,
so the present
expected also because the patient was already on immunosuppressants. So rashes are a mark of hypersensitivity. So that also is possible. The patient did not have rashes. So I was just wanting to ask you these specific questions. Did you look for rashes and they were present or absent?
Rashes were not present, ma'am, in this case.
Yes. Okay. Thank you very much.
Any questions from the participants? Participants, any questions?
No, sir. I think we can go on with the next participant.
Yes, madam. We can go for the next presenter.
Thank you, sir.
Hello.
Dr. Ramesh, sir?
Yes, madam.
Are you
Am I
there?
audible?
Yeah.
Okay, madam.
Yes, sir.
I can start.
Madam, is it visible?
No, sir.
At present, is it visible?
No, sir. Have you shared, sir?
Is it visible now?
No, sir. Yes, sir. Now it is coming.
Visible now?
Yes, sir.
Can I start now?
Please start. Go on to
Yes,
slide
sir.
10 and start.
Good afternoon, my respected teachers. Myself, Dr. Ramij Yusuf. I am PGT of Calcutta National Medical College. My topic is an isolated pulmonary hydatid cyst, a rare case presentation in a young female from Kolkata. Echinococcosis or hydatid disease is caused by the larval stage of the tapeworm Echinococcus. 4 species are recognized, and the vast majority of infections in humans are caused by the Echinococcus granulosus. Echinococcus granulosus causes cystic echinococcosis, which has a worldwide distribution. Humans are exposed less frequently to Echinococcus multicularis, which cause alveolar echinococcosis. Echinococcus vogeli, Echinococcus oligoarthurs are rare species and cause polycystic echinococcosis.
Dr. Ramesh, sorry to interrupt you. Your slides are not changing.
Is
Make
it changing
sure,
now?
if
Is it now?
You have to go to slideshow.
Slideshow. You'll have to go to slideshow and then change.
Welcome, Rosie.
Is it visible? Is
Good
it
afternoon.
visible?
Yes, sir. Yes, sir. It is visible, but you have to go to slideshow so that it will change.
Now it is slides. I plug into the slides. So now is it visible? Now the slides are changing.
No problem with the visibility, sir. It is not changing.
Press F5, sir.
Please go to slideshow. At the bottom of your screen, you have the full screen, no slideshow.
Or press F5.
F5, madam, I have already done already. F5. Is it visible?
It's not full screen. Yes, now I guess. No.
Is it full screen now?
No.
Not yet. It is not full screen, sir.
Okay, I can read.
manually change your change your slide.
Change your slide by pressing arrow key.
Sir, you click on the slide number 3 and go on, sir.
Okay, ma'am. Now the pathogenesis.
Click the slide.
Should I start from the beginning? Hello?
Continue, sir. You can continue where you left.
Yes, you can proceed, sir. Where you
Okay.
want.
Now the pathogenesis. In man, the larval form causes unilocular hydatid disease. Hydatid disease is now renamed as cystic echinococcosis. So, what are the mode of infections? First, by direct contact with infected dogs and by allowing the dog to feed from the same disk. And the third one is by uncooked vegetables contaminated with infected animal fishes. Although larvae can enter the lungs through the lymphatic system or bronchial system, it is thought that cysts settling down in the lungs are usually larvae that have passed through the hepatic sinusoids. Pulmonary cysts typically increase in diameter.
Sorry, doctor, I think you have to change your slides. You have to go down. Okay, I think you're going to the next one.
Madam, actually my sharing is not working.
Madam, shall we present the slide,
yes,
madam?
Yes, exactly.
Yes. Please, organizers present the slide.
Yes, ma'am. We will do it.
Madam, should I continue?
Yes, sir. Once the slides have come, you can continue.
Continue.
Although larvae can enter the lungs through the lymphatic system or bronchial system, it is thought that the cysts settling down in the lungs are usually larvae that have passed through the hepatic sinusoids. Pulmonary cysts typically increase in diameter at a rate of 1 to 5 cm per year. The fully developed cysts are composed of 3 layers. The outer layer of pericyst is composed of inflamed fibrous tissue derived from the host. The exocyst is an acellular laminated membrane, and the innermost layer or endocyst is a germinative layer of the parasite, give rise to brood capsule or secondary cyst, which actually bud internally. An intact cyst, if large, may be filled with liters of fluid. The fluid, which is antigenic and may contain debris, contains hooklets and scoliosis and is referred to as hydrated sand. Daughter cyst may develop from the endocyst. Here is the structure of the cyst. Now my case presentation is that a 35-year-old female patient came to OPD of Calcutta National Medical College, Kolkata, from a local area of Kolkata, presented with intermittent fever, chest pain, dry cough, and heaviness of left side of chest for last 3 months. She was treated conservative, denied tuberculosis contact. She belongs to a farmer family. On clinical examination, she was cachectic. Her auxiliary temperature was 36.2 degrees centigrade. Pulse rate was 86 per minute. Blood pressure 108 by 72 mm Hg. On auscultation, there is decreased breath sound along with vocal phobitus. Next slide, please. Now the investigation part. Hemoglobin is 11.6 gram per deciliter. Her ESR is 90 mm per hour. Serum electrolytes are normal. ECG is within normal limit. Chest X-ray, homogeneous opacity in the left lower zone of the lung. CCT showed well-defined homogeneous cystic lesions with low density and smooth walls of variable thickness in left lung. The patient undergo lobectomy. Here is the patient X-ray. Now the method of lab diagnosis. Post-left lobectomy of the lung, a piece of tissue sample with cystic fluid was sent to microbiology department for definitive diagnosis. Wet mount preparation from the centrifuge deposit of cystic fluid was done. Histopathological examination with hematotoxylin and eosin stain was done. Now the result, next slide please. Now the result and laboratory findings. Microscopic finding by wet mount preparation from the centrifuge deposit of hydrated or cystic fluid showed hooklets. Histopathological examination shows the layer of cyst wall. Now I am showing the wet mount preparation. It's actually showing the hooklets of Echinococcus granulosis. Discussion part. Cystic echinococcus is one of the major zoonotic disease. They are also of public health importance due to their geographical distribution. Hydrated is endemic in sheep rearing countries where sheep are in close contact with humans. Humans are infected as accidental host in the life cycle of the worms. Majority of the cases are silent. Uncomplicated hydrated cysts are discovered incidentally during routine chest X-ray. Diagnosis is made upon clinical, radiological, and microbiological findings. What is the differential diagnosis of pulmonary cyst? Is tuberculosis, lung abscess, aspergilloma, bronchogenic cyst, primary or secondary benign or malignant tumor. Next slide, please. Now, my conclusion is that from the above history, as well as radiological and the laboratory findings, it can be concluded a case of isolated left-sided pulmonary hydrated cyst. The infecting organism being Echinococcus granulosis. Pulmonary hydrated cysts are rare events in endemic areas. Hydrated cyst is actually a public health problem. Cystic echinococcus remains a significant problem worldwide in terms of medical, social, and economic impacts. Surgery is the mainstay of management of pulmonary hypertension. Can be shout in patient with surgical contraindications. Surgery carries a high risk of rupture, especially if the cysts are considerably big due to late presentation, and thus pose a risk of fatal outcome, particularly in resource-limited settings. Disease control can be improved by increasing awareness among healthcare workers and educating the community before more importance is given to increasing diagnostic capacity and implementing prevention and control strategies suggested by the WHO and the Centers for Disease Control and Prevention to prevention. To overcome this, education on proper hygiene and healthy livestock management practices are encouraged. Now my references are
Thank you, sir, for the presentation. I have two questions. One question is, in the laboratory investigation, the hemogram was...
Madam, sir, actually, their investigation, the complete blood count was normal in this patient.
Rise in eosinophils for this kind of helminthic infection.
Yes, sir.
So what will be the reason for that?
Excuse me, sir. What?
He is having absolute. He is having a normal eosinophil count. We expect eosinophil increase.
Yes, sir.
Both eosinophils as count as well as absolute eosinophil count.
Yes,
can you explain that?
In case of parasitic infection, there is increase we found, and other.
Or similar cases in your place, you have similar findings?
Sir, I have not found any similar cases like that that which has a increased eosinophil count actually.
Okay. Madam, you can add on.
I don't have any questions. Does anybody among the audience have a question? If there are no questions, then we'll move on to the next speaker. Thank you very much, Dr. Ramesh.
Thank you, madam. Thank you, madam.
Thank you, Dr. Ramesh. We'll move
Thank
on
you,
to the
sir.
next presenter.
Thank you, sir.
Sir, Dr. Jayaprakash, are you ready, sir? Sir, Dr. Jayaprakash, sir?
Yes,
Ah,
ma'am.
yeah.
I will.
Okay, you can start presenting.
Yeah.
Yes, sir. Dr. Jayaprakash can present, sir.
Yes. Yeah, yes, sir. I will try to. Yeah, yes, sir. So that will not show any.
Okay, Dr. Jayaprakash,
Yes,
you
ma'am.
want us to present your PowerPoint so that you can read
Yeah, yes,
that?
ma'am.
Okay, if you want me to turn to the next page, just say next. Okay.
I think
Yeah,
there's
yes.
something wrong with the entire hookup. So make it a routine to present. Unless the presenter is able to, you just make it a routine, because it's happening for every subsequent speaker. So
Yes.
thank you.
Yeah.
Yeah, that will be the better mode of
Okay.
presenting. Okay.
Okay.
Yes, sir. Jayaprakash, sir, you can start.
Sir, 1 second, sir. I will share.
Can you see?
Yeah, yeah.
Not yet.
Yes, sir. Sure. Sir, you want to see that slide, sir? Sir, you see that slide?
Yes, sir.
Slideshow.
Yeah.
Can you go to
Yeah,
slideshow?
Yeah, yes, ma'am.
Yes, good.
Yeah. Thank you, ma'am. Everyone, good morning, ma'am. This is my topic. A study on the correlation of intestinal parasitic infection and hemogram in symptomatic and asymptomatic patients attending a tertiary care center. Introduction. Parasitic infection caused by intestinal parasites such as protozoa and helminthes are a cause of health concern globally. Major factor are prevalence of intestinal parasitic infections in developing and underdeveloped countries include poor sanitation, poor personal hygiene, illiteracy, increasing population, unavailability of potable water, poverty, and tropical climatic conditions. 60% of the world population is affected with parasitic infection in the intestine, resulting in high morbidity and mortality rate. The prevalence of intestinal parasitic infection in India caused by enteric parasitic ranges from 12.5 to 67 in various studies. Common parasitic infection reported globally included Ascaris lumbricoides, bookworm, Trichuris trichura, and Entamoeba histolytica. The frequency of parasitic infections varies with age, gender, and the place of which the population is chosen. Advancement in the detection of diagnosis parasitic of infections can improve the sensitivity and decrease the incidence of manual errors. Elements are increasing association with the increased risk of anemia, malnutrition, low pregnancy, no pregnancy weight gains, and intrauterine growth retardations. Intestinal parasitic infections can cause a series of characteristic changes in blood constituents, hypoalbuminemia, or more commonly decrease in serum total protein concentration. Bookworm can cause blood loss in the intestine. It is the result of anemia. Major challenges to in developing countries in order to decrease the incidence of parasitic infections include lack of sufficient research, neglect of symptoms, and lack of follow-up. Better awareness among people can also aid in the reduction of parasitic infections. My aim and objective is to find out the prevalence of parasitic infection among the patients attending a tertiary care center. Find the correlation between the intestine parasites infection and the hemogram in the study populations. Find any significant in the result between the asymptomatic patient and the symptomatic patients. Material and methods. This is a cross-sectional study, the period of 2 years, 3 months in SRM Medical College, Katamuluttu, Kotteri. Total 903 patients we are getting. Symptomatic patient is 653 in the age of more than 18 years with the presence of gastrointestinal symptoms, and the 250 people belong to the control group were included. Exclusion criteria, less than 18 years we are excluded in this study. Result. Study population is 903, among which 250 samples were from asymptomatic patients. Positive parasitic infection were detected in 23.9% of the total study population, among which 8.8% were asymptomatic patients. Men were predominantly affected comparative to women, and the most common affected age group was 20 to 25 years in our study. Entamoeba histolytica were the major parasites found, close followed by helminthes among the affected study populations. Among asymptomatic patients, Entamoeba histolytica was the most predominant, followed by Giardia lamblia, 18%. The decrease in the level of hemoglobin, less than 6 milligrams percentage, was found to be higher in symptomatic patients, 23%, compared to asymptomatic patients, getting that 4.5%. Based on the geographical location, symptomatic patients were mostly distributed in Chengalpattu region, while majorly asymptomatic patients will hail from the West Bengal. This is Indian. West Bengal is 11%. In the asymptomatic patient, Chengalpattu also will get the 11 numbers. This is Tamil Nadu. Chennai we get that 5%. Mostly the Chengalpattu we get that more percentage in 73%. The abnormalities in hematological parameters are more evident in symptomatic patients compared to asymptomatic patients. The erythrocyte sedimentation rate is more than 20 mm per hour was found in 68% of the affected populations, although the HP and the protein level was normal. Eosinophil ranges more than 80 were found to be more predominant in parasitic infections. Conclusions. The total prevalence in our study was found to be 23.9%, similar to other study than in Indian populations. Among the parasites found, Entamoeba histolytica, Giardia lamblia, and hookworm predominate the intestinal parasitic infection. Early diagnosis, better sanitation, and effective health management are prerequisites to control and manage infections in developing countries. We plan to compare the use of automated stools testing and a conventional diagnostic method in our future. These are all my references. Thank you.
Thank you, sir. You have done a study comparing symptomatic and asymptomatic.
Yes, sir.
How to explain the phenomena that Chengalpattu, most of the cases are symptomatic. How many percentage was asymptomatic when compared to the other group?
In Chengalpattu, yes, sir. We got that 73 patients or 73% were symptomatic patient in Chengalpattu.
Yes, sir. Among the asymptomatic in Chengalpattu, what was the predominant worm infestation?
Chengalpattu.
Yeah, so that we can tackle that, no? This getting transmitted silently. You can work on that also. What was the predominant path? Helminthic or Entamoeba histolytica?
Entamoeba histolytica, sir. Predominantly, we'll say in the Entamoeba histolytica. Yeah.
So asymptomatic cyst passes
asymptomatic patients also having Entamoeba histolytica predominantly seen.
So that's a problem in and around Chengalpet as well as throughout the country. Asymptomatic
Yes,
cyst passes. Yes, sir. Madam, you can add on.
I have only one thing about your study. Why is it that you, knowing that parasitic infections are more common in children, you have restricted your population to above 18 years of age? You have
Hello.
excluded those below 18 years.
Yeah, ma'am, we are starting that in our PhD topic. This is for we included the adolescent only, ma'am. We will not according to the reactive arthritis. My topic is going to the reactive arthritis. It is more in the, so I will take that adolescent group only.
I will agree for that topic, but not for this presentation for the tertiary care center study on intestinal parasitic infection. Then you should have also included these. Okay, but anyway, or you should have retitled it as in adult.
एक मिनट दो मैं मेरा लेक्चर है.
Okay.
Yes, ma'am.
Okay.
Yeah. Okay.
If anybody else has any questions, please go ahead.
Yes.
Can I comment? Make a comment? No, Dr.
Yes, sir.
Rakesh, you want to ask question? Okay, you ask. I'll ask another question.
Yeah, I just wanted to add about Amoeba histolytica, because that asymptomatic Amoeba histolytica, probably they are just diagnosing Amoeba histolytica morphologically, and this is probably Amoeba dispar or Moshkowski, which we have not differentiated. You can't differentiate morphologically. So so high Amoeba histolytica infection is really not possible. We have to really differentiate actually. So that is you have to do a molecular study to differentiate that.
Okay.
That is exactly. Am I audible now? Can you hear
Yes,
me?
sir. Yes, sir. Yes, sir.
Yeah, and nice. Thank you for good paper. Just I like to supplement what Dr. Rakesh has told. You cannot tell it is Entamoeba histolytica. So please write. If you are not used any molecular technique, you write Entamoeba histolytica / Entamoeba dispar or Entamoeba muscosi. This is a WHO recommendation. Okay. Thank you so much.
Okay, thank you, sir.
That is a very valuable comment from Dr. Parija for all the audience. So hereafter the diagnosis, please make it a point, medical identification, especially when the patient is asymptomatic. You can never ever conclude. Thank you, sir.
Okay, thank you, ma'am. Thank you, sir.
Thank you, sir. We'll go for the next presentation.
Dr. Hussain?
Yes, ma'am. A very good afternoon to all of you.
Yes, ma'am. You start presenting, ma'am.
Yes, ma'am.
Madam, you are not audible.
Madam, not audible.
Am I audible?
Yes, madam.
Okay. So my topic of presentation is clinical radiological profile of a case of neurocysticercosis at a tertiary care center in Uttar Pradesh. To start with the introduction, neurocysticercosis, which is considered a neglected tropical disease, is most feared due to its life-threatening complications like epilepsy, stroke, and hydrocephalus. The disease is widely prevalent in North India, with states like Uttar Pradesh, Bihar, and Punjab being endemic for the disease. My objective was to focus on clinical evaluation that traces the suspicion of neurocysticercosis and its diagnostic approach to avoid incorrect diagnosis. So my case was a 7-year-old boy belonging to district Lucknow, admitted in pediatric ward with chief complaints of headache for the last 3 days, vomiting for the last 1 day, abnormal movement of the body followed by unconsciousness for 5 minutes at home. It was a single episode. There was no history of trauma, jaundice, skin rash, cough, and cold. Past history was nothing significant, and patient was immunized as per routine immunization schedule. Development history was within normal limits. Then in general examination, weight and height were below normal. Patient was afebrile, vitals were sinuses, clubbing, edema, lymphadenopathy were all absent. And in systemic examination, eye, verbal, motor response were all normal. Pupil was normal size, normal reacting. Part, tone, deep tenderness was extensive. This was a significant finding. There was no sign of pharyngeal irritation for abdomen. There was no organomegaly, serious there was no any significant finding in CVS or chest. Then coming to complete blood count, all the values were within normal limits, including hemoglobin, TLC, KFT, LFT, electrolytes were all normal. Coming to microbiological investigations, HbS, AGH, IVSC were non-reactive, blood culture was sterile. In radiological investigation, an MRI finding, ring-shaped lesion with eccentric dot in right facing frontal lobe was seen, along with inflammatory granuloma, which was suggestive of neurocysticercosis. Fundoscopy was within normal limits, and routine examination and microscopy, there was no any significant finding. Coming to the treatment, patient was given injection mannitol for 3 days. Injection ceftriaxone started as the patient developed fever, but once culture report came out to be sterile, antibiotic was stopped. Patient was also given omnacotic, which is steroid, for 6 days, then tablet albendazole for 21 days, then anticonvulsant phenytoin was given. Initially, it was given IV, then followed by oral, and then supplements like calcium and multivitamin. So to summarize, on the basis of clinical history and examination, a working diagnosis of intracranial space occupying lesion was formed. Patient was managed symptomatically and injection mannitol was started. During course of hospital stay, child developed fever, hence IV antibiotic was started. Once the culture report came out to be negative, antibiotic was stopped. MRI was done, which was suggestive of neurocysticercosis. The condition of the patient gradually improved. He was conscious, hemodynamically stable, afebrile, accepting oral feeds well with lesser episodes of headache, and hence was discharged with advice to follow up in pediatric OPD. This is the Del Bruto's diagnostic criteria of neurocysticercosis, and in my case, there were 2 major along with 1 minor, and 1 epidemiological criteria were being fulfilled, so it is falling under the definitive diagnosis. So neurocysticercosis is the most common parasitic disease of the nervous system. As we all know, it is caused by ingestion of embryonated eggs from feces of individuals infected with fork tapeworm, and also by autoinfection. Seizure is the most common manifestation, followed by headache. The disease commonly affects adults in the 3rd or the 4th decade of life, less commonly in children and older population. Prasad et al. Reported a high incidence of intestinal teniasis, 38%, and 3 cases of neurocysticercosis as evident by MRI examination from 3 villages near Lucknow, indicating more vigilant action to be taken by clinicians to combat the disease burden. So to conclude, the present study highlights the fact that the children presenting with neurological features should be screened for neurocysticercosis so that this deadly disease is not missed in endemic states like Uttar Pradesh. Thank you.
Thank you, madam. And this study, it's mainly based on the radiological finding. Since hemogram was also normal, you are mainly basing on the radiological finding.
Yes, sir.
What any attempt was made to diagnose it microbiologically? Any serological test?
No serological test was done.
If given a chance, what all serology is possible in your place or in a reference center?
Western blot can be done.
Yes.
Other tests which can be done are the molecular, which are based on the mitochondrial DNA, and we can differentiate between tinea solium and tinea solium ischiata on the basis of molecular methods, lymphocyte transformation test, and recently LAMP has also been added in the list of the diagnostic tests. These all can be done.
Yes, madam. Because now microbiological diagnosis is also very vital for this kind of a study, apart from our radiological screening. And as a routine, even Dr. Parija used to insist on that, and many studies have been done in Pondicherry. Even for many places, neurocysticercosis is very common, and it is a treatable condition. So microbiological screening has to be initiated at all our centers.
Yes, sir.
Any questions
Because
from the?
I was there in the initial part of the when the cases came, so MRI findings were suggestive of neurocysticercosis, so nothing was done on microbiology.
Oh. Madam, Rosie, madam, would like to add on?
What was the specific treatment that was given?
Tablet albendazole for 3 weeks.
Pardon?
I'll
For
bring
how
this water
long? How long?
for 3 weeks, 21 days,
Okay,
at the rate
and
of 15 milligram per kg per day.
yeah, then how long is it since the patient is on follow-up? Did you find out the findings, the MRI finding? It's of interest, isn't it, to know what
Lesion was
happened?
resolved.
Lesion was completely resolved because you told clinically the patient improved. Now, because your conclusion itself is on radiological finding, so reversal of the radiological finding is the minimum that we can expect. And as Dr. Ravindran said, an attempt should surely be made to make a laboratory diagnosis. Okay, then there is no difference, isn't it? Laboratory diagnosis we should include for presentation if you make locally, and then you will be able to. So if you make it at your institute, and then that will raise the awareness, and you will be able to have the gamut of investigations for your diagnosis in that place. However, laboratory diagnosis is one of the pillars which any young researcher like you should insist on.
Yes, ma'am.
Anyway, thank you. And is there any other questions?
Ma'am, I like to add
Yes, sir.
On the laboratory diagnosis.
Yes, sir.
There are good studies from India, from Pondicherry, and from our laboratory also. And we are taking the cue from Pondicherry studies. We have done some work on use of LAMP also for diagnosis, and it is working out to be a very good method. And though we still are in the process of but then also even in urine samples we tried, and we found that we could detect the DNA there using LAMP, but not in all, but say around 60% of the cases we could detect, like what in Pondicherry also they could detect that in urine sample. So lab diagnosis is improving. Then there are other examples, EITB and so many other examples which we can use, and it is improving, and molecular diagnosis is changing the whole scene. And we are hopeful that we will improve upon it, and some way we can supplement on what radiological diagnosis.
Sir, Parija sir, you want to add on sir, your expert opinion on this?
Yeah, I think it's a good paper in the sense that the candidate has presented a paper on neurocysticercosis. I like because they are young people, they must understand that your intention is good, but you must supplement with your clinical report. That will confirm. Otherwise, you see, typically we used to tell the students, the old cases by CT or MRI, you cannot always difference between the cases of a neurocysticercosis. Typically, we will tell, but this is a children, otherwise it should have been adult. It is difficult to distinguish between a case of tuberculoma of the brain from that of neurocysticercosis. That is the major problem. But definitely, good, congratulations. But please keep in mind that to have such studies, you must take the help of laboratory diagnosis. Okay, thank you very much.
Participants,
Thank you,
any
sir.
questions?
We can
Thank you,
just
sir.
go on to the next speaker. Thank
Yes,
you.
madam. We'll go on to the next presentation.
Dr. Gobiru Shravani.
Yes, ma'am.
You can present now. Madam, are you able to share, madam?
Oh, 1 second.
Yes, madam. You can present, madam. Go to slideshow.
Oh, yes. You can see my slides, sir?
Yes, madam, it is visible, madam. You are audible. Please go ahead.
Okay. Okay, my topic is on prevalence of parasitic infections among patients attending tertiary healthcare center. Introduction. Intestinal parasites are widely prevalent around the world due to poor sanitation and personal hygiene, due to consumption of contaminated food and water, and tropical climatic changes. Globally, around 60% population have been infected with intestinal parasites, of which 21% are Indians, having high morbidity and mortality rate. These infections are causing major health problems in developing countries. The most commonly reported parasitic infections are Ascaris lumbricoides, hookworm, Trichoris trichiura, and Entaviva histolytica, among which E. histolytica is responsible for more than 15 million infections worldwide. The overall prevalence of parasitic infection ranges from 12.5 to 67% in different parts of the country. The major causes of gastrointestinal disturbances are enteric protozoa and soil-transmitted elements. According to WHO, although the disease burden due to intestinal helminths is high in India, only around 9.97% has received anti-helminthics. The aim and objective of the study is to find the prevalence of parasitic infections and multiparasitic infections among the patients attending a tertiary care center, and to find the prevalence of normal and abnormal constituents in stool among the patients affected with parasitic infections. The study type is a retrospective study. The study period is 2 years and 7 months at SRM Medical College and Hospital Research Center. Inclusion criteria is all stool samples suspected with parasitic infection are included without any bar on age or gender. Exclusion criteria, other samples suspected for parasitic infection were excluded from the study. The results showed that total samples were among study population were 3,899, among which only 4.6% were found to be positive. Among the affected study population, men were predominantly affected compared to women. The parasitic infections were found to be more prevalent in the age group of 21 to 30 years, around 19%, and followed by the age group of less than 16 years, around 17%. The parasitic infections with abnormal constituents in stool like mucus, blood, pus, and RBCs were found to be less compared to the parasitic infections with normal constituents in stool. Among the affected population, the protozoal infections were the major cause, followed by helminthic infection, and around multiparasitic infections were around 7%. Among the protozoan infections, Entaviva histolytica has the more prevalent, followed by Giardia lamblia, Entaviva coli, Trichomonas hominis, Idamoeba, and Blastocystis hominis. Among the helminthic infection, hookworm followed by Strongyloides stercoralis, pinworm, Hymelepsis nana, Tinea, Trichoris trichiura, and Ascaris. Among the multiparasitic infection, though there are 7.7%, the most common was found to be Entaviva histolytica, hookworm infection, Ankylostomum duodenale, Entaviva coli, and other infections were also seen. The discussion of the prevalence of positive parasitic infection in our study was et al. With 5.1 prevalence for over a period of 3 years. The protozoal infections were more compared to the helminthic infection in our study population, similar to the findings reported by Manoj Chitra K et al. And although the parasitic infections were less prevalent in our study, multiparasitic infections were predominant in our study population, which was comparable to the study by Mathuria YP et al., in which they reported prevalence of around 13%. The total prevalence in our study was found to be 4.6, which is less compared to the other prevalence studies in India. Due to failure, this might be due to failure of patients to recognize the symptoms at an early stage. Among the parasites found, Entaviva histolytica and hookworm predominated the intestinal parasitic infections. The early diagnosis, better sanitation, and effective health management are a prerequisite to control and manage infections in developing countries. These are my references. Thank you.
Thank you, madam. It's a good epidemiological based study. You described the finding of multiparasites.
Yes, sir.
What was the percentage and what was the combination of multiparasites?
So
multiple
most of
parasites.
the major parasites were found to be Entaviva histolytica along with Giardia, sir. Mostly they were found. Entaviva histolytica was 23%. Around of 7 cases, Entaviva had around 4 cases, with hookworm and Ankylostomum duodenale were major, and the Tinea, Blastocystis, and Trichoris very rare cases they were able to find, sir. Around 3 or 2, sir.
Sorry, sir, not audible.
Dr. Ravindran, you have to unmute.
Sir, please unmute. Dr. Ravinder.
Yes, sir. Only one sample you took, or consecutive samples you took, madam?
Only one sample, sir.
Yeah, because this kind of study, consecutive samples will be more valuable.
Yes, sir. I took only one sample, though there are many other repeated samples I excluded.
Yes. Add on, madam. And also insist again the same point Dr. Parija mentioned, Entamoeba histolytica bar dispar, whenever we use the microscopy. If we go to molecular, then we can specifically name them.
Okay, sir. Okay, sir. Thank you.
I think locally also you will have to take into factor the anthelmintic distribution and compliance through the primary health centers and other, because the other papers might be in other states. So we can also, it will be worth for somebody as an add-on to look at the distribution of anthelmintic locally in Chengalpet district, as well as look at the compliance also. Usually in schools, they are giving it orally to the children, monitoring and giving the anthelmintic. So then they amongst the children especially, there's a compliance. Also, another point I wanted to note about your study was 17% in children and 19% in adults among your total number of positives, I think you have calculated. But now it will also be worth to find out whether that 17% also the denominator should be among the people who are less than 18, in which case you're likely to get a different number, which probably may be a higher percentage in children.
Okay.
Because you should classify, the denominator should be the population in your study population, which is less than 18 years. Then you will get a different number, is my guess. Okay, provided you know, I'm thinking of what could have gone wrong, or probably. Okay, so these are the things. If there are any other questions, a good presentation, Aparna. Any other questions?
So, ma'am, I have one question.
Yes, sir.
Yes, sir.
Can I come in?
Yes, sir.
Go ahead, sir.
Yeah. I wanted to ask, like 23% of Entamoeba histolytica you have put in. So, how many of the patients you have followed it for? How many patients?
So, this is a retrospective study, sir. Actually, this is a retrospective study.
Okay.
Yes.
Okay. Okay. But then, what are the diagnostic method has been used to diagnose Entamoeba histolytica?
Sir, we use only microscopic examination for stools only. That's it.
But morphological examination is actually not very, sensitivity is quite low. So in those cases, personally, I feel that you have to write down Entamoeba histolytica bar Entamoeba dispar. Okay. For sure, we cannot say that it is Entamoeba histolytica unless until the patient is having dysentery or any liver abscess. If you are clinically diagnosing it, not clinically diagnosing, you have to confirm the laboratory diagnosis by doing additional tests.
Okay. Sir. Yes.
All right. Thank you.
Thank you, sir.
Thank you, madam. We'll go for the next presentation.
This emphasis on the diagnosis morphologically, you will have to put as Dr. Parija and the other experts. All of them are emphasizing the same thing. It can never ever be overemphasized. So hereafter, let us look at parasitology reports as in, you know, if it's morphological, let us also entertain the other species. And let us give a report accurately. Thank you very much, sir, for your valuable comment, Dr. Vinay.
Thank you.
ma'am.
Can we go on to the next speaker?
Yes, madam. We'll go to the next speaker.
Dr. Lavina Shekhawat. Madam, are you ready?
Good afternoon, everyone. My presentation is visible.
Yes, madam.
Okay.
Your audible presentation is visible.
Oh. Today I am going to present my paper on hydatid cyst. A patient was diagnosed in a Gitanjali Medical College, and I am presenting this report under the guidance of Dr. Sheetal, Assistant Professor, Department of Microbiology. Introduction. Hydatidosis, it is a chronic cyst-forming helminthic disease of domestic and wild ungulates. Human beings can accidentally harbor the cystic forms once infected, and it is caused by the infection with larval stage of dog tapeworms belonging to us. And it is also referred as echinococosis. There are two most important forms. One is cystic and the next is alveolar echinococosis. Cystic, it is caused by Echinococcus granulosus, and alveolar, it is caused by, there is another one is polycystic, which is caused by Echinococcus vocale. Epidemiology. It is a cause of substantial morbidity and mortality in most parts of Europe and the Americas. It accounts for more than 95% of estimated 2 to 3 million global cases. Cystic echinococcus is endemic in India, especially in the states of Rajasthan, Tamil Nadu, and Andhra Pradesh that are poor pastoral regions. Normally, the patient is asymptomatic with the presence of single cystic lesion in the liver. That is, right lobe is affected more than left lobe, because once the cysts are ingested, it enters into the small intestine, and via portal vein, it enters into the right lobes. That's why right lobe is affected more than left. Preliminary diagnosis can be done by USG, and it can be later confirmed by CT scan. Surgical excision is the treatment of choice, and anti-helminthic prophylaxis with albendazole is given for 3 weeks. In our hospital, a 29-year-old male who was driver by occupation presented with pain in right upper quadrant, which was radiating towards back, and abdominal fullness was present since 5 to 7 days. There was no history of contact with dogs or sheep, but the patient had a non-vegetarian diet, and there was no other significant past history. On examination, there was no pallor, icterus, edema, or lymphadenopathy. The patient had a regular pulse of 79 beats per minute and a regular BP of 120 by 80 mm of Hg. On per abdominal palpation, cystic mass was present in the right lobe of liver. It was palpated in the right lobe of liver. The physician investigated and he was advised for CT scan. On CT scan, it was found out that the patient had an enlarged liver with a cystic lesion that was measured 10.4 into 7.7 into 7.4 centimeter in the 7th segment of liver. Mesenteric and para-aortic lymph nodes were enlarged. The patient had a normal routine investigation of hemoglobin of 16 mg per ml. Total leukocyte was also normal, and a eosinophil count of 27.5 mm. Scan report of the patient. There was a presence of an echogenic cyst which was present in 7th segment of the liver. Treatment: the cyst was removed laparoscopically by deroofing, and omentoplasty was performed. The patient was advised for anti-helminthic prophylaxis with albendazole for 3 weeks. Post-surgical investigations were done. The centrifuge deposits of hydrated fluid was sent to histopath and our department microbiology. On histopath, there was a presence of multiple fragmentous cysts that measured 7 into 5 into 1.5 centimeter. It had a rough external surface with internal glistening surface with the presence of multiple cysts. In microbiological tests, we did there were 3 bed mount, LPCB staining, and modified Ziel-Nansen staining were done. In bed mount, we found colorless hooklets. In LPCB, we found blue-colored hooklets, and in Ziel-Nansen staining, pink-colored hooklets were present against a violet background. Conclusion: Hydatid cyst, it is a zoonotic disease which occurs in areas inhabited by the cattle-rearing population. Man is an accidental host, and the life cycle alternates between herbivores and carnivores. The life cycle, the liberated ova burrows through the intestinal mucosa and are carried out into liver by the portal vein, where they develop into adult cyst. Echinococosis is a very challenging clinical problem throughout the world. Early diagnosis by imaging technologies is helpful in endemic countries, and the eradication of the parasite surgically is essential along with the prophylactic dose of albendazole. These are my references. Thank you.
Thank you, madam. A nice study about the case.
Thank you, sir.
And so many stains you have done for microscopy. Which stain you prefer, and you advise others so that it can be very effective method to diagnose?
I prefer wet mount stain because in xylene cell staining, I tried different methods. Then only I could find this picture of xylene cell. Otherwise, I took 5% H2SO4 for the decolorization process, and in primary staining with carbon fission, I did it for 40 minutes. Then only I could get the pink colored booklets. It was tough for me to do ZN stain. So I prefer wet mount according to my personal experience. I don't know what is the ideal method.
Yeah, all are equally good, and better staining visualization can be done both in wet mount and lactophenol as well as in Zinlinsen. Even 3 can be done because these are cost-effective stains. Just 3 mounts also can be done conveniently. Madam, I need to ask about any molecular studies or serological studies were done.
No, sir, no serological and molecular techniques were done.
Because again, insisting as a microbiologist, we need to specifically diagnose using molecular techniques. And if it's not possible at your center, serological tests can be used for diagnosis. Rosie, madam, would you like to add?
Yes, I would think for the former part of the your discussion, I think essentially these are unstainable, and therefore what the stains are giving is only a background to what you're seeing. So wet mount, if you can just adjust the phasing contrast between the rest of the medium, then you will be able to see the refractile, it will enhance it. Otherwise, you would go in for anything which is dark, giving a background. So that you will be able to visualize. I think stressing on one stain or the other, I think is maybe not very useful at this stage. And as rightly said, serological investigations and other microbiological investigations should stamp it. Of course, hooklets are so classic. So at least here you have a microbiology diagnosis. You've seen it under the microscope. Hooklets are very classical. So yes, I would give it that you have done a etiological laboratory diagnosis. But also, there are now at this point of time, you can outsource. We have so many investigations and molecular biology. So just having the serum and the fluid and giving it, exchanging it at least for the sake of an academic exercise will be very useful to the youngsters. So that you can follow. I have one more question. That picture that you showed about hydatid cyst, is it your own, this particular case, or have you just given it as an example and a picture?
Ma'am, this was the case which was found in the gastroenterology department in our medical college.
Okay, it was a previous case. It is not this current case. It will be nice if you had taken a pic of this current case. Yes. Thank you very much. Any other questions from the rest of the audience?
Audience, any questions?
Ma'am, with your permission, can I do something?
Sure, sure.
Ma'am, we recently had in Stanley, we had a case of hydatid, ma'am. And we stained with gram staining, ma'am. It was wonderful, ma'am. Gram staining itself takes up the stain.
As gram positive or gram negative, please? I think it's the background that we were just talking about. Maybe the rest of the serum is, you please correct me if I'm wrong by, repeating. Because in Stanley also I have done. So it's possible that especially the rest of the background being proteinaceous could get stained. And
actually, ma'am, can I come in, ma'am?
Yes, please, sir. Please.
Actually, we have already we also have done the staining, but we only tried modified ZN stain. Okay, modified ZN parasite, so it can take up that stain. But the whole of the hooklet will not be stained properly. Exactly. So as you said, only the partially it can be stained.
Yes.
As such, staining will not be important, but the background will be very important in these cases.
Yes. Thank you, sir. Thank you.
We have come to the end of the...
Yes, ma'am.
So we have come to the end of the oral presentation. Now let us start with the poster presentation, sir. The first presenter for poster presentation is Dr. Vishaka Ashokan.
Yes, madam, she can, they can present.
Dr. Vishaka, are you ready with your presentation? Dr. Vishaka, are you there? Ma'am, shall we go to the next?
Yes, we can go to the next,
Next
madam, if the person is not ready.
Yes.
Dr. Hariharan,
Yes, ma'am.
are you ready, sir?
Yes, ma'am. I am ready with the
Sir, you can
presentation.
start.
Ma'am, I couldn't show my share my poster. Can you
Yes, sir,
share
we
my
will do it.
poster? Oh,
We
thank
will
you.
do it, sir.
Thank you, ma'am.
Hariharan, poster presentation. இல்ல இவங்க பண்றாங்க.
Yes, sir. Screen is visible, sir.
Yes, ma'am. Yes, sir. Thank you. Good morning, dear professors.
Yes, go ahead, sir.
Okay, sir. My poster title is Prevalence of Intestinal Parasitic Infection Among School-Going Children and its Correlation with Anemia. General introduction. An intestinal parasitic infection is a condition in which a parasite infects the gastrointestinal tract of humans. Intestinal parasitic infection is a major causative agent of iron deficiency, anemia, and malnutrition, especially among school-going children located in rural areas. In our study, mainly conducted to estimate the prevalence of intestinal parasitic infection and iron deficiency anemia among school-going children. This aim of this study is it is to find out the prevalence of intestinal parasitic infection among school-going children along with its correlation with anemia. Methodology is it's a cross-sectional study. Totally 85 school-going children were taken. It took conducted parasitic infection analysis and anemia. The stool examination was done by wet mount to detect the presence of O1 cyst and hemoglobin estimation by using XT1000I hemoglobin analyzer. Here, this is the wet mount picture. Here I identified Entamoeba histolytica or Entamoeba dispar. I would like to correct my mistake from here itself. As professor, you have already instructed me a lot of time. Not differentiate Entamoeba histolytica and Entamoeba dispar. So that's why I'm correcting my mistake now itself. This first picture shows Entamoeba histolytica cyst. Then second and third one are Hupeworm cyst. Then fourth one is Hymenopis nana cyst. Okay. So the results are, in my study, boys are more prevalent, that is 47%, and girls are 38%. In anemia status in my studies, 40 patients have come with low hemoglobin level, that is 47.05%. The total intestinal parasitic prevalence is 8.23%. Among them, Entamoeba histolytica, Entamoeba dispar comes with 57%, and hookworm infection with 28.6%, and Hymenopis nana with 14.3%. So my conclusion is, since intestinal parasitic infection can be controlled by following a few healthy hygienic practices like regular deworming, awareness and hand washing technique, intake of iron deficiency and folic acid supplement irrespective of the nutritional status. Usage of proper toilet can reduce the incidence of iron deficiency anemia caused by intestinal parasitic infection in school-going children. Thank you, sir.
Thank you, sir. You have studied about the anemia
Yes,
in
sir.
various worm infestations.
Yes.
And what was the correlation with hookworm and correlation with Entamoeba histolytica? Since a lot of cases you have got with Entamoeba histolytica, whether the children or adults were suffering with anemia or not?
According to my study, the age group between 16 to 18 years with more cases, that is almost 36 cases comes between 16 to 18 years. Within this population, I got more number of cases with Entamoeba histolytica and Entamoeba dispar cases, along with anemia. Then
So
second
anemia,
most common.
yes, sir. Anemia is common in both histolytica bar dispar,
Yes, sir.
as well as in cases of hookworms.
According to my study, it is both between these. Yes.
Okay. Any questions from the audience? Sorry, Hari sir, you like to comment? Any questions from the audience? If there are no questions, we can move to the next presentation.
So the next presenter is Dr. Ramya.
Yes, ma'am.
Are you ready, madam?
Yes, ma'am.
You can start your presentation.
Audible and poster is visible.
Okay. Good morning. Myself, Dr. Ramya. Today, I'm going to present about a case report of a Jane nasal polyp, which is caused by a spelt parasite, Rhinosporidium seaberry. Rhinospordiosis is a chronic granulomatous infective disorder that is caused by Rhinosporidium seaberry. It usually presents as a soft polypoidal pedungulated sessile mass arising from the nasal mucosa. Most common sites of occurrence of rhinospordiosis are the nasal cavity and the nasopharynx, and it can also be found in the conjunctiva, larynx, and maxillary sinuses. So here, a 72-year-old male, he presented with the complaints of nasal mass, nasal obstruction with nasal discharge, and occasional epistaxis. And a tissue biopsy was done, and the tissue was sent to the laboratory, where we macroscopic.
Sorry to interrupt, ma'am.
Yes,
Dr. Fayaz, can you please stop presenting? Dr. Ramya is presenting, so Dr. Fayaz, you stop your presentation. Ramya, ma'am, can you again present it, ma'am?
Yes, ma'am. Ma'am, my poster is seen, ma'am.
No, madam, no.
Dr. Fayaz, you have to close the sharing so that the other person, Ramya, can present. Dr. Fayaz, you have to put the command stop sharing so that she can present.
Sir, my poster is seen, sir.
No, madam.
Yes, madam.
Yes,
Now you can see, madam. You can go ahead, madam. You are audible, your poster is seen.
Okay, sir. Good morning, myself, Dr. Ramya. Today, I'm going to present about a case report of Jane nasal polyp caused by a spelt parasite, Rhinosporidium seaberry. Rhinospordiosis is a chronic granulomatous infective disease that is caused by Rhinosporidium seaberry. It usually presents as a soft polypoidal pedungulated sessile arising from the mass arising from the nasal mucosa. The common sites of occurrence of the rhinospordiosis are the nasal cavity and the nasopharynx. It can also be found in the conjunctiva, larynx, and maxillary sinuses. In this case, a 72-year-old male, he presented with the complaints of nasal mass, nasal obstruction, nasal discharge, and occasionally he had epistaxis. And a biopsy was done, and the tissue was sent to the laboratory. Macroscopically, we received multiple fragments of grayish to white brown colored soft tissue bits. And when it was stained with the hematoxylin eosin stain, microscopically we can able to appreciate a thick-walled sporangium that is containing numerous basophilic spores. And after that, we extracted the genetic material of this. We confirmed it as Rhinosporidium seaberi by this HE staining. And then we extracted the genetic material of this Rhinosporidium seaberi and we did whole genome sequencing. After sequencing, we used this BLAST-N, that is the basic local alignment search tool for the nucleotides. With the help of the sequencing, we can able to compare the similarities between the what we have sequenced and able to find out the similarity between the organisms which has been already sequenced there. So here we can able to find out that Dermocysterium species was about 63% similar. There was a similarity between this Rhinosporidium seaberi and Dermocysterium species, and 51% similarity was found between Ochlacida and Rhinosporidium, and 47.1% similarity was found between Cyanobacterium and Rhinosporidium seaberi, and 24.85% of similarity was found between chloroplast of a flowering plant and the Rhinosporidium seaberi. So, in conclusion, this phylogenetic analysis of this Rhinosporidium seaberi or 18S rRNA gene suggests that this culture-resistant organism is not a member, but rather this is the first known human pathogen from a novel clade of aquatic protestant parasite. So this is a branch in the evolutionary tree near the animal and the fungal divergence. So the similarity between the Rhinosporidium seaberi and the Dermocysterium species was around 63%. Sir? Sir?
Yes. Is there any questions from the audience?
Hello. Any questions from the audience? Madam, I have a question. Again, what is the clinch? You have told so many points. What is the clinching point to tell it is a parasite?
Because this Dermocysterium is aquatic protestant parasite, sir. So the similarity between the Rhinosporidium and this Dermocysterium is around 63%, sir.
Because of that, you are classifying Rhinosporidium also into the
Yes.
parasite.
Yes, aquatic protestant parasite, yes, sir. It is coming under the order of Dermocysterium.
Okay. Any questions from the audience? Madam, like to add to it?
Yeah, you have done the sequencing for your parasite.
Yes, ma'am. Yes, ma'am.
And now it should be identical to the sequence that's already registered for the parasite, isn't it?
We
In
did
the
the
official
sequence.
sites. Yeah, you got
Yes.
the sequence. It's already, it is already available, isn't it?
We did the sequence and we got it by using the universal
I
primer.
understood. I understood
Okay,
that. And you
okay.
compared it with those of other organisms
Yes, ma'am.
whose sequence is already known.
Yes, ma'am.
Isn't it?
Already. Yes, ma'am. It's already.
Similarly, was
Yes, ma'am.
Rhinosporidial sequence already known by others prior to this is my question. Like, I
No.
want to know if anyone else had sequenced Rhinosporidium, or you are the first person to sequence Rhinosporidium.
We have searched for the literatures, ma'am. We didn't find any sequences regarding the Rhinosporidium. Yes, ma'am. Yes,
Prior to your search, your
Yes,
sequencing, you did not find any recorded evidence of Rhinosporidium sequencing.
Yes, ma'am.
Okay. Have you really done a good search? Then I should congratulate you for that, for attempting to sequence. And
Thank
if
you.
there are any, yes, ma. And if there is anybody in the audience or even otherwise who can, actively search on this and see if this study is if this sequencing is the first of its for this particular species, then it will also aid her to record it. Okay. So I would also urge others to check out if she's the first or the first from India or something should be there for us to document here. Okay.
Experts, any suggestions?
And any other questions also, we'll just entertain.
Questions from the participants or any suggestions from the experts regarding this study?
But rhinosporidium, as far as I know, has already been sequenced. And that is why when they compared the genome, that is why it was thought that it is a parasite and not a fungal. Previously, it is actually still mycology people are diagnosing rhinosporidium. We are not actually diagnosing rhinosporidium.
Yes, sir. If it is well characterized, then we need to diagnose and classify under parasitology.
Yes.
Okay, sir.
Any more suggestions or questions? We'll move on to the next presentation.
Thank you, sir.
Dr. Ramya, can you please stop your presentation?
Yes, ma'am.
Next presenter is Dr. Fayaz, ma'am. He is not there, ma'am. He is told that not able to join. So shall we go to the next presenter, ma'am?
Yes, madam. We can go for the next one.
Dr. Saraswati.
Yes,
Dr.
madam.
Shashwati.
Dr.
Yeah, you can start.
Dr. Shashwati.
Yeah, yes, ma'am. You can start your presentation. Shashwati, yes, ma'am.
Madam, I cannot show you my presentation because I'm in a COVID lab and working here at the present moment. Could you just show me my present?
Yes, ma'am. 1 second, we will put.
Yes, ma'am.
Yes. Ma'am, you are able to see, isn't it, ma'am?
Yes, madam. You are audible. Your
So, madam,
Ma'am Shashwati, ma'am, you can start your presentation.
Yes, madam. You can start.
Ma'am Shashwati, ma'am, are you here, ma'am?
Shashwati, madam, you can start your presentation.
चल क्या हो गया भाई
31 मार्च
हैं
मार्च
अच्छा
Ma'am Shashwati, ma'am.
Madam
Shashwati, ma'am. You can start your presentation, ma'am.
You can start your presentation, madam.
Okay. Phyllitis is an endemic problem in India and it's mainly and Brugia malai. Sir, one day a patient, a 30-year-old patient, she had come to the Department of Surgery actually, with a painless lump, which was the breast lump, which was previously painless, but for the last 15 days, she started having side pain. So therefore but all her vitals were normal, everything was normal, no cause FNSC. And the pathology department called us also, the microbiology, as to what the cause was. On finally aspiration, we could that when the smear was made and Leishman stain was stained with Leishman stain, we found a micro amongst the cellular. It was just a cellular smear and a microphyllaria was identified. So it's actually this microphyllaria. We found that there was absence of any nuclei in the tail, and it was a sheath microphyllaria. So probably, actually, it was belonging to the Ucheridia branchophylla. So, madam, the breast is a common site, but being isolated by FNSE, microfilaria isolated by FNSE, it's a rare incident. So therefore, Madam, I made this my case presentation actually. And the lump was, madam, it was freely mobile. It was not attached to the skin. The axillary nodes were free, and the lump was in the left breast, while the other breast, it was absolutely normal, and the lump was also not attached to the underlying skin. So, and it was in the left upper subareolar region. So this was actually the case, madam.
Madam, have you, it's a nice case you have studied. I have one question. What made you to suspect
yes, sir,
filariasis in this breast lump from this lady? What was
actually,
your,
sir, breast is a
one of the known site? What
filaria
else
is
made you
anything
suspect?
there. So it was a weird, so and it was freely mobile, not attached to the underlying skin, not attached to the. We were thinking of fibroadenoma first. So then the patient was because West Bengal is it's an endemic area. Being Bardhaman is quite close to Bankura and all. So filaria was one of our DD. So that's the reason we thought probably it could be filariasis.
Okay, madam.
Therefore, we just on a basis of filariasis, therefore we just did it.
A good case.
Purulia, Bankura, sir, it's quite adjacent to Bardhaman. So therefore.
Yes, madam. Yes.
Madam,
So
sir?
you had a suspicion because of
Madam,
your location as well as common site, one of the site for filariasis. Rosie, madam, you can add more.
Yes.
It's a good case. Does anyone else know if any report of filariasis affecting the breast? And so some search also can be done. If there may be other cases, so we can just search for it as an exercise.
Okay.
And also, what I found was the skin is free. This we don't see in case of filariasis of the lower limbs or scrotum, which we are more familiar with. We find that the skin is also involved. The skin is not free.
Yes,
because
madam.
the lymphatic drainage of the subcutaneous tissues would tether the lesions altogether because of the proteinuria. Yeah. So in this case, you find that the skin was free and it was a mobile
Absolutely.
lump. It was a mobile lump. So
Yes,
that
madam.
makes it a little peculiar to me. So it's worth following it up. And was a lumpectomy done for this patient?
Yes, madam. It was done. And the patient following lumpectomy, she was also given medical was given.
Yes, a DEC would have been given. Now, the thing is, if you had shown me a picture of the excised lump, it would have been very nice, where we would have seen whether it is, well delineated, capsulated, etc. Anyway, in any case, please follow this up. Congratulations for the good case that you chanced upon and could present to us. Is there any other questions from the audience?
If there are no questions, we can
go on to the next one.
Yes, madam. We'll go on to the next
Thank you,
presentation.
Thank you.
Thank you. Thank you.
Thank you, madam. We can go for the next presentation.
Can I audible? Yes, ma'am.
Yes, madam. You can present, madam.
Yeah, madam. You can start.
Good afternoon, ma'am. Okay, sir.
Carry on, please. Yes.
I'm Dr. Vishwapriya, first year postgraduate from the. Okay, ma'am. Okay. Okay, ma'am. I'm Dr. Vishwapriya, first year postgraduate from Department of Microbiology in GN Government Mohan Kumar Mandala Medical College, Salem. I'm going to talk about malaria vaccine. The first challenges to the eradication of malaria, the causes are insecticide resistance, emerging and anti-malarial drug resistance, and the presence of asymptomatic and submicroscopic infections. Pyrethroids are the most effective for long-lasting insecticide nets, but 30 countries develop resistance for pyrethroids also. In Southeast Asia, where mutant parasites resistant to anti-malarial drugs are more likely to survive due to the lower level of acquired immunity, poor adherence to the administered drugs, and high parasite burden. So we will need ideal malaria vaccine to eliminate the malaria, also prevent the transmission. Ideal malarial vaccine should be reduce the transmission, should reduce the clinical disease severity, prevent the infection at the first instance. But if this is not possible, should decrease the intensity of the infection and should be effective in preventing the malaria transmission. Goal of malaria vaccine initiative is by 2025, a malaria vaccine with 80% or greater efficacy that at least lasts for 4 years. Coming to the novel strategies of malaria vaccine, these include 4 approaches. First one, the parasite-focused approach. Second one, reverse vaccinology. Third one, structural vaccinology. Fourth one, immunofocused approach. Now coming to the reverse vaccinology, it starts with obtaining the genomic sequence of the pathogen and identifying all open reading frames to predict protein antigens. The predicted antigens are subsequently expressed in suitable vectors to produce the recombinant protein. First used bacteria under this category approach is Meningococcus cerebrus B. So next, the structural vaccinology mainly to overcome the antigenic variations. The second one is the path of malaria vaccine development. It includes the erythrocytic stage, and the transmission blocking stage. First one, the pre-erythrocytic stage. Especially the liver stage vaccines are prevent the malaria in human host by producing antibodies against the infective form of sporocytes. However, because of the higher rate of replication of sporocytes, a single parasite may be sufficient for the infection to proceed to the blood stage. The liver stage Plasmodium falciparum is an attractive therapeutic target for the development of both anti-malarial drugs and vaccines, so as it provides an opportunity to interrupt the life cycle of parasites at critical earliest stage. The most advanced pre-erythrocytic vaccine candidate is ODS, which consists of truncated circumsporocyte protein of Plasmodium falciparum directly fused to the hepatitis B surface antigen. This vaccine shows 30 to 50% efficacy only in human field trials. In Africa, second one, blood stage. These are designed to elicit anti-infection and anti-malarial response. Underlying principle of this strategy is that if vaccine would block the invasion of erythrocytes by merocytes, it would prevent malarial disease. At present, several blood stage vaccines are in clinical trials, especially phase 1. The surface proteins are mostly present in the Plasmodium falciparum. Examples are erythrocyte binding protein 175, glutamate-rich proteins, merocyte surface proteins, serine repeat antigen 5. All these antigens are highly expressed on the surface of merocytes. Also, Plasmodium falciparum erythrocyte is only present in the infected RBCs. So next, coming to the transmission vaccine stage. This prevents the parasite development in the mosquito midget. Aim of this vaccine is to induce antibodies against the sexual stage of antigens to block oocyte transition, so stop the subsequent generation of infective sporocytes, so prevent the transmission between mosquito to human. These vaccines are intended to protect the entire communities from the infection. Examples of this: post-fertilization antigen Plasmodium falciparum 25 and 28. Pre-fertilization antigens include Plasmodium falciparum 230, Plasmodium falciparum 48, and Plasmodium falciparum 48. Now coming to the RTS present and future, the most recent developed recombinant vaccine. The ODS is attempted by fusing the protein circumsporocyte protein with a surface antigen from hepatitis B, hence creating more potent and immunogenic vaccine. This vaccine shows 30 to 50% protection in human field trials in Africa, 43% in children aged 5 to 17 months, and 27% in children of 6 to 12 weeks old. Goal by 2025, cases that at least for 4 years. Now, project in India and the status about malaria vaccine includes: first, Jaiva 1. These target antigens are Plasmodium falciparum, merocyte surface protein 1, and Plasmodium falciparum receptor binding F2 domain of erythrocyte binding antigens. This target antigen given in IM route under oil emulsion, given at 3 doses, includes a day 0 at 10 microgram of target antigen, a day 828, 25 microgram of target antigen, on a day 180th day, 550 microgram of target antigen was given, but no serious side effect was noticed still.
but poor immunoresponse to Plasmodium falciparum myelocytes protein 1. Now next is JVAC2. Myelocyte surface proteins are used under Plasmodium falciparum receptor binding F2 domain of erythrocyte binding antigen 175 used as a target antigen in this group, given in IM route and are manufactured for phase 1 trial. Thank you, sir.
Thank you, madam. You have listed a lot of candidate vaccines. Out of this, which is most probable to be used in the field and at large for the community purpose? Which one you recommend for the field trials? You have listed a lot of vaccines, no? Which will be more effective?
Sir, ODS AS01, sir.
Any specific reasons, madam, why it's effective?
Sir, this vaccine should only come under phase 3 trials, sir. Also, efficacy was up to 50%. No other vaccines are should under this efficacy, sir.
It's having efficacy of more than 50%. Is that right?
Yes, sir. Yes,
okay.
Sir.
Rosie, madam, you would like to add?
Actually, a good attempt to classify and update. Do you think if they receive the vaccines, if you can look at them as a candidate vaccine, it should be a vaccine which the subject after receiving should be able to even take another type of vaccine. Okay, another vaccine should also be accessible in case they did not develop the immunity. So that should also play a role in identifying, because we have only 50%, isn't it? So till we get a vaccine which has a very high efficacy, the factors to look at. If there are any other questions, we'll entertain. Otherwise, we'll move on to the next speaker.
Nice thematic presentation, madam. We'll go for the next presentation.
Yes.
Ma'am. Dr. Shikha Pandey, ma'am. Dr. Shikha Pandey, are you here, sir? Dr. Shikha Pandey. Ma'am, shall we go to the next presenter, ma'am?
Yes, madam. Yes, madam.
Dr. Usha Verma. Dr. Usha Verma.
Ma'am, please upload the poster.
Oh, yes, ma'am. Yes, ma'am, we will do it. Usha Verma.
हाँ उसी तुम्हारा पोस्टर चल रहा है, ज्वाइन करो फटाफट वो पोस्टर
भाई yes madam, poster is visible. You can go ahead, madam, you can present.
Sir, can I, because my student cannot connect.
Organizers, okay.
Sir?
No, it's a poster.
Yes, sir. Yes, sir.
Yes, madam, you can continue.
Sir, this is a case report of a co-infection of Strongyloides stercoralis infection. A 4-year child admitted in a tertiary level hospital. He was having continuous diarrhea, vomiting, and he was diagnosed as having a co-infection of the diaphragm and anything else. Then the larvae of the Strongyloides stercoralis, and patient was diagnosed as a celiac disease patient. As on serological examination, as this serology was positive for celiac disease, and later on patient was treated with albendazole and gluten-free diet was given, and patient was referred to the PMC center of the our institute.
So basically, in this case, we want to emphasize that in a celiac disease patient, apart from the gluten-free diet, we have to think of the co-infection, and strongyloid stercolis infection is very rare in Western Rajasthan. I think it is the first case report which you have reported from the Western Rajasthan. Thank you.
Thank you, madam. Good case study. What made you to suspect strongyloidosis in a celiac disease patient?
So it
Usually
is
we don't suspect strongyloidosis.
just a accidental finding, because patient was diagnosed, patient was referred to the tertiary care center for the SEM as a severe energy malnutrition. Even we substituted his gastric aspirate sample for CB-NAT, and CB-NAT test was negative. All other serological tests were negative, and accidentally on the stool examination, this strongyloids larvae were detected, and patient responded very well to the treatment, and then it was discharged.
Is malnutrition status improved after the treatment, madam?
Yeah. Patient was, we are having one unit of which are dealing with protein energy malnutrition patient, children, and she was admitted there for a long time. But patient responded very well.
That's nice, madam. And these days, strongyloidosis cases are on the increase for various other conditions.
HIV negative. Patient was HIV.
That is very, very unique finding. So HIV negative, but due to other comorbid condition like celiac disease, he has developed strongyloidosis. And we have to be on the lookout for strongyloidosis because many conditions it is coming up. Even in this presentations, we were seeing post-renal, some long-term steroids, it's coming up. So nice case study, madam. Thank you. Madam, Rosie, madam, you need like to add on?
I just would like to know whether the burden was high or was it just minimum, the larvae burden? Was it more in number or was it?
High burden. Hello?
Yes,
yes, madam. Yes, madam.
Was it more in number? Hello?
Yeah. Even after the treatment, in the
how many larvae?
Madam is asking how many larvae estimated?
Total number I cannot estimate, but in one field we were seeing 2 to 3.
Yes, madam. Any other questions, madam? From the participants, anyone wants to comment or any clarifications? Thank you, madam. We'll move on to the next presentation. A nice case study.
Thank you, sir. Thank you. Sir, however, one more is left. Vishakha, one number.
Vishakha.
Organizers.
Yeah. Was not uploaded. First number posted.
Vishakha. Dr. Maidin has started, ma'am, so we will wait.
Yes, the first one.
Okay, ma'am. Yes, madam, it's visible. You can go ahead, madam.
Good afternoon, sir. I'm Dr. Maidin Bibi, postgraduate.
Yes.
Hello, sir. Am I audible?
Yes, madam, audible. Your poster is visible. You can go ahead, madam.
Yes, sir. Okay, thank you, sir. My topic on e-poster presentation is prevalence of intestinal parasitic infections in hemodialysis patients from developing countries, a systematic review. Introduction. Intestinal parasitic infections is the main cause of gastrointestinal complications in hemodialysis patients due to their impaired immune system. Patients with chronic kidney disease or end-stage renal disease have a weakened immune system due to uremic toxins. This weakened immune system increases susceptibility to various infections. And infections by bacteria, viruses, fungi, and parasites And these infections are the second most common cause of death among patients with ESRD undergoing hemodialysis. Globally, intestinal parasitic infections are among the most important causes of morbidity and mortality, particularly in low and middle income countries. According to WHO reports, over 1 billion people are affected by intestinal parasitic infections throughout the world. Therefore, an accurate and a timely diagnosis as well as treatment of these infections is essential to improve patient health and quality of life. Herein, we performed a systematic review to evaluate the pooled prevalences of intestinal parasitic infections in hemodialysis patients. Methodology. A systematic literature search was conducted as on PubMed, Web of Science, Google Scholar, Medline databases, and relevant studies were selected up to November 2020. A meta-analysis model was used to estimate the pooled prevalence, odds ratio, and confidence interval. The totally 26 studies were included from various countries, 13 using case control design and 13 using cross-sectional study. The population of the study in case control design was 1,225 hemodialysis patients and 1,099 healthy controls. In cross-section study, a total of 1,854 participants were included. The main characteristics of the included studies are presented in Table 1 and 2, mentioning the first author, country, year of publication, sample size, diagnostic method, study design, number of infected people in cross-section studies, and number of patients and healthy people in case control studies, and the type of parasites detected in each study. You are seeing Table 1, summary of studies with cross-sectional design investigating prevalence of IPIs in hemodialysis patients. This table shows the summary of studies with the case control design investigating the prevalence of intestinal parasitic infections in hemodialysis patients and control subjects. Coming to the results, in studies with the case control design, the pooled prevalence of intestinal parasitic infections were estimated to be 32.48. Here you are seeing this is the forest plot for hemodialysis patients in case control study. This diamond shape is the pooled prevalence you are seeing at the end of the plot, which is 32.48. You are seeing all the individual studies which are clumped, and the average is taken as a diamond shape. The study, all studies are statistically significant. That is, these studies do not cross the line of null effect. Here, then, in studies with the case control design, the pooled prevalences of intestinal parasitic infection were estimated to be 15.38% in healthy controls. This forest plot shows the controls in case control studies. The pooled prevalence is 15.38. Then this is the forest plot for cross-sectional study. The pooled prevalence here is among the hemodialysis patient is 33.93. Of the 13 articles analyzed that used a case control design, 9 reported a significantly higher prevalence of intestinal parasitic infections in hemodialysis patients in comparison to the controls. Our systematic analysis revealed that intestinal parasitic infections had an overall prevalence of 29% in hemodialysis patients in developing countries included in our study. Our findings showed that Cryptosporidium species had a higher prevalence in hemodialysis patients, being 13% than in controls 2%, with the prevalence rate in dialysis patients being similar to that reported in other immunocompromised patients in various other studies like HIV patients, parasitic infections in HIV patients. Our research demonstrated that Blastocystis species as another parasite with a high prevalence, second to Cryptosporidium, high prevalence rates in hemodialysis patients. This Blastocystis is a molecularly divergent microorganism with many subtypes reported among humans and animal hosts. The role of this blastocystis in the development of inflammatory bowel disease and irritable bowel syndrome was also reported recently, warranting further studies. Our analysis found that prevalences of Giardia lamblia, Entamoeba histolytica, Entamoeba dispar, and Helminths were no significant higher in hemodialysis patients than in controls. Although the prevalence of helminthic infections was very low among dialysis patients, one should not neglect these infections as they can still cause hyperinfections in immunocompromised individuals. Conclusion: the result of this systematic review revealed a higher prevalence of intestinal parasitic infections in hemodialysis patients compared to controls, especially of severe Cryptosporidium species infection. In endemic regions, providing a suitable personal hygiene and also regular health checkups for intestinal parasitic infections in hemodialysis patients presenting with gastrointestinal symptoms are highly recommended. Thank you, sir. Thank you, ma'am.
Thank you, madam. A nice meta-analysis type of study. And you have referred a lot of articles with the case control study design. What was the prevalence of strongyloides in this hemodialysis patient? Everyone talked about strongyloides in the previous presentation also. Apart from Cryptosporidium and other protozoans, a very significant finding about blastocystis hominis was there. What was the percentage or significance of strongyloides in this analysis? Whether it was present or
most of the studies took account of Cryptosporidium species and some other parasites, sir, but not they are taken strongyloids, so I didn't mention about it, sir.
Okay, madam. A nice comparative study. Rosie, madam, you would like to add on?
Nothing, sir. No comments. It's good study.
Yes.
Can we move on? Is there any other speaker?
Yes, madam.
Any other posters there? Okay.
Thank you, sir. Thank you, madam.
Thank you, madam. Nice study, comparative study. We'll go for the next presentation.
Dr. Maideen, can you stop your presentation? Dr. Priya, are you ready, ma'am?
Yes, ma'am.
Dr. Priya?
Yes,
Yeah, you can start your presentation.
Is my screen visible, ma'am?
No, ma'am.
How to share my screen, ma'am?
Shall we share the screen, ma'am?
Yes, ma'am. Please.
Yes. 1 second, we will do it, ma'am. Ready? Are you able to see, ma'am?
Yes, ma'am, but I want to zoom it.
Zoom in.
Yes, okay.
Yeah.
Good evening. I like to thank the organizing committee and my HOD, Dr. V. Dilirani, for giving me this opportunity. I'm going to present a study on the prevalence of cryptosporidiosis in renal transplant recipients in a tertiary care center in North Chennai. Introduction: cryptosporidiosis is intestinal opportunistic infection commonly occurring in immunocompromised patients, including renal transplant recipients receiving continuous immunosuppressive therapy. The clinical manifestations of cryptosporidiosis are dependent on the immunological state of the host. Infection among the immunocompetent host resides in the area that is typically self-limited, whereas in immunocompromised host, the infection may be protracted and life-threatening with no reliable antimicrobial therapy. Hence, this study plans to study its prevalence. Aim of the study is to study the prevalence of cryptosporidiosis in renal transplant recipients. Materials and methods: 90 stool samples were studied in detail. 2 consecutive samples from 30 renal transplant patients and 1 sample from 30 healthy adults as control. Stool samples were subjected to microscopic examination by direct wet mount method. After stool concentration by formal ether sedimentation technique, modified acid fast staining was used for detection of oocysts of Cryptosporidium species. In light microscopy, oocysts were viewed as pink to purple partially stained spherical bodies measuring 4 to 6 micrometer in diameter against a blue background. This is the figure showing modified acid fast staining. Out of 30 renal transplant patients, 8 showed positive for Cryptosporidium, and in the control group, all samples were tested negative. So, conclusion, the prevalence of cryptosporidiosis in this study was 26% among renal transplant patients. Since this infection can be effectively managed if diagnosed early, a high index of suspicion by the practitioner for its diagnosis is important. It is recommended that in all transplant recipients presenting with acute diarrhea, modified Ziehl-Neelsen staining should be done to rule out cryptosporidiosis.
Thank you, madam. It was a nice study, and I appreciate the samples, consecutive samples were taken. That is ideal sampling for this kind of a study. And any concentration method was used, madam, since you have extensively studied about Cryptosporidium?
Sedimentation, I used the sedimentation technique by formal ether method, sir, because the stool has at least minimum of 50,000 oocysts has to be present to be seen in acid for staining. So I used sedimentation technique.
It would have been nice if you would have also used Sheether's sugar flotation.
Okay,
specific
sir.
For Cryptosporidium. This study is for Cryptosporidium. That would have been appreciable.
Okay,
madam, you would like to add, madam?
No, I have a good presentation. No more comments. Any other comments from anybody else?
Ma'am, I have one comment.
Yes, sir. Please, sir. Nice, sir. Please.
Yeah. The thing is that you are looking for these patients, renal transplant patients. Why only have you looked for Cryptosporidia? Why haven't you looked for Isospora or Cyclospora? Only Cryptosporidium purely?
Sir, in 2 cases, strong elites were seen, but not Isospora or Cyclospora.
Was not there. Okay. Okay. All right. Thank you.
Sometimes past
yes, madam.
positive reports are there, especially Cryptosporidium.
Ma'am, not
then students does this mistake.
What,
the concentration of the stain is not appropriate, then false positivity is being reported.
Yeah. Actually, especially E cells can be reported as Cryptosporidia by students. So actually, we require 2 experts to see Cryptosporidia. Only if the students are seeing, then because of over enthusiasm or to give more positivity, the E cells can be reported as Cryptosporidium. So have you measured the size, our participant?
Sir, we do simultaneously gram staining, but these are appeared as ghost cells in gram staining.
Ghost cells?
Yeah, it won't take up the stain. The Cryptosporidium will not take up the gram stain, whereas these cells will take up the gram stain. So I did a simultaneously gram staining to rule out yeast cells.
No, whether how many microscopists has seen the slides?
How many?
Microbiologist or microscopist?
Apart from you,
Yeah.
Who else have seen?
Assistant professors and associate professors confirmed it, sir.
Okay, fine.
So you have senior members, okay. Because I also abstain from commenting on that. Very often, yeast cells, Candida species will appear, but they'll appear more darkly stained and more uniformly stained.
Right.
Another feature is that the size will not be uniform if it is yeast cells.
Absolutely,
Since yeast
absolutely.
cells during the growth will be of various sizes, so you will find a range of size. Whereas in Cryptosporidium, you will find them all to be of identical size.
Yeah, and Cryptosporidia appear as a rough, ground glass type of appearance.
Yes, exactly.
But E cells will appear as very smooth. Okay.
Modified trichome can also be used.
Yes.
Thank you very much, sir.
Okay, thank you.
Thank you for the discussion.
Thank you.
Dr. Ravindran, is there anything more to?
Yes, that's all, madam. Sir has added a valuable point along with Madam Professor also.
Yes.
We'll go for the next presentation.
Yeah, sure.
Next presenter is Dr. Lakshmi Shankar. We will do the presentation. We will put the slide.
Thank you, ma'am. Am I audible, sir?
Yes, madam. Yes, ma'am.
Yes.
Thank you. Thank you, sir. Thanks. A warm good evening to one and all. I am indebted to presenting this case to my seniors, including Beena Madam, who is my HOD. So today's case discussion is Phonius gangrene, a twisted presentation of Enterobius vermicularis infection. So Enterobius vermicularis, also known as pinworm, is due to the pin-like structure and seen in the female worm. It is one of the most common nematode infections worldwide, and humans are the only known natural host. It is most commonly seen in children, and household crowding, immunosuppression, and poor personal hygiene are identified as major risk factors. Coming to the case report, it is a 66-year-old male who was presented to our surgery casualty with a gangrenous scrotum. He was a diabetic who was on oral hypoglycemic agents, and he was complaining of pruritus ani for the last 2 years. And there is also, on history taking, there was a history of perianal abscess, which was he was having 2 months back, which was ruptured and formed an ulcer, which extended to the scrotal area, which is now presenting as a gangrene. Coming to the lab diagnosis, lab investigations showed hemoglobin 9.8 gram percentage, fasting blood sugars 169 milligram percentage. Total count was elevated 12,600 with a polymorph predominance of 92%, and even with a higher range of eosinophils and an ESR of 130 millimeter per hour. Coming to the microbiological, were isolated from the gangrenous area, and it was identified as Enterobius vermicularis based on the morphology. On gross appearance, it was of size 10 millimeter long and 5 millimeter thickness, white thread-like worm, and end of the worm we could identify the cervical alley as well as the double-bulbed esophagus, as it is very evident in the picture. And in the middle portion, there were the reproductive and intestinal portions were clearly visible. And then the posterior end, there was a pointed, curved posterior end was visualized. On pressing the middle portion, that is the reproductive organs, the release of eggs were demonstrated, which was a plano-convex shaped typical eggs of the Enterobius. The other specimens received in the lab was pus aspirate, which was collected from the gangrenous area, which grown a multi-drug resistant Escherichia coli, which is sensitive to piperacillin, tazobactam, and meropenem. And stool examination revealed eggs of Enterobius vermicularis. Aerobic blood culture was sterile after 48 hours of incubation. And the gangrene was managed with surgical debridement and later on with a flap. And piperacillin, tazobactam, according to the susceptibility report, was given to the patient. And the pinworm infection was treated with albendazole, 400 milligram single dose, followed by a repeat dose after 2 weeks. And the diabetes was controlled with an injection insulin, and his blood sugars were under control after 2 weeks at the time of discharge. So he clinically improved and with a very good blood sugar level, and the wound was healed with less of a scarring, and he was discharged after 2 weeks. On review, there were no eggs demonstrable in the stool samples. Coming to the conclusion, even though enterobiasis is an infection of children, it is seen in all kinds of debilitations like diabetes, old age, or immunotherapy. In this case, it is the chronic excoriation by the pinworm infection and the constant trauma produced due to that led to the secondary bacterial infection, which is here Escherichia coli, and which act as the actual reason for perianal abscess and later on Phonius gangrene, which is a very serious condition. So with this case, we can understand that even though the pinworm infection very common can lead to more serious infections like Phonius gangrene, even though it is not directly involved through acting as a cause for a secondary bacterial infection. Thank you so much.
Thank you, madam. Are there similar cases reported often?
Yes, sir. Actually, 2 cases of perianal abscesses were reported, and also subcutaneous nodule presentations are also reported for enterobius. But Fournier's gangrene, I couldn't find any case in the literature.
Because incidental finding along with the Fournier's gangrene due to his diabetic condition, or as you told, it can cause excoriations, and that might lead to bacterial
Yes.
infection.
He has he is giving the history of 2 years of pruritus ani, and it was very worrisome for the patient, and he is very much relieved of that symptom actually now. Pardon, sir?
So Fournier's gangrene, in this case,
Yes.
actually, the patient was treated with a broad spectrum antibiotic. Though you
Yes,
have
sir.
isolated E. coli, Fournier's gangrene is a polymicrobial infection, and it is mostly predominantly anaerobes.
Yes, ma'am. But Piptas covers the anaerobes also.
Okay, but so E. coli may be only the commensal, which will also be present because of the stool spillage, as may perhaps be the cause for the enterobius also surviving very well in a good bed. Okay. Because what could come to the perianal area when the gangrene is there, it is a very rich site
flora
for it to feed
yes,
on.
ma'am. Yes.
So that could also be the reason. Anyway, it is a very good finding and a good presentation.
Thank you, ma'am. Thank you so much.
People should be on the lookout more for Fournier's gangrene, and usually Fournier's gangrene, 2 weeks getting discharged is also phenomenal one thing. So we have to look at that. Okay, whether it was
Actually, this gangrenous area was starting at the presentation, ma'am. It was not as a florid gangrene.
It was an early presentation, perhaps.
Yes, ma'am. Sure, ma'am.
More care was given to the patient. Since
He was
you could
very
go
much
and
willing.
take a sample from the patient, itself shows that some care was given. Otherwise, usually Fournier's gangrene patients are often neglected because they come in advanced stage. Yes, that's very good. And secondly, 2 weeks at discharge, you don't expect any scarring, isn't it? So that is another thing. So you expect scarring much later. But the fact that it healed is what you're trying to say, I think.
Yes, ma'am. Yes.
Point well taken. Okay. If there are any other questions or discussions, most welcome.
Ma'am, can I come in, please?
Yes, sir.
Please, sir.
I think I'm troubling a lot.
No, sir.
It's a pleasure. It's a pleasure.
It's a pleasure. It's good for all of us. It's a nice discussion that you're always spurring. Go ahead, sir.
Okay. Thank you so much, ma'am. Only actually, according to me, the patient improved. Maybe he might not be having any risk factors like diabetics or HIV or maybe some other underlying risk factor. That's why the patient improved. That could be the reason for that.
The patient was a diabetic, sir, because the fasting glucose he said was 168, and they put the patient on insulin.
Insulin, yes, sir.
Perhaps
So
the patient
even
was not treated early.
after getting the risk factor, the patient improved within 2 weeks is phenomenal. Maybe the resistance has not been started yet against the antibiotics. So that is a wonderful thing which you people have done. Another thing that you said that the pinworm itself has caused this excoriation. That is doubtful because the excoriation occurs because of constant itching which has happened in that area. And Fourier's gangrene is basically necrotizing type of fasciitis which affects the scrotum mostly. Okay.
Actually, sir, he was having a perianal abscess 2 weeks back.
Okay.
It was ruptured. And this area extended to the scrotum. Now it is having a necrotic area.
Okay, that's why you are saying it's a Fournier gangrene.
Yes, sir.
So it was basically a perianal abscess.
But it is now having a necrotic area over the scrotum, sir.
Scrotum itself. Okay.
Yes.
So maybe the constant itching which the patient is doing for a long period of time.
Yes, 2 years. Since 2 years, he is complaining of perianal itching.
The patient was very patient then, because I cannot sit for 5 minutes if some itching is happening in that
Yeah,
area.
yeah. But he was very, very happy after the treatment, sir. He was very,
Obviously,
very much
very
relieved of
happy.
the.
Yeah.
Yes.
What was the age of the patient?
He's 66 years, sir.
66 years. Okay.
Yes.
Okay. Nice, interesting case. Actually, some of the parasites which has been reported in the past is Paragenosis, Fournier gangrene. If you go and search for the literature,
Yes,
you
I
will
have
see-
seen one case, sir.
You have seen, yeah. Okay. So
Yeah.
Paragenosis has been associated with the Fournier gangrene. Actually, that is a disease of animals, zoonotic disease.
Yeah. Zoonotic. Pardon, sir?
Yeah, I can see you. Sir.
Pardon, sir?
No,
Yes,
sir
sir.
is actually on phone, I think.
Okay, thank you.
Okay.
Sir. Thank you.
Any more questions? A nice case study, madam. We'll go
Thank
for
you,
the next presenter.
Dr. Aradhana Singh, are you ready?
Yes, ma'am.
Yes.
Yes,
You
madam,
want
we can
to
present,
the
madam.
Ma'am, can you share the presentation or you want us to do it, ma'am?
Ma'am, I'm sharing. Just a moment. Is my screen visible?
Yes, ma'am.
Okay.
Yes, ma'am.
A very good
Visible,
evening.
madam. Yes.
Okay, sir, I'll start. A very good evening. Myself, Aradhana Singh, and the topic for my presentation is molecular epidemiology of pathogenic entamoeba species E. histolytica, dispar, moshkoski among different clinical outcomes of entamoeba infection. Starting with a bit of introduction. Entamoeba infection, they are ubiquitous in nature and have a major impact on millions of people worldwide, especially with low socioeconomic status. Among the different species of entamoeba which are found in human, entamoeba histolytica has long been accepted as the only pathogenic species. However, recent reports of other entamoeba species like entamoeba dispar, entamoeba moshkoski, and recently entamoeba bangladeshi in symptomatic cases has raised a question on their pathogenicity. Additionally, the prevalence and epidemiology data about the entamoeba case very earlier times and mainly based on microscopy, which cannot differentiate between the commonly found species. So with this background, the objective of our study was to determine the recent epidemiology of the pathogenic entamoeba species in different clinical outcomes. Coming to the methodology, a total of 473 stool samples and 125 liver aspirate samples were collected and then categorized as intestinal and extraintestinal and asymptomatic cases. Under the intestinal cases, different groups were made like the gastrointestinal discomfort patient, the retropositive case, patients having diarrhea. Under extraintestinal cases, we had only the cases from liver abscess, and in asymptomatic cases, the community population, these samples, they were subjected to microscopy using the iodine mount and saline mount, and then they were further subjected to nested multiplex PCR to screen the presence of entamoeba histolytica, dispar, and moshkoski in the samples. Coming on to the results, the microscopy, it showed the presence of entamoeba species in 5.4% of the stool samples and 6.4% of the liver aspirate sample. The figure 1, it shows the presence of trophozoite and the cyst in the different samples. Through nested multiplex PCR, the presence of entamoeba species was found in 6.6% of the intestinal cases, in which the dominant species was E. histolytica with 4.3% of the prevalence. In extraintestinal cases, we found only the presence of E. histolytica species with a prevalence rate of 86.4%. And in the asymptomatic population, we found the prevalence of entamoeba species in 10.5% with a dominance of E. dispar
in 5.6%. And when the microscopy and molecular method was compared, we found the sensitivity of the microscopy to be approximately 25% sensitive only. Now, from this study, we concluded that in this geographical region, the extraintestinal manifestation, they are more as compared to the intestinal infections of entamoeba, and E. histolytica is still only cause of invasive extraintestinal infections in this geographical region. And E. dispar was also found in the cases of the symptomatic intestinal infections. And this changing paradigm of the entamoeba infection can be better studied using the molecular methods. I'd like to acknowledge Banaras Hindu University and Department of Science and Technology for providing me the facility to conduct this study. And I'm very grateful to the patients and the community providing me the samples for this study. Thank you.
Thank you, madam. The molecular study was done for stool samples to distinguish between various species of the entamoeba, mainly the histolytica and dispar. I have a question, madam.
Yes,
You
sir.
told about extra-intestinal manifestation.
Yes, sir.
Common. In those cases, any dispar was found, madam? Or
No,
what was the
sir.
percentage?
No, sir. In the extraintestinal manifestation, we found only the presence of E. histolytica.
So still dispar is a non-pathogenic.
Sir, in our study, we concluded that only.
Yeah, some studies they have shown the pathogenicity.
Yes.
Yes, thank you very
Some
much.
research can have.
Can you hear me?
Yes, sir.
Yeah, thank you very much, Dr. Aradhana, for your nice paper on this entamoeba species. You have mentioned that this entamoeba dispar can cause symptomatic infection in the intestinal amoebiasis. Have you documented in your study?
Sorry, sir?
Did you document any case of entamoeba dispar from the symptomatic intestinal amoebiasis in your study?
Yes, sir. We have found the symptomatic cases, the intestinal from the GI discomfort patient in which the presence of E. dispar was found.
That is what I am telling you, because you see these symptomatic cases from where you have got entamoeba dispar,
Yes, sir.
you have ruled out other bacterial and viral causes. I think could be due to other causes, no? I understand that there are some symptomatic diarrhea, something like that were there, and you have isolated entamoeba dispar. But
Yes,
I
sir.
want to know how could you incriminate that this entamoeba dispar was the responsible for this syndrome of intestinal amoebiasis and not other pathogens, bacterial, all viral pathogens. Have you ruled out?
Sir, actually, we have the inclusion criteria for our study was to include such cases only, in which we have excluded any other associated factors.
Okay, thank you. Very interesting paper. Very nice. Well designed study. And do you have any idea of, did you not try for entamoeba Bangladeshi?
No, sir, we have not included in this one, but
Right, because entamoeba Bangladeshi being recently reported neighboring
yes, sir, it has been. Yes, sir, it has been recently reported, but we have not included it in this study.
Thank you very much. Very good paper. Thank you.
Thank you, sir. Thank you, sir.
Shall we go to the next presenter, ma'am?
Yes.
Sir?
Yes, it's a good, very good
Yes, madam.
study. We can move on to the next paper.
Yes, madam. Next presentation we can move on.
Dr. Saurabh. Dr. Saurabh, are you here?
Yeah, if it
Dr.
is,
Saurabh?
we can move on to next, madam. If the candidate is not ready.
Dr. Fayaz?
Okay.
Dr. Fayaz?
Yes, ma'am, I am in, ma'am.
Okay, sir. We will put the slides, so I present it, sir.
Yes, sir. You can present, sir.
Will you please share the slide?
Yes. 1 second, sir.
Dr. Fayaz, you can proceed right now.
Very good evening to everyone. Now I am going to present a case report about a rare disease, that is a scarabeiasis. An abstract: scarabeiasis is a rare ectoparasitic infection of the gastrointestinal tract caused by the beetle, commonly known as dung beetle. Actually, this beetle usually flies away from the anus while passing stools. Herewith, I am presenting a case of scarabeiasis in a 4-year-old boy. Case report. A 4-year from a village in Pudukottai, which is in Tamil Nadu, to our medical college gastrointestinal department, with a history of insect flying while defecation. There are several episodes had occurred during the past 1 year. Each time he was treated with albendazole, and he is found to be free from the episodes for at least 2 days. Then eventually he is having the symptoms again and again. The case has no history of any native treatment, and during the current episode, the stool sample containing the insect was collected. Actually, on examination, it was found to have alive and black colored beetle with yellow bands, 0.7 centimeter in size, and it is positively diagnosed and confirmed as scarabeiasis by an entomologist. The dung beetle is a scarab species, belong to the superfamily Scarabeeidae. It has 2 subfamilies, Scarabeinae and Aphodinae of the family Scarabeeidae, which is commonly known as scarab beetles. And on clinical examination, the child was found to be alert, active, not anemic, and there is no signs of malnutrition. Coming to the microscopic examination, and there was no evidence of ova, cyst, or any larva in the stool sample. And we have also done the colonoscopy, and it was found to be normal, and there is no insect, and the mucosa was found to be normal. The child was treated with the saline purgatives, oral metronidazole, along with the advice to use the tight underclothing, especially during sleeping and also while playing, to avoid contact with the cow dung. And the parents are also counseled regarding the personal hygiene. And coming to the conclusion, scarabeiasis is a temporary infestation of the digestive tract by the dung beetles. It is commonly known as scarabeiasis or canthriasis. Actually, diagnosis is usually done when beetles fly away from the anus during the time of defecation. The mode of entry, the most recent accepted theory is that the larval form of beetles enter through the anus, especially in naked children. It eventually developed into adult ones and fly out of the rectum. The most common presentation is usually the beetle flying away from the anus during defecation. Though scarabeiasis remains a rare entity, there are cases reported from several parts of our country. In Tamil Nadu, there are 2 cases reported, one from Tiruchi and another one is from Chennai. And also in Kolkata, also a case reported. And in Sri Lanka, there are some cases reported. I acknowledge Dr. Mr. Varadarajan, who is a professor in entomology, for identification of the beetle. Thank you.
Thank you, sir. A nice observation and a nice study. Actually, what was the treatment given for this child?
Actually, the treatment we have given only the metronidazole, antibiotic metronidazole, 3 times a day for 1 week, for 10 days. And we have given saline purgative.
Okay.
This was done. But the child, we are following the child, but it is reduced, but still he is having the beetles coming from his anus.
Yes, sir. Hope we cured of it. Any questions? Will any more presenter is there, madam?
One more.
Thank you, sir, for your good study.
Vishakha.
Dr. Saurabh alone has to present.
Yes, ma'am. No, one more Vishakha, madam is telling you.
First.
Already, madam, you have presented for one student, madam. So again, you cannot present.
Yes, madam. Already one presentation you have made. Madam, next.
She's trying to connect.
Next presenter, please connect. Organizers, connect the next presenter. Slides are visible. Please present, sir. Yes, go ahead.
Dr. Saurabh. Our present பண்றாரு.
Yes, he can present now. Dr. Saurabh can present.
Dr. Ravinder.
Yes, madam.
After this,
yes, ma'am.
We will go in for the validity. Okay.
Yes, madam.
Madam,
please.
Ma'am, am I visible?
We will go for validity after the presentation of these two papers.
Madam.
Okay.
Yes, ma'am. Am I visible?
Yes, sir. You can go ahead, sir.
My presentation is visible, ma'am.
Yes, yes, sir.
Yes,
Yes,
ma'am.
sir. It's clear, audible, visible. Please present.
My presentation is visible.
Yes, sir. Audible and visible. Please present.
Good
sir.
evening to everyone. Yeah. Good evening to everyone. I'm Dr. Saurabh Kadil from Krishna Institute of Medical Science, Secunderabad.
Yes, please proceed, sir.
Hello. My interstitial plasma cell pneumonia or pulmonary pneumocystis in post-COVID-19 pneumonia and retroviral disease positive patient. It's a case study. So, a brief introduction. Once considered to be a protozoan, and pneumocystis cerevisiae existed in cyst and trophozoite form, and now classified under fungus based on molecular typing. Pneumocystis pneumonia, that is PCP, is an opportunistic infection predominantly present in patients with impaired cell-mediated immunity, as in AIDS, when absolute CD4 count is less than 200 cells per microliter. Some other conditions that predisposes immunosuppression are child with nephrotic syndrome on steroid, ankylosing spondylitis patient on infliximab, and transplant patient. Pneumocystis have tropism, that is affinity towards lung, and infection is considered to be transmitted person to person by respiratory droplet or reactivation of latent infection. We received a 52-year-old lady with chief complaint of shortness of breath, fever, generalized malaise since 2 weeks, and she had history of LRTI COVID-19 pneumonia 15 days back, for which antivirals were given, that is remdesivir was given, and the steroid to subside cytokine storm were given. And on examination, she was in tachycardia 152 beats per minute with a blood pressure of 140/80, and she was febrile 99.6 degree Fahrenheit, and SpO2 was 100% with NIV. An examination, we found a bilateral coarse crepitation present in bilaterally in chest, and rest systematic examination was normal. She was provisionally diagnosed as type 1 respiratory failure with metabolic acidosis. And after careful clinical evaluation, she was admitted in ICU exclusive to COVID-19 patient, where detailed investigation was sent, and patient treated with 2-unit convalescent plasma transfusion, 200 ml each, IV meropenem, IV ticoplanin, IV steroid, and subcutaneous anticoagulants, and high flow oxygen mask. Now, investigation and result. On the day of admission, detailed investigation of the patient showed COVID RT-PCR negative, lymphopenia with relative neutrophilia in CBC, and all raised D-dimer, procalcitonin, CRP with hyponatremia. That indicates a poor prognosis of the patient. And chest X-ray indicated both hyla obscured and diffuse consolidation of bilateral lung field and pleural effusion. Also, CT test showed diffuse consolidation of bilateral lung field. And also blood culture negative after 36 to 48 hours, and sputum showed GPC and GNVs and without fungal elements, and ZN stain showed no AFP, and NT-HCV, IgG. In the mid-hospital course, WBC count, D-dimer, IL-6, ferritin, PCT, CRP normalized without much clinical and radiological improvement, with persistent lymphopenia and relative neutropenia. In the further course, patient diagnosed HIV-1 reactive. After then, absolute CD4 count is done. It became 6 cells per microliter, and HIV viral load came 15600 international unit per ml. So after that, we used induced sputum following mucolysis and centrifugation was investigated for AFB and PCP. And for pneumocystis, we cyst observation, we done 3 type of staining: toluidine O staining that visualized the blue colored cyst of pneumocystis that I have put the picture, figure 1 I have taken. And next is GMS, gomerin methamine silver staining. And cyst seen as a black colored crushed ping pong ball against green background. This is shown in the figure 2. And also I did Giemsa staining, for which internal nuclei of cyst up to 8 in number are seen, that is under 100x high power. And on sudden staining, no AFP was seen. After that, we treated the patient after being HIV tested positive and PCP. Patient treated with tablet dolutegravir integrase inhibitor, tablet lamivudine or zidovudine, and injection fluconazole and cotrimoxazole 4 ampoules TDS. And also for Mycobacterium avium complex prophylaxis, tablet azithromycin 1 gram per week for 3 months was given. And following that treatment, clinical and radiological improvement was seen with decreased oxygen requirement and hemodynamic stability of the patient. So eventually patient is shifted to ward and discharged after 20 days of hospitalization, and after explaining all the warning sign and follow-up. So after this whole case, we conclude that PCP and COVID-19 shares numerous characteristics as both diseases presented with similar clinical feature, radiological findings, and blood findings. HIV and PCP were missed initially, as PCP with HIV is less commonly to antiretroviral therapy. This highlights the importance of considering HIV and PCP as differential diagnosis in an immunocompromised patient. So here I rest my case. Thank you.
Thank you, sir. It's a good study. If any questions are there, we'll answer it shortly. We'll go. Organizers, is it over?
Yes, sir. Yes, sir.
Hope all the papers
That
are
was a
over.
wonderful array of presentation.
Still it is there.
Ma'am. And Dr. Ravinder for joining. Roshi and Ravinder, they can conclude the session. Roshi and Ravinder, Dr. Roshi and Ravinder.
Yes, madam. Yes, ma'am.
You can just conclude.
Yeah, it was a good presentation with extensive studies, thematic poster, as well as the original article. And I thank the organizers for giving me this opportunity. I thank Geeta Lakshmi madam, Dr. Parija, Dr. Kalavati madam, and the whole team. And also I thank Rosie madam for being with me and sharing the dais in this presentation. Thank you, one and all.
I second Dr. Ravindran's comments on the good profile and standards that were exhibited throughout the 2 days of this session. And it was a great privilege for me to sit through these paper and poster presentations. I thank Dr. Ravindran for being a wonderful co-chair with me, and we are at it. So we just join you shortly. Thank you very much.
Thank you, Roshi.
Thank you, madam.
Thank you, madam. Thank you.
That was a wonderful array of presentation. Thank you so much, judges. Prizes will be announced at the end of the valedictory ceremony. Now we are in the penultimate session. Over to our VC, ma'am.
Thank you. I thank you for all the delegates, participants, and Dr. Ravi who are on the online. Dr. Saurabh and Dr. Rakesh is here with us. Thank you, Dr. Rakesh, being with us. And I think we will be having the valedictory function right now, and the announcement of prizes would be given by our president of the Indian Academy of Tropical Parasitology, and still more he has lots to say. And with this note, I will just hand over to Dr. Parija. Dr. Parija?
Yes, yes. We'll start now. Started.
You can start now.
Yeah, you want me to speak now?
Yeah.
Yes.
And what about Subbalakshmi, about the prices? Have you all decided and have you sent to Dr. Parija?
Ma'am, Rosie Venila ma'am and Ravindra sir will announce. They have to announce.
Okay, over to Dr. Parija.
Yes, ma'am.
Hello.
Dr. Parija, it was an amazing conference. I should say that we had been toiling within a matter of 15 days with my IT department, and the experience given to me and to my team really was a wonderful retreat for us. I would say it's not work. We thought we were busy and we enjoyed this moment given by you to us, and it was a challenge for me and for my team. And really, I thank again Dr. Parija and the Almighty who have given me the courage to go ahead with this national conference, 14th national conference. And today, my team, and I should say that everything is possible. That's what in only one word I can say. Everything is possible. You have to have the heart and mind into it. That's the only thing we should do. And for the youngsters to go ahead and never think about the outcome, and just go ahead with whatever you have in your hand. And now I will introduce my whole team. And Dr. Kalavati Victor, really, she's a wonderful person. I have been for more than 20 years, I've been there, 25 years I should say, and a wonderful person to move with and to have a friendship with. And immediately, age is not a factor at all for anybody, and she is one of the examples of that. And Dr. I should appreciate her who took the challenge immediately when I just called her to my room and said that Dr. Parija wants to do this 14th National Conference as E-TROPACON. And she immediately obliged and said, Madam, we can do it. So age is not a factor at all. She is still young with us. Dr. Kalavati, can you remove your mask? And she's such a great person. I should appreciate her. And she has a head of the Department of Microbiology. And we were colleagues in Madras Medical College. And doing a PG also postgraduate. I was there at Madras Medical College. We were working together. And now she is head of the department and the principal at the ACS Medical College. When she has given a very good lead and for the whole team. Thank you, Dr. Kalavati. She is Dr. Kalavati. Dr. Kalavati, yeah. Dr. Parija, she is Dr. Kalavati.
Yes. Thank you.
Right. The next one.
Yeah. Thank you.
Dr. Subbalakshmi, take your mask and remove your mask, and can you sit on the
Sir, Parija sir. Parija sir, can you hear me?
Yes, yes. You want me to speak now?
No, I'll talk, sir. So thank you for giving us the opportunity to conduct, all because of the guidance from our VC, Dr. Geeta Lakshmi.
Thank you.
From the scratch, he helped us to do, and we are able to finish it successfully, sir. Hope you all will appreciate our team.
Thank you very much.
Thank you, sir. Thank you.
Appreciate their team.
So, can I now speak now?
Yeah,
no,
Yes. Okay.
Subbalakshmi, can you put the camera on your, over to you? Subbalakshmi. Yeah, she is Dr. Subbalakshmi. professor in the Department of Microbiology, a very enthusiastic, really the wavelength was so good that she understood each and every movement of the from the top to the bottom, to the registered delegates, to the speakers, to the resource person, and really appreciate. I mean, she could understand what we wanted to do without a small, even though there were a little bit of changes, we could manage with her help and Dr. Kalavati's help. And she was only to say every early morning at 4:30 also she will just send a message once. Because I get up very early, she also says, give me a message, madam, this is going on today. And this is what is the program, I mean, what has been happening the whole day, because previous night I didn't have that. Thank you, Dr. Subbalakshmi, for your enthusiasm and zeal to conduct this conference. I really appreciate you.
Thank you very much, ma'am. Thank you. Thank you, sir. Thank you for your kind words.
Yeah, thank you. So next, Dr. Balasubramaniam is there?
Ma'am, he has left, ma'am.
Right. The next person was Dr. Balasubramaniam. He is a professor of ENT, but he is not here. He has left. And he was a great, he is a computer engineer, ENT surgeon, and what not. He knows everything. He was working with me for last 20 years, where I brought him here as an additional registrar of this university. And he had been toiling with the whole team to make this event successful. So thank you, Dr. Balasubramaniam. Next is Dr. Kalpana. She is a professor there. She is here. Dr. Kalpana. She is she is a professor and she is my student, and they are all really enthusiastic personals. I think so, handpicked and chosen in our medical college. Thank you, Kalpana, for your wonderful job.
Thank you so much, ma'am.
She was an ardent student of me.
Thank you.
Thank you. Next person, is any other person here? Along with you, your team? Any other person in the team there?
Madam, Dr. Anandi. Madam, Dr. Anandi.
Anandi. Where is Anandi?
Madam, she is there.
Dr. Anandi, she is Dr. Anandi. Raise your hand. Dr. Anandi. Right. She is Dr. Anandi, one of the good, and she is also my student who had been in the scene behind the whole success of this conference. Thank you, Anandi. Next one.
Thank you, ma'am.
Next.
Madam, Dr. Sukanya is there.
Next.
Madam, Dr. Sukanya is there. Dr. Sangeeta is there. Sophia is there. Say hello, ma'am.
Just please come in, because I want to see all. Dr. Suganya is there, who has been coordinating. I think so. You would have seen her speaking with all the resource person and all things she was doing. It is Dr. Suganya. Thank you, Suganya. Dr. Parijat is Dr. Suganya.
Dr. Sangeeta is there.
Right, Sangeeta. Dr. Sangeeta, thank you, Sangeeta, for coordinating with the whole team and making this the grand success, this conference. Next.
Sophia is there.
Yarik?
Sophia.
Sophia, thank you, Sophia, for being here with us. She is also in the department as associate. Enna work ranga?
All my teams are there. Show your face. All of you show your face to me. They all did wonderful job, madam.
Yes.
Yeah.
Right. Thank you, all of you. Thank you, all my postgraduates. Did a wonderful job. Take off your mask and just we want to see you. Take off your mask. Right. Thank you. So this is what the whole team and my IT. I think so. She's Dr. Mrs. Kanya and Saravanan is there.
Saravanan is there. Yeah. Why not?
Saravanan.
Yes, ma'am. Can I see, ma'am?
Mr. Saravanan. A known person in the whole IT team. But for him, nothing would have happened with all the technical problems, everything he was doing in time. And he is also the main technical person in our university who is dealing with everything regarding IT in our university. Thank you, Saravanan.
Thank you,
Dr. Parija is Dr. Saravanan. He is
Thank you.
Mr.
Thanks,
Saravanan.
Yes,
Thank
to
you,
be given
Saravanan.
big applause, madam, because he has been there for the last 2 days.
Yeah, he's 40 years has been here in the medical college rather than being in his own department. Thank you, Saravanan.
Thank you, ma'am. Thanks, ma'am.
Yeah.
With pleasure, ma'am.
So I'm over to Parija for the event for the valedictory address.
Good evening, and respected Professor Gita Lakshmi, Vice Chancellor of the University, and all his team members. Professor Kalavani, I think we met in that 2007 also Chennai conference, and Dr. Shiva Lakshmi and other members of the team, and my esteemed colleague Professor Segal, Ravindran, and other people, and all the participants. I take this opportunity to thank each and every one of you, in first and foremost being the most dynamic lady, Professor Gita Lakshmi. I suggested, I requested, and she with a strong team has made it possible to host E-TROPACON 2020 in the virtual mode. I congratulate her, and I congratulate her entire team, all the members of the team, for making a strong effort for hosting this excellent conference. Academically, it was a very nice experience. We had interesting paper on the e-posters. We had interesting oral presentations. Above all, we had eminent speakers from different parts of the country and abroad to share their experience of zoonotic parasitic disease. It was really an academic society, and even I have learned a lot during these 2 days of conference. I thank each and everyone. Again, particularly, I must thank Dr. Shiva Lakshmi, because she was the coordinator with whom I was talking a lot. And I must, she was very systematic, and the way she was handling and coordinating with me and Dr. Gita Lakshmi, two different people at two different places, and giving shape to a program. Really, congratulations. I congratulate Dr. Shiva Lakshmi for your nice job of organizing.
Thank you, sir. Thank you.
I also take this opportunity in thanking my coordinator, Dr. Namrata, who was behind this scene and was trying to coordinate. I must thank her. Again, I was so happy to see Dr. Kalavani here. I remember with pleasure. We met first time when Dr. Gita organized the first conference of the E-TROPACON 2007 in Chennai. I remember you, and I'm so happy that you are still with Dr. Gita Lakshmi and has given the same strong helping hands to Dr. Gita Lakshmi for organizing this conference. I am confident such conference will motivate our young people. This will make an opportunity for all senior people to know the young upcoming parasitologists, and I hope this IATP will provide a good platform for all young people and experienced people to interact and promote the activities of the parasitic disease. We are moving forward in the right direction. I hope we have more meeting in future in exemplary way. Again, last but not least, on behalf of Indian Academy of Tropical Parasitology, on my behalf, I wish to record my sincere thanks, our sincere thanks to the organizing chairman, Gita Lakshmi, and our team for hosting such a wonderful, nice conference at Chennai. Thank you. Thank you very much.
And they need an appreciation letter for all of them from
yes, I forgot. I promised within a week or two, I sent certificate of merit and appreciation letter to all of you, and I'm waiting for that list of the names from Dr. Geeta Lakshmi. It will be my pleasure, and this really will be privileged to issue such certificates of appreciation to each and everyone.
So there are a number of participants also went up. It's really a breakthrough in the conference that there are more than 800 participants for delegation here, and it's really breakthrough. And the next one is also many of them wanted to become members, so we uploaded it in the YouTube chat so that everybody can make membership in our Indian Academy of Tropical Parasitology. Not able to hear. So can you hear me? Yeah.
Yeah, that's absolutely.
Kashinath is here, and Dr. Ravindra is here, staying with us, and thank you, Kashinath, being here with us, and Ravindra being staying with the constant support from him from outside. Thank you, Ravi, and Dr.
Thank you, madam.
So we will be ending this session with a very good virtual feast in our mind. Really, I myself wonder, like, it was such a good event which had happened, and which can happen in future also. And I want Dr. Kalarati to give a vote of thanks.
Before that, I wish you all the best for happy and prosperous New Year 2021.
Yes, wish you a happy New Year. Kalarati, over to you.
Prizes. Oral presentation. Poster prizes.
Hello, Dr. Ravindra.
Yes, madam.
First and second prize, doctor.
Yes, madam.
You have the list. You have the list, please.
Yes, madam. For the oral presentation, second prize will be Jaya Banerjee, Krishna Institute of Medical Sciences, and first prize will be Dr. V. Stanley Medical College. For the posters, second prize will be Dr. Sashwati Chattopada, Bardhaman Medical College. First prize will be Aradhana Singh, Banaras Hindu University. Congratulations all the winners. Thank you, madam.
Thank you, Dr. Ravindra. Thank you.
Thank you, madam. Thank you.
For sparing your time. Okay, thank you.
Thank you very much, madam, for giving me this opportunity.
Or Subbalakshmi, anyone. Dr. Subbalakshmi or Subbalakshmi, can you give vote of thanks?
Yes, yes. Good evening, all. I take immense pleasure, and I'm.
Yes.
On behalf of the organizing committee, I extend my heartfelt thanks to the Honorable Founder Chancellor of Dr. M.G.R. Educational Research Institute, to Dr. A.C. Shanmugam, a renowned entrepreneur and educationalist. I would like to thank our young, dynamic, Honorable President, Engineer Through A.C. Arun Kumar Sir. I take this opportunity to thank our Honorable Secretary Through A. Ravi Kumar Sir for his constant presence and encouragement. I would be failing in my duties if I did not thank Dr. Geeta Lakshmi, Vice Chancellor of Dr. M.G.R. Educational Research Institute and the organizing chairman of TROPACON 2020. She is the biggest pillar of our support till all along the way from the beginning till today. I extend my sincere thanks to Dr. Subhash Chandra Parija, Vice Chancellor of Sri Balaji Vidyapith University and President of IATP, and all the office bearers of the IATP for giving this opportunity to conduct the national conference and also helping out throughout for organizing this conference. I thank our registrar, Dr. C. Palnivelu, our beloved Dean, Dr. Pragna Dolia, our special officer, Dr. Dhanavel, retired IAS officer and advisor, Dr. Jayachandran, and our medical director, Dr. Shah Nawaz Khan, and our medical superintendent, Dr. Palani, who have all been extremely supportive throughout, irrespective of the time and situation. I take this opportunity to thank the man behind the scene, or additional registrar, Dr. Balasubramaniam, who burnt his midnight oil, helping us with all this platform he created for us to conduct the conference without any disturbances until today. I also thank Dr. Hariharan, Principal Pharmacy,
for his support to initiate this conference. And I extend my sincere thanks to Dr. Kanya, HOD of the IT department of our university, and Mr. Saravanan from IT department of our university. He has been there throughout the conference, rendering his constant support. I also thank Mr. Raju, who belongs to IT department of ACS Medical College, for his constant support in sorting out all the technical issues now and then. I also thank our event coordinators, Mr. Bharati Kannan and Mr. Kribakaran, for their support. I extend my heartfelt gratitude to all the speakers, chairpersons, panelists, and judges who are the biggest reasons for the support of this event. We thank all the heroes of the event, the delegates, and the presenters who have made this event very interesting through their enthusiastic participation. Last but definitely not the least, I thank all the faculty and postgraduates of the Department of Microbiology, that is my department, and other coordinators who also have worked day and night to organize this conference in timely and orderly manner. Thank you one and all. Thank you, the IATP President, Vice President, and all the members. Thank you, sir.
Thank you so much, ma'am. Thank you so much, ma'am, from the bottom of our hearts. We request you all to rise for the national anthem. Signing off, marking a memorable end to E-TROPACON 2020.
Okay, thank you everyone. Thank you, sir. Happy New Year. Happy New Year to everyone.
Happy New Year to everyone. Thank you very
sir.
much for nicely organizing the conference.
Thank you, sir. Thank you.
Thank you. Thank you.
Thank you, everyone, and a very happy New Year, and meet again in the next conference.
Definitely, sir. Happy New Year, Saigal, sir. Happy New Year to you also.
Thank you very much, and same to you, all of you.
Thank you, sir. Thank you. Thank you, sir. Thank you.
Okay, thank you.